Chandu. Yeah. Good morning, all, and welcome to the second day of this conference. We are starting with now scientific program of the day, and I would like to invite Dr. Pranjali to introduce our speaker and invite the chair and co-chair. Okay. So I invite Dr. Harinarayana Rao, preclinical consultant, Hyderabad, as a co-chair. And then as a chair, and Dr. Gopal Krishna from TIFR as a co-chair to head the session. I would like to invite Dr. Satish Kumar sir. He's an ex-chief scientist and group leader, Center for Cellular and Molecular Biology, Hyderabad, to deliver his talk. Thank you, sir.
Okay. Good morning. It's a pleasure to be here, and particularly I'm thankful to Mahesh and Jayant to give me this opportunity to meet old friends. And let me say in the outset, the empty chair doesn't show my popularity or lack of it, but it's Harinarayan Rao, because he's the chair. So for this morning, what I will do is, as the title suggests, I will take you through some of the stuff which may at times appear trivial, but at times a little bit of science. So just I don't know how many of us are aware of this. Mendel would have been the first mouse geneticist. Is there anybody in the audience who knows that? He started his experiments not on peas, but on mouse. And his interest was coat color. And particularly encouraging for those I gather that many of you are the facility in charge of the animal houses or facilities. And most of the time you must be facing this dichotomy of your directors or bosses telling you not to do science, right? So you are not alone there. In fact, Mendel was stopped by his boss, Bishop Anton Ernst Schaubgosch, not to do mouse genetics. And the reasoning was fantastic. And the bishop said that the mice are going to have sex and how he can allow something like that in the church. But the poor bishop did not know that even the peas are going to have sex, and that's to an extreme form of sex, that is incest, because self-pollination. The great guys are always great. In fact, we should not be really feel powerful. It was Haldane in 1928 in a very popular lecture, sort of paper that was on being the right size, that was the book. He wrote this, that to interfere in a single gene without interfering the others, it's a real challenge. So that means that nothing new. Even in those days, these guys were thinking about these things. That's in 1928, and it took about 60 years, 61 years in 1989 when the first mouse gene was knocked out, and it got published in PNAS and Cell simultaneously. With this little bit of history, what I'm going to do today is that I will talk around a couple of these things, primarily based on the experiments which my group did at CCMB. So if some of these things they appear outdated, that tells my age, nothing else. So genetically modified, is there color mic? Because I have to be on each side. Genetically modified laboratory animals, as we all know, gene regulation, gene function, creation of animal models of human diseases, what you call lately humanizing mouse, or mouse here, I mean, when I talk about mouse, that I also mean other laboratory animals. And we will see this genome editing things have made that blurring much more, whether we talk rat, mouse, or whatever species. And of course, discovery and validation of drug targets and drug developments in pharma, development of gene therapy protocols, and laboratory animals for veterinary livestock biotechnology systems wherever you want to use. So you use these laboratory animals. I will pick up a couple of examples from these, and then I'll go ahead with that. Now, thank you. Should be okay. You can switch that off. Hello. 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 I hope it's audible. Sorry for the interruption. Now, when we say of transgenic animals, or for that matter, whatever transgenesis, essentially it comes from the property of the cell, that's DNA repair. Somehow the eukaryotic DNA, particularly even again, including mammals, don't like the loose ends of DNA. So the moment there is a loose end of DNA, the cell has to deal with it. And the dealing with, there are different ways of dealing with it. That is, that non-homologous end joining, that whatever that comes on the way, stitch it together, and or homologous, that you find the homologs and then change, or some kind of excision repair if there is a damage from UV lights or other insult to the DNA. So that's basically, that's the basis for making the transgenic animals or transgenic organisms. And that's what, so we have not done anything new. That's what we, the scientists, have taken the help of the DNA repair mechanisms, which are already available in the cell. Now, the whole story starts with this gentleman, Rudolf Jinisch. He showed that if you take the viral, some of these viral sequences, put them into a blastocyst, they can get into the genome, and then you can get the mouse on the other end. So this was the first, I showed for the first time that you can make what is called transgenic animals. And of course, you have seen this figure a million times, that the transgenic mouse expressing human growth hormone genes effect, not only that there is the gene is transferred, but there is an effect on the phenotype, and there is this guy is containing that gene. Now, standard techniques, again, this is a textbook thing, is that you introduce the DNA and then transfer to the pseudo-pregnant female, the blastocyst, and then you go back, and some of these animals will carry that transgene. That's a standard technique, and we will come back to that. But though it is very, very old, that is almost 42 years old, but this has become very, very relevant again in the context of what we think today is, or what we call is genome editing. Now, this example I have picked up, this was the least being that this gentleman was my supervisor, PhD supervisor. This paper was a landmark in many ways. A sheep gene was transferred to the mouse genome, and 50% of the milk protein was nothing but the sheep. And that demonstrated that really, really you can do wonders, that can be a phenotypic effect. And not only that, you don't require anything. The mouse recognizes all the addresses which comes from the sheep with millions of years of difference in evolution, and nothing else is needed. You get that whole gene. No manipulation was done. It was a sheep 16 kb sequence put into the mouse. And this gentleman used to tell me that this paper took them 3 weeks in total, and the 3 weeks was the time for the mouse to get pregnant and deliver the babies. So that was the quickest project ever done by this guy. In fact, this appeared in Tomorrow's World BBC program in those days. So that's, now I will quickly go through this. One example which I picked up from the work which was done by colleagues at CCMB, me included, of course. This example I'm showing that you may be aware that they are egg-laying mammals, what we call echidna, and then again some couple of hundreds of millions of years difference between mouse and this echidna. So these animals, it turns out that these animals have got we identified one cDNA into the milk of these animals. These are egg-laying mammals, and we did a lot of molecular biology and the stuff, and took that gene, put them into, first we showed in the cells that indeed this gene or this protein, which we called ECHEMP, shows antimicrobial properties. And this is again, when you make this protein in the E. coli, again, it interacts and works against so many organisms and its antimicrobial properties. And more importantly, we made this transgene and made a transgenic mice, beta casein promoter, and then this is what really interesting, that if we take the milk, and this is what happens, and I will not get into the details of this, wherever you are seeing everything clean, that's a milk coming from the transgenic animal expressing this gene, which has come from Echidna, some more than 200 million years away from, and this gene doesn't exist in extant mammals. This is in vivo experiments where the animal was challenged with the organism, the mouse was challenged, and then again, seeing that wherever again you are seeing clean plates, that means that this protein is working. Unfortunately, the level of expression was very low, but fortunately, still we could see the phenotype interestingly. So this is one example, again, going through the mammary gland sections of these transgenic animals expressing. The point here is that when you have these animals which are treated with this protein or having this protein, their mammary gland is clean. These are the wild types where the mammary gland has got these all symptoms of infection. Now, let's move on further. So people were making this 82 was the first time when piece of DNA was put in, and then animal was made, and millions of, I would say, the animals could have been made throughout the world in animal houses. Whatever we understand about the gene regulation in mammals, thanks to the mouse, that how a gene works, which is the promoter, what elements are needed, this all animals made, and whatever knowledge has come from there. This knowledge was also used for creation of disease, animals, models of human diseases. Like, for example, hepatitis C virus was introduced and then produced, and there are so many examples, plenty of them. What was a changing scenario that, in fact, in 2007, fast forward, these three gentlemen were awarded Nobel Prize, and some of you would have met those who are old, like Hari, or seen this guy in Hyderabad. Martin Evans, he came to CCMB in Hyderabad, and incidentally, he happened to be my external examiner at that time for the PhD thesis. These guys, they showed, the challenge was, and this is particularly a younger student I need to be impressing upon, the challenge, what was the challenge? That you put the DNA into a cell or egg, the DNA gets stitched randomly anywhere in the genome. So that means what you need is a piece of DNA, which you know what you want to do, and a cell or an embryo which can produce you the animal. There were two aspects of it, and that's where the whole transgenes were based on. The challenge was that how to target a gene directly into the genome, that I don't want the random integration, but I want to change a gene which is endogenous sitting on the chromosome. Now, for that, now it may look trivial, but it was not a trivial exercise. This gentleman came out with what we call embryonic stem cells. In 81 was the first paper, that you can have these cells which are pluripotent, and these cells you can use them to make animals out of them, and you can grow them in culture like as you grow bacteria and get the whole animal out of it. These two gentlemen simultaneously, Mario Capecchi, Oliver Smithis, they were trying to develop the protocols how you can target a gene on the chromosome, because the problem was that anytime you put any piece of DNA in, it will go and randomly integrate. Not rather than going for homologous recombination, because in case of mammals and most of the eukaryotes, homologous recombination is rare, random integration is the rule. So that was the real challenge which these guys solved, and we will see one or two examples. So they were nicely, they were awarded for the Nobel Prize, obviously. And by the way, that reminds me, Oliver Smith's first paper as a PhD student was in 1955, and that was starch gel electrophoresis. So that means that no surprise that at the end of some couple of decades or several decades, he gets a Nobel Prize for gene targeting. And the paper which made that, there is the Indian connection. Kucherlapatti was the first author of that paper in Nature. Kucherlapatti et al., Oliver Smith's lab, that experiments were done when they showed that indeed you can target a gene on the chromosome. This is the risk you run when you invite old people to talk. There will be always history. Anyway, so this what this Martin Evans did, that you have got these ES cells, and when you put them back into a blastocyst, there's a composite embryo, you get chimera, and extensively they can contribute. The yellow color is coming from these ES cells, black color is the host embryo, and when you breed them through, you get these chimera that can produce the next generation. And the trick is that if you are able to manipulate these cells in vitro, when they are growing, then you can get the animal, which will be different from the what you started with. So that will be very precise. That's where the word came, gene targeting. And interestingly, again, that reminds me to Martin Evans did not get the Nobel Prize for embryonic stem cells for whatever discovery he made. He got the Nobel Prize for gene targeting, which most of his gene targeting experiments failed later on in his life. But then that's the irony of life, that he got it for ES cells, not for, and there were, anybody can discuss with me the couple of, there were international politics there. As usual, science is not free from that. So what do? You get the ES cells and go through this homologous recombination. This was Mario Capecchi's idea, that you can have this, without again getting into detail, if this is your targeting vector, this is your normal chromosome, you do going for homologous recombination. This kind of arrangement will discourage random integration, and then you can select against random integrates. And sometime you could, with luck, you could find out of 100, 200, 300 clones, one ES cell clone which has got the right mutation, what you wanted, and then you can go ahead and make the animal out of it. So this is what essentially meant was this change. Using these kind of things, there's again these examples I'm putting because these slides are readily available with me, that many, many years ago, we knocked out one gene, 94, that was beta casein, that one of the milk protein genes, and then we showed that nothing happens basically. It was nothing exciting except the size of the micelle went down. And then again at CCMB working, then we showed that indeed, if you remove kappa casein, this gene turned out to be, and very exciting, that it was essential for lactation. Remove the gene and there's no lactation. So this is regarding that what you can achieve using these type of technology, that understanding gene functions. Now, coming back to the models of this another example again from the CCMB work. There is a gene WDR13, and when we knocked it out, it turned out that the insulin level goes up, and the reason for that, these cells which are producing insulin, they proliferate, and then you get phenotypic effect on this. And indeed, in fact, you will see this kind of picture again today in the presentation. Sachin may be talking about it. What we did, DBDB, that's a typical model of diabetes. We put that mutation into this, and it turned out that this animal double knockout survives nicely, while in case of the DBDB homozygote doesn't survive more than 4 months or something. But in this case, we could demonstrate that yes, this gene, knocking out this gene helps in there. And this is to give you a little bit of data that the fasting glucose levels, they are maintained in the double knockouts. These are the double knockout animals that they are, and this is the DBDB, and similarly fed glucose, they are maintained, and there's a phenotypic effect. Now, this is again the summary, that whatever I have spoken about is that knocking out this gene helps. Indirectly, what that means is that this protein can be a target for finding some drugs that you can hit this gene and lower the concentrations, and maybe that can be helpful. Okay. This is very, very interesting. Somebody did an exercise. He took the best selling drugs, this 100 best selling drugs which are available for human, and then went back to the target. Now, retrospectively, that this drug, where is the target in the protein that is it's, and then looked for the phenotype, and it turned out there was 100% match. That if the technology would have existed, if these guys would have knocked out the genes, they would have gone back and found the target for these drugs. I'm not saying that drugs would have been discovered, but this target would have been discovered. So that's the way, the powerful, this fantastic review in Nature Genetics. Now that brings me to the next level, and gene targeting or changing the making the mouse through embryonic stem cells was a TD process, chance factor, and generally, if you are a well-established lab, then you'll take some 2 to 3 years to get to the mouse and start doing the phenotype. And finally, it may turn out, if I can paraphrase in Hindi, pahad khoda nikli chuya aur wo bhi mari nahi. So if your mouse was not dead at the end of 3 years, then you were depressed because there is no phenotype and you struggle hard. Okay. Parallelly, people knew it. Again, there is nothing exciting. People knew it. That if we can introduce these changes in the genome, that we can introduce breaks in the genome, then homologous combination will be preferred. Okay, I'm getting a signal. So this is again the talons and then these zinc fingers. Using now came the Crisprs. Just the take-home message I want to give is simple, and that is that using RNA and an enzyme, you can introduce a targeted break in the genome, and then homologous recombination will become the rule. And that brings me back to that slide of microinjection into the eggs, that you put these into the egg, instead of going through the ESL route, you can now make directly an animal which will be knocked out using this CRISPR. And that's where there's a lot of excitement all these years about the CRISPR technology, that the difference it has made: one, you don't require ESL route; two, directly go to the eggs. And that also means that you can work on species where there are no ESLs. And hardly there are any species other than mouse and human, there are ESLs which can be used. So that was and these are again the same thing, is going through that superovulation and directly inject and get the genome edited progeny, then 15% of your animals will be all right, what you're looking for. This is, we can skip, and I'm last 1 or 2 slides, and somebody is saying time is up. So this, where again, in one of these experiments at CCMB, what we did, one of the casein locus, milk proteins, and I've been interested in milk proteins, we wanted to remove some 140 base pair of DNA, and we did that in a targeted way through going through the CRISPRs. And again, we are able to understand the function of this. Now, to conclude a couple of things, I will just, I want intelligence from you now specifically. Too much precision is bad, okay? On one hand, it is good, but on the other hand, it is bad. One is that you are going to do away with serendipity. In biology, most of the discoveries, they happen because of the serendipity rather than being too much precision. Second, remember that if you are going precise, if you are going to discover a drug, that will be only specific context, not something generally. Next, humans are not inbred, fortunately. Most of the time, we work on the inbred strain. Third, human are human and mouse is mouse. Okay? So each time when we are interpreting, we'll have to be very, 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 very concerned about it. And finally, your animal house, and I assume there are a lot of people who are in the animal house facilities, how the animals are kept, that will determine your phenotype. It's extremely, extremely very important how you are managing it. That will determine your phenotype. So one has to be careful. And finally, this again, I was told that somebody will be speaking about these kind of transgenic mice. Biosafety is a real issue when we are talking of these kind of things. There is I don't know there's evidence or not, but one of the reasons that they said Chinese, always there was this COVID coming from bats or pangolin or human directly, very transient, or at least I don't have any evidence, but what I suspect is these guys had something like that in their transgenic facility, and fantastic. This can jump very easily and would have been incubated, and you can mutate or whatever can happen. So one has to be extremely, extremely careful about the animal facilities. And when we are talking of precision, and this is what I do these days, by the way. This is, those of you who have not seen Kashmir, this is Kashmir. And this is my final slide. Just if I can, I don't know whether I can have something here, but that's a shame that we can't have it. That's where I finish here, and I am ready to take any comments or. And thanks. Now I think I can see it now. It was Sachin did something. Yep. Play. I think it's taking time. It's okay. If it works, it's okay. It doesn't work, it's okay. Fine. Okay. Sorry. Oh, it's okay. It's okay. It's not very important. It was more for fun. Yep, that's it. So this is just 3 kilometers away from Pakistan border. If you happen to go to Kashmir, don't go to Gulmarg, Sonmarg, go to this place called Karen, Karen. So it's a wonderful place. Other side of the river bank is Pakistan, this side India. You can shout between.
Thanks, Satish, for a wonderful presentation, taking us through the journey of genetic manipulations, right from random mutations to gene editing. As usual, you are a superb speaker. Thanks. And then number 2, next time count with me and I will be along with you to that place. We sail in the same
Sure.
boat now.
Yeah.
We'll have fun.
Yeah, I'll put you in front of me to escape from
In
the
case.
bullets.
We'll do that. Thanks a lot. Any questions, please?
Yes, please.
This kind of gene therapy and gene editing, all these things.
Can you
Well-
can you please take the mask so that I will be able to hear you better slightly? Yeah.
Sorry, I am Ravindran. I am a professor of pharmacology in Jipper, Pondicherry. This kind of gene therapy, gene editing and all these things, well, they were touted to be, very good approach and all these things. But then when you see, they failed. They didn't really work. Even though the gene therapy is given for 1 or 2 diseases, even then, no, it's not very effective. So what do you think about this? Technology is good. I mean, no, it is working in
See,
mice and mouse
okay.
and
If I
animals.
have
But when it
got
comes
your,
to disease,
Yeah.
patients, it is not really working well.
okay, all right. If I get your question, is that the transforming this research to translation level in terms of the gene therapy, the success rates have been poor. Okay, these when we come to, okay, I'm not going to oversell you the technologies or whatever. If you have to work out protocols, human is the last species where you can do the protocols. You will have to start with the laboratory animals. Now, failure from animal models to the human, there are so many reasons for that. And one of the reasons, I'm not an expert in gene therapy, but one of the reasons as a layman what I can think of is that it's the delivery systems. Most of the time, it's the delivery system that how you deliver things to the human that many times we are unable to sort it out. Okay. But one thing that, again, it's my personal bias that may work nicely is that you have human cells, make changes in the cells and use those cells in the therapy. That will have more promise than any kind of thing that targeted sort of doing it, changing something, the lung cancer or whatever, liver disease or something. So, but it's the problem is different. So it's nothing to do with the animal protocols or these techniques working in animals. And there is no substitute for that. Right? Is it okay? Okay. Yep.
Once they sort out the delivery system that survives, like many people died, adenovirus associated something, but we have not yet arrived. How to take this to the human cell?
And of course, the alternate, which is again a dream, is that everybody will have their own ES cells, and you can have differentiation of them, or you make changes in ES cells of your own ES cells, make the cells type what you want to replace, and then use it. But of course, part of this is lost. A lot of it is fiction, but then only the dreamers will reach somewhere.
Yeah. Also, in mice, whenever we do a gene therapy or knockout, we do it in a specific background. So we may not see the same effects in an outbred background from mice. In humans, there is no such thing called as a background. So there has to be a global effect of the same gene throughout all species, which is why there is variation from individual to individual, because there is changes in genetics from one person to the other. So we cannot have a consensus about the effects which you are getting as per one species.
And also another robust system is Krelock's system, where you can target the gene in not whole body, like you can have the specific gene. If you can tell about something about that.
Well, okay, let me give a political answer to this. What changes you can introduce is limited only by your imagination. Think of anything you want to do, and the technology exists. You can make it tissue specific, you can make it developmental stage specific, you can make point mutations. The technology exists, and whatever has been developed, this LOXP, basically the idea of LOXP is that you will have a knockout only in the target tissue and not in the whole body. That has been known for many now in decades now, but the same things now being fused along with the genome editing. What genome editing has done is that you don't require ES cells, and the reason for that is that predominant reaction will be homologous recombination, simply because you can cut the endogenous gene and then ask this vector what you are putting from outside to repair it. Rather than it going in random, it goes at the site most of the time, and that's where the advantage is, and that's where all these things that are doable, whatever technology was people have, and that's why I wanted to include in the background what from where we are coming. So it's not that the gene editing has replaced everything else. So it's a cumulative effect, and just adopting all those things together in the context of gene editing. Right?
Thank you, sir.
Thanks, sir, for wonderful.
There would be many more questions. I request you to speak to the speaker straight during coffee time. Thanks for your cooperation. Thanks,
you, Hari.
Satish.
Thank you, Hari. Yeah, can I take it?
Thank you so much, Satish sir. I would like to request Dr. Hari Narayana Rao sir to felicitate our speaker, honorable speaker, Dr. Satish Kumar sir.
It should appear who is giving to whom, okay? Thank you, Hari.
Thank you so much, Satish sir. Thank you so much, Dr. Hari sir. Now, the next lecture will be delivered by Dr. Lakshman Annamalai. The topic is mitigation of gastrointestinal toxicity associated with combined anti-PD1 and anti-CTLA4 immunotherapy animal model development. So I would like to request Dr. Lakshman Annamalai to come on the dais and please deliver the next session.
Hey, I can't, it doesn't allow me to share my screen. The host disabled participant screen sharing. So could you please allow me? Okay, I believe. Okay, got it. Okay, good morning, everyone. I'm going to, give me 1 second. I'm going to, okay. Okay, so the monoclonal antibody therapy has changed the therapeutic landscape of various cancer in human patients. So the reason why it is so is because, are you seeing my little screen as well? Or am I sharing the right screen? Okay, I don't hear anybody, so I believe it's going well. So,
You're seeing your smaller screen.
Sorry?
You're seeing entire screen.
Okay, wonderful. Okay, that's in presentation mode. That's great. Okay, so why immunotherapy is superior?
Can you put in the full screen mode?
That's what I did. I thought. Give me 1 second.
Yeah.
Okay.
Yes.
Okay. How about now?
Yeah. Great. Please go ahead.
Okay. So, the immunotherapy, why it is superior compared to chemotherapy? Because it's highly efficacious, and you don't see the toxicity that you see with chemotherapies. Most of the toxicities. However, we do occasionally see some toxicity, and I'm going to show you some data on about what toxicity we encountered with anti-PD1 and anti-CTLA4 combined immunotherapy, how we mitigated it. Okay, so this work was carried out at Merck Research Laboratories in California, and all the data shown in these presentations are published either in journals or it is part of international patents. So there is no proprietary right concern. So these are the publication and the parent, which is publicly available. Okay, I'm going to start with a simplified version of T cell activation. So this cartoon depicts how T cells are activated in general. An antigen presenting cell usually presents the tumor antigen via MHC molecule, and which interacts with TCR. That is the first interaction. Then second interaction between antigen presenting cell and T cells are via CD28 and B7 molecule. So this combination sends a very positive signal, which is very strong, and activate the T cells, which results in production of IL-2. There is clonal proliferation of T cells, and there is increased survival. If it is a tumor antigen, then there is these T cells are activated against tumor cells. They go and kill the tumor cells. So that is the basic basis of T cell activation. On contrary, there is another molecule which is called as CTLA4, which stands for cytotoxic T lymphocyte antigen 4, which is an intracellular protein. But once in a while, it goes to the membrane. Once it goes to the membrane, it competes with CD28 to interact with B7 molecule. Once this combination occurs, then it sends a very strong negative signal. And in fact, the T cells undergo quiescence, and they're resulting in reduced IL-2 production, reduced proliferation, reduced survival of T cells that results in reduced tumor cell killing. Okay, so this is a negative impactor of T cell activation. So not only the CTLA4, there are a bunch of other molecules which the interaction happens between the antigen presenting cells and T cells, but I'm going to focus on PD-1 molecule. So PD-1 stands for programmed cell death protein 1. It is exclusively expressed by T cells, and the ligands PDL1 and PDL2 is expressed by antigen presenting cells, T cells, and more specifically tumor cells. So this cartoon shows the PD-1 axis, which happens in the tumor cell. The tumor cells produces tumor antigens that is presented by the dendritic cells via classical MHC molecule, and the T cells are activated. So the T cells are ready to kill the tumor cells, but tumor cells are super, super smart. So they express PDL1, which interact with PD-1, and in turn, the activated T cells are, in fact, they're suppressed. So there is no more tumor cell killing. So so people have discovered, and this PD-1 molecule discovery won a Nobel Prize as well. There are two FDA-approved immunotherapy for cancer. One is PD-1 axis blockade and CTLA4 axis blockade. So these two are different molecule, but how about combination of these two? So the CTLA4 activation occurs at the lymph node, probably a draining lymph node of a tumor, and then the PD-1 axis occurs in the tumor environment. So when you combine these two, the survival increases enormously. At the same time, there are adverse reactions that also happens, and the frequency and severity increases in human patients. So this particular clinical trial, which measured the objective response rate, which is a very common parameter measured in human cancer clinical trials. This one is for melanoma. You can see that the dual therapy increased the objective response rate to 61%. However, the monotherapy for each is only 22 and 34% respectively. There is another clinical trial in which the combo therapy did much better than the combination therapy. However, if you see the adverse reaction of grade 3 and grade 4, which are severe, and grade 4 and beyond is fatal, in fact, you can see with combination therapy, it increases really a lot. So what are these adverse reactions? The adverse reactions of immunotherapy can affect any organ, any system, and more severe, and I'm going to focus on GA-related adverse reaction, which are more severe and fatal, and the frequency is higher than any other systemic organ. So This shows the endoscopy of colitis patients who received combo therapy. You can see the blood here in this particular one. It's a fatal one. And almost 9 out of 9 patients in this particular clinical trial had a grade 3 and above. So FDA restricted, of course, US FDA restricted the combo therapy to 3 mg per kg compared to 10 mg per kg, which was recommended after preclinical trial because of dose-limiting toxicity. So this project came to my group while I was working at Merck, and with the objective of to find the mechanism of GI toxicity with dual combo therapy, and to mitigate the toxicity and to escalate the anti-CTLA4 antibody concentration to 10 milligram per kg to achieve maximum objective response rate or overall survival rate. So when it came, the generalized hypothesis we had was maybe the increased T cell effector function and also depletion of regulatory T cells causes this, or the combination of these two causes this. CTLA4, I forgot to mention that CTLA4 is constitutively expressed in regulatory T cells. For people who are unfamiliar with regulatory T cells are anti-inflammatory cells. When that and the inflammation subsides, for example, pneumonia, there should be reduction in inflammation, and the regulatory T cells takes part in reducing the inflammation. So combination of these two probably causes the colitis in patients. So we wanted to dissect what happens with anti-CTLA4 and anti-PD-1. So with individual therapy, the most common CTLA4 related adverse event was gastrointestinal toxicity. However, with anti-PD-1 antibody, it is pulmonary toxicity. However, it was milder compared to this one. So anti-CTLA4 causes severe toxicity, GI toxicity. And moreover, the CTLA4 monotherapy with active FC functional antibodies actually causes really high toxicity. So we know already that CTLA4 FC function actually causes this in humans. So most of the anti-CTLA4 monotherapy antibodies are either IgG1 and IgG2. But anti-PD-1 antibodies are IgG4. I'll explain in a minute why I'm stressing on this. So, okay, got messed up. Anyway, so as I mentioned, the anti-PD-1 or IgG4. IgG4 is the one which has the least ADCC activity compared to any other isotype. So anti-CTLA4 is IgG1, and it causes a lot of FC-related toxicity. So we know that. So in this case, in dual therapy, we want to see whether to blame the FC effector region. So what we did was we used the DX400, which is a very standard mouse surrogate for anti-PD-1 antibody. And to generate the anti-CD4 surrogate, we generated 3 different clones. The first one was of IgG2A backbone, which has a full FC receptor, which can interact with FC gamma receptor. From now on, I'm going to call it as FC effector. The second one has a mutation in FC region. So it has an FC region, but it's mutated, so it can bind to FC gamma receptor. I'm going to call it as a FC mutant. The third one is a nanobody. Again, people who are not familiar with nanobodies are first discovered in a camelid, in llamas. The normal antibodies do not have light chains, and also the variable region is just 15 kilodalton. So it's a very small molecule. So we use this just 15 kilodalton molecule, which doesn't have this lower region. So it's just FC null region. So I'm going to call it as FC null. So what we did was we injected all these separately in 3 groups of BALB-C mice with these dose levels for 7 weeks, biweekly for 7 weeks. And we measured body weight, blood parameters were measured. We did gene expression profiling, and finally, we also carried out the golden standard histopathology. So I'm going to talk about the histopathology scores because I'm a pathologist. This was published in the paper as well. So the upper region showed the normal colon. This is, you can see the ragged region here. There is inflammation here. The inflammation was scored separately. And then degeneration was scored separately, and regeneration, which is mitotic index, was scored as well. And a combination score was generated. It's called as inflammation score in this publication. Okay, so to show this, to explain, I'm going to quickly walk through this slide. The black one here is the combo therapy, and this is anti-CTLA-4 antibody. You can see the body weight is kept increasing for this is 7 weeks. This is in x-axis in weeks. And you can see the body weight went down only for the combo therapy, followed by the FC effector therapy, the CTLA-4 monotherapy. And also, you can see that the intestinal permeability actually enormously increased with combo therapy and CTLA-4 effector function. So there was severe damage to the intestinal epithelium and the barrier. So again, the inflammation was scored in all these. And I'm going to quickly show that, only in the CTLA-4 effector and combo therapy, there was inflammation. In FC mutant and FC null, there was no inflammation at all. This is the inflammation score. And with gene expression profiling, we also found that with combo therapy, and then you can also see similar trend, but in lower intensity with CTLA effector function, there is enormous increase in or upregulation of pro-inflammatory genes. Also, it alluded towards that colitis is mediated by FC effector function in combo therapy as well. Also, we found that T lymphocytes, B lymphocytes, macrophages, and NK cell genes are upregulated in this colon with combo therapy. So, what are the immune attributes of colitis with combo therapy? So, to address this, we used RAC to knock out mice where there is no BRT cells. In another group of BALB/C mice, we depleted CD4 lymphocytes. In another group, we depleted CD8 positive lymphocytes. And the last group, we depleted macrophages for 8 weeks, and then gave the combo therapy and compared with the isotype control. And I'm going to again quickly go through the data. Basically, when you deplete macrophages and the lymphocytes, basically there is no colitis at all. So this colitis is actually mediated by macrophages and lymphocytes, especially the CD4 positive lymphocytes with the combo therapy. So remember, I want to remind the hypothesis was whether it's effector function or not. Yes, we confirmed that it was effector function. The second is Tregs, regulatory T cell, is causing colitis. In the interest of time, I'm going to just fly through this slide. So with combo therapy, we haven't seen any difference in the Treg population. Tregs are measured by FOXP3 positive cells, which is CD4 positive, CD25 positive, and FOXP3 triple positive cells. So there was no difference at all. So this was wider. Basically, there was Tregs were not involved in the colitis induced by the combo therapy. So basically, it's the effector function. So why can't we use the effector null mutants then, in the therapy instead of using the effector positive mutant? So just a reminder, if effector function is very essential for anti-tumor efficacy in the mouse surrogate model. So we don't know what happens in the human, but in this model, definitely FC function is needed. So what we did was we used syngenic animal models where we use BALB/C mice, and we transformed CD23, MC38, or MB49 tumor cells on the back of these mice. And then when the tumor grew on the hunch of these animals to 100 cubic millimeter, then we started treating them with with isotype control or other effector positive or mutant or null mice. And then this is a tumor volume. If you see, only in the combo in FC effector molecule treated animals, the tumor volume went down. In all others, it was comparable to the isotype control, meaning that the without FC function, the antibody did not reduce the tumor volume. Basically, it was not effective at all. However, when we did the combo therapy, to our surprise, anti-PD-1 and anti-CTLA-4 actually reduced the tumor size to very minimum size, and also it was little lesser in efficacy, but comparable to these. So what we concluded was anti-PD-1 with unknown mechanism helps FC-less CTLA-4 and works in a better way. So we also checked, this is the data from other syngeneic molecule. With the interest of time, I'm not going to go through it, but it actually confirmed that this is happening in other syngeneic animal models as well. So to summarize the data, so prolonged anti-CTLA-4 FC effector in BALB mice induced colitis that mimic the adverse reaction that we saw in animal models. And to study that. So this is the best animal model to study these adverse effects. And colitis associated with combo therapy is mediated by FC function. And CTLA-4 blockade does not require FC function for potent anti-tumor activity with combined with anti-PD-1 antibody in mice. Of course, this clinical trial is going on to see what happens in the human. This probably answers the question somebody asked before. And CTLA-4 nanobodies lacking FC domain may provide superior combination benefits with PD-1 blockade by increasing the therapeutic index. So I would like to thank my group, and David Boucher was my postdoc who did most of this work, and Anandi was the antibody engineer, and Drake was the in vivo scientist at Merck. None of these guys actually work at Merck anymore, including me. Smita retired, and everybody left Merck. It's very common with immunotherapy in cancer. So. With that, I would like to take any question. Thank you very much for your attention. But before somebody asks questions, because I worked in gene therapy field for almost 20 years in the US, I would like to answer the question somebody asked about why it's not working in mouse. It's the statement, I'm afraid that it was wrong. Many gene therapy, as Sir said, AAV therapy is very successful, especially for alpha smooth muscle actin, which is sold in India for 16 crores. I've seen in news that this is a very successful one. It is not the gene therapy, so the delivery is very important. It's delivering to each and every cell of the body. However, the toxicity associated with AAV makes it a little failure. So we are there. 10 years ago, we were not there, but we are there. Apologies for taking another question, but I would like to answer the question which you might have from my talk.
Thank you very much for the talk. It's pretty clear, and your analysis and your staging of the different groups literally makes even a not so expert in your field to understand the subject. And thanks again for taking the question of the other speaker. That's extremely nice of you to follow and then give an answer. And my last inquisitiveness, are you talking now taking this call from US?
Yes, I am.
Wow.
I live in California, so I live in San Francisco.
Thanks for keeping you awake. And then I'm sure you are on the other part of the time zone. And
yes,
thanks for the interest, and you are immensely benefited by your wonderful talk. And I now leave the talk for the other scientists if they have any questions to ask. A few questions, please.
Hello, sir. Can you hear me?
Yes, I do.
Can we develop gene therapy for neurological disorders like Alzheimer's, Parkinson's? Because the discovery of the drug for these diseases are extremely challenging. And
excellent question. That's where I am. My background is neurosciences. I'm a neuropathologist trained in Harvard Medical School. So I worked in NeuroAIDS at Harvard, and right now I'm working for Denali Therapeutics. Denali Therapeutic is a neurosciences company. I can't talk more, but we are targeting molecules which are like TMM 176, TMM 106, and p-tau and other molecules which is involved in Alzheimer's disease. We're trying our best, but we are also doing small molecule therapeutics and large molecule therapeutics. We don't have there yet, but hopefully in another 5 to 10 years, we'll be doing something. Right now, we are trying.
I just have a question, technical question actually. Did you look at Th17 cells at all?
Th17 cells. Wonderful question. That is one thing I couldn't talk more because I left Merck. Yes, Th17 cells and IL-12 and more molecules were involved. Unfortunately, I couldn't talk more about it because it's Merck's property. The part I showed you is published. So yeah, definitely. It's a very good question. Very good thought. You followed it thoroughly. Definitely Th17 cells are involved in this.
Thank you. If there are no more questions, I thank again for the wonderful talk.
Sure.
Oh,
thank you very much.
Sorry, my apologies.
I was the one who asked the question about gene therapy, not successful in therapeutics. Maybe 30 years back, when I was a pharmacology student, at the time, gene therapy was touted to be, it came with much fanfare, and it was used for cystic fibrosis. Okay, they recommended for cystic fibrosis, but then it was not very effective. Even after that, the many things now were not effective. Now, well, maybe they would have tried hundreds, maybe one is effective, maybe more there may be effective therapies may come in future. Thank you very much for answering the question. Thank you.
Absolutely. I would like to thank one person. When Dr. Satish Kumar gave an excellent talk when he began with the embryo transfer, I would like to thank Dr. Taru Sharma ma'am. 25 years ago, she was the one who taught me in vitro fertilization and embryo transfer. I know she's somewhere in the auditorium. She's one of the motivational scientists for me. Thank you so much. Thank you, everyone. Have a wonderful rest of your day.
Thank you so much, Dr. Lakshman, sir. He's a very good friend and classmate of our own organizing secretary, Dr. Mahesh Kumar, sir. So thank you so much, Mahesh Kumar, sir, also. I declare that this session is over. Now, I would like to request Dr. Kalakumar, sir, to come on the dais and felicitate our chairperson for this session, Dr. Hari, sir. And I would like to request Dr. Ashok Kumar Devasity to felicitate Dr. Gopal Krishna, TIFR, Hyderabad. Hence, we will move to the next session. The next, thank you so much, Dr. Hari, sir. Thank you so much, Dr. Gopal Krishna, sir. Yeah. Yes. So the next session is the management and accreditation of the animal facilities. For this session, I would like to request Dr. R. Ravi Krishnan, Vice President of RCC Laboratories India Private Limited, Hyderabad, to come on the dais for the chair, for occupying his chair. And I would like to request Dr. Irfan, the scientist from NARF BR, as a co-chair for this session. So first talk for this session, Dr. Ekta Kapoor. She is on live for the talk, Regulatory Requirement and Accreditations of Animal Facilities, GLP. So she will be with us on the live.
Very good morning. This is Ekta, and I'm highly delighted to participate in this session on GLP accreditation. It is an honor and privilege to be here and address the audience for giving you an insight into the GLP certification system and the details about the National GLP Compliance Monitoring Authority, where we are fully adherent to the OECD principles of GLP and the OECD Council norms. So with this, a very good morning and namaskar. I would like to thank the organizers for giving me this opportunity to participate and share my thoughts about GLP certification in the country. I would start my talk if there is any other person who would like to speak. I would just pause for a while and then I will start.
A very good morning, everyone. I take this opportunity to introduce Dr. Ekta Kapoor. Dr. Ekta works as a scientist chef in the Department of Science and Technology, Government of India, and a full-time GLP inspector of National Good Laboratory Practice Compliance Monitoring Authority, NGCM of India. She is a doctorate in pharmacy, specializing in pharmacology. Dr. Ekta has over a decade of experience in GLP compliance monitoring, and she is a lead inspector of NGCMA and very recently promoted as head of NGCMA. The committed and sustained efforts of Dr. Ekta led to the quantum growth and visibility of NGCMA and its activities. She had been instrumental in the rigorous exercise for attaining India a full adherence status to mutual acceptance of data in the OECD Working Group on GLP. Dr. Ekta represents India in the meeting of the OECD Working Group on GLP. She has conducted on-site evaluation of GLP program in Canada, Japan, and Thailand. And with this, I request Dr. Ekta to proceed with our presentation. And Ekta, you have to make sure that your presentation should cover in less than 18 minutes. Thank you.
Many thanks for the kind introduction. I will stick to my timelines. I request the organizers to please share the presentation. Thank you so much. So I will be speaking on the regulatory requirements in the country and the requirement for GLP certification. This is on behalf of the National GLP Compliance Monitoring Authority, Department of Science and Technology, Government of India. Next slide, please. My presentation would be covering the regulatory framework in the country, the birth of GLP principles, where did they come from, how were they incepted, and what is GLP, the applicability of the quality system of GLP, the benefits of the system. I'll tell you briefly about the mutual acceptance of data agreement concept of the OECD. I'll tell you about the NGCMA, the National GLP Compliance Monitoring Authority and its functioning, and the status of India in OECD today. Also covering the future plans of NGCMA with a conclusion of my talk. Next, please. You are aware the regulatory data is the data, including research data, pharmacology, chemistry, manufacturing and control data, including preclinical data and clinical data, which is submitted to regulatory authorities for grant of marketing approval of any new drug, any pesticide, cosmetic product, food additive, feed additive in a respective country. Per se, for the regulatory authorities, these new drugs, pesticides, cosmetics, etc. are also called as the new chemical entities. And it is essential that for receiving a marketing approval for any of these NCEs, the approval of the respective regulatory authority is sought. Next, please. If we look at the drug discovery, development and timeline, it is almost more than 15 years that any one compound, if we start from approximately 10,000 compounds, it is a very lengthy process of even one compound to be approved by the FDA. And as we can say, the entire drug discovery process of a new chemical entity goes through a series of phases, including the discovery part, then the preclinical testing, then the clinical trials, filing of IND, then, first filing of IND, and then phase 1, 2, 3 clinical trials, filing of the NDA application, and finally, submitting of the entire gamut of data as a dossier to the regulatory authorities. It also takes a number of years plus a lot of monetary investment goes into the process. Next, please. Next slide, please. So if we go back to the history, we all have read about the thalidomide tragedy of the 1960s, which happened in Europe, particularly in Germany, when a number of children were being born with, malformed limbs. And this was the reason that the regulatory authorities were highly alarmed about the safety data of the drug which was given to the pregnant women for morning sickness. This drug was nothing but thalidomide. And when the data on thalidomide was looked into, it was felt that the data had not been generated, or it had not been, the safety data had not been gone through by the regulators. Therefore, a mandate was born for all manufacturers of pharmaceuticals, agrochemicals, industrial chemicals, cosmetics, food and feed additives to provide safety data in order to have their products registered or licensed. And the safety of these drugs was to be, data had to be generated based on the tests or studies decided on by government regulators. Next, please. As the regulatory authorities required a number of tests to be conducted, so a wide range of studies were to be done on microorganisms, in vivo studies on rats and mice, studies on non-human primates, and a number of environmental studies for agrochemicals and industrial chemicals. All this led to the emergence of the entities known as the contract research organizations, the CROs, with the claimed ability to conduct these studies. Today, we have many CROs in our country which are working at the global level, and they are having state-of-the-art facilities and GLP certifications for conducting these types of studies. So at that time, there were, since a number of studies had to be conducted and a lot of data had to be generated, there were certain incidences. For example, the industrial biotest laboratories of USA, which showed that incompetence, lack of knowledge, and sometimes outright fraud could jeopardize the studies or could reduce the value of results presented. It was this time that the principles of GLP were born under the OECD. Next, please. So what is GLP? It is actually a quality system concerned with the organizational process and conditions under which non-clinical health and environmental safety studies are planned, performed, monitored, recorded, reported, and archived. I would just like to spend 2 minutes on this slide because each word in this definition has its own importance and its meaning. When we say GLP is a quality system, we have to, ensure that the quality has to be maintained according to written procedures, and they are in accordance with the standard guidelines or standard OECD principles. GLP is again applicable to non-clinical or preclinical health and environmental safety studies. So it's not just tests that are being done. If we are going to certify the tests or accredit a laboratory for conducting certain repetitive type of tests, there is another quality system, ISO IEC 17025, which is under NABL in our country. And this GLP is not only establishing the safety of, the quality of tests being performed, rather it covers the entire safety studies that are being performed for the new chemicals. So the process of GLP monitoring actually involves how the study is planned. There is a written down document called the study plan, and written down SOPs or standard operating procedures according to which the studies are to be performed. So when we say planned, we always look at the study plan, which covers the entire details of the study that is to be conducted, including the materials required, the methodology to be followed, who will be doing, who personnel that will be involved. And of course, the study plan is a document which is approved by the study director, who is the single point of study control. Then the studies performed in accordance with the study plan and the SOPs. Of course, any quality system doesn't go without being monitored. Here we have a special component of GLP, which is called the quality assurance, which is going to monitor each and every study that has to be conducted under GLP. The study results are recorded not on any piece of paper or any notebook, but on raw data formats, which are specially designed and approved for use in a GLP test facility for conducting GLP studies. The study results are again compiled, collated, and reported in the form of a written down study report. And finally, the study plan, raw data, study report, and all the specimens generated during the study, including the test and reference item used therein, are archived at a safe place within the test facility or at a designated location for a distinct period, which is decided by the regulatory authorities or the GLP compliance monitoring authorities. Next, please. So we come to the applicability of GLP. I already mentioned GLP is applicable for various test items or NCEs covered under the following categories: industrial chemicals, pharmaceuticals, both human and veterinary, agrochemicals, cosmetic products, food additives, feed additives, medical devices, or others where specified. We have 9 areas of expertise for which a GLP certification is given, and these include the physical and chemical testing, toxicity studies, mutagenicity studies, environment tox studies on aquatic and terrestrial organisms, studies on behavior in water, soil, and air, residue studies, studies on effects on mesocosms and natural ecosystem, analytical and clinical chemistry testing, and again, an area of others which need to be specified. These may include areas like safety pharmacology, DMPK studies, or even bioefficacy studies based on the WHO protocols. Next, please. Advantages of GLP is that it limits wastage of resources, it ensures high quality of results, ensures comparability of results, and promotes mutual recognition of results thus helping to remove technical barriers to trade. Next, please. The main goal is to help scientists to obtain results which are reliable, repeatable, auditable, and recognized by scientists worldwide. Next, please. Coming to the mutual acceptance of data system of the OECD, the MAD is a multilateral agreement which allows all the participating countries to share the results of various non-clinical tests done on chemicals. Today, there are 38 member countries of the OECD. India is a non-member, but we are full adherent to mutual acceptance of data system. This system is based on the simple concept of tested once, accepted for assessment everywhere. It means that if a MAD adherent country has a test facility which is submitting data as per the principles of GLP and as per the OECD test guidelines, in that case, all the other OECD member countries, that is the 38 countries of the OECD, plus the non-member MAD adherent countries will accept that data generated by that test facility globally. This has huge economic and trade benefits for any economy. The MAD agreement is composed of 3 OECD Council Acts, the 1981, 1989, and the 1997 Council decisions. Next slide, please. The benefits of MAD are both monetary, because it helps in reducing the duplicative testing and creating a framework for sharing of results. There are many qualitative benefits, that is, the governments are able to evaluate and manage more chemicals than they would if they were working independently. It helps in avoidance of delays in marketing new products, reduction in non-tariff trade barriers, reduction in animal testing, and development of technical instruments that improve the quality of chemical evaluations and regulations. Next, please. Testing in compliance with GLP is conducted in synchronization with 2 different types of documents: the OECD principles of GLP, which are dynamic, with new and revised guidance and consensus documents being introduced whenever issues need explanation. As on date, there are 22 documents of the OECD principles of GLP. In addition, we have the OECD test guidelines, which are the technical guidelines introduced after many rounds of ring testing and discussions in all the OECD countries. The benefit of having the GLP studies conducted in accordance with these 2 types of documents helps that they are common guidance documents having global consensus, which ensure that the data generated is robust and can be relied on by regulators worldwide. This also enables the regulators to take informed decisions which favor the population and the environment. Next, please. Just taking you through the history of GLP in India and the challenges faced by Indian test facilities way back in the 1990s. So the exposure started in India to GLP in 1991. Prior to the National GLP Compliance Monitoring Authority being set up, the Indian manufacturers were largely dependent on foreign test facilities for generating non-clinical data. This led to a major foreign exchange burden because they had to pay for the tests that they were getting done. Non-GLP data from Indian test facilities was an important technical barrier for international acceptance. Also, the Indian test facilities had to approach the GLP monitoring authorities of other countries like the Netherlands, Switzerland, Germany, and Belgium, which was expensive and yet limited the global acceptability of the data. Next slide, please. So with the requirement of the Indian test facilities, the NGCMA was set up by a cabinet decision in August 2002 under the administrative control of Department of Science and Technology with the following objectives: to operate in accordance with the OECD Council norms, to grant GLP certification to test facilities by examining their procedures and practices to carry out safety studies on chemicals, and to provide a network of GLP certified labs in the country for the regulatory authorities and assure regulatory authorities that safety data generated from GLP labs can be relied upon. Next slide, please. The slide shows you the functional chart of NGCMA. At the highest level, we have the apex body, which is chaired by Secretary DST. It is under the apex body, we have our technical committee on GLP, that is a recommending body, which recommends whether or not a GLP certification is to be granted to a facility based on the inspection results of that particular test facility. We have the NGCMA Secretariat at DST, and we have trained GLP inspectors in our setup. Taking you through the journey of NGCMA. So when the NGCMA was born in 2002, we had the compliance procedures built up, documented and implemented. The first certificate was issued by NGCMA in 2004. We had to train our inspectors, sensitize our test facilities, train the test facility people, and of course, we were to be evaluated by the OECD for the compliance and for adhering to the MAD agreement. We were evaluated twice in 2008 and 2010. Next slide, please. And based on the results, on 3rd of March 2011, India was conferred with a full adherence status to mutual acceptance of data. As per the OECD's publication, saving costs in chemicals management, the net savings to the government and industry brought about by the MAD program is approximately more than 309 million euros a year. This also saves tens of thousands of animal lives annually. Today, India is the only country in South Asia to be a full adherent to MAD, and it is, it was really proud to be one of the countries, the only one in South Asia, where countries like China are not even a provisional member of the MAD. Next, please. So after attaining the full member status, we have been participating in all meetings of the OECD working group. We have been attending various OECD training courses for GLP inspectors. We got the chance to organize and host the 12th OECD training course for inspectors in Hyderabad during October 2015. We have been conducting joint GLP inspections and study audits at the request of various regulatory authorities, including the US FDA. Next slide, please. We have been also participating in the on-site evaluations of other countries, which has been designated to India by the OECD. And I myself have participated in the evaluation of most developed countries and the the best GLP programs of the world, including the programs of Canada and Japan. And Thailand, of course, we did because Thailand was to be added as a non-member MAD economy in 2019. India has now been designated as the vice chair of the OECD's Working Party on GLP for 2021 to 2023. The on-site evaluation of Indian GLP program is again scheduled this year in July, and we are getting ready and gearing up for the same. Next, please. The details about the Indian GLP program certification in India on GLP is still voluntary in nature. The certificate is valid for a period of 3 years, and 6 months before the expiry of the current certificate, the test facilities have to submit an application for recertification. The certified test facilities are subjected to mid-cycle surveillance, and all the procedures and practices of NGCMA have been documented in the form of procedures which have been implemented through a documentation control system. Currently, there are 21 procedural documents to cover all procedures with respect to compliance monitoring and administration of the GLP program. Next, please. So, there are different types of GLP inspections that we conduct within NGCMA, and all these inspections are documented in our procedures. The first inspection that we do for any new applicant test facility is a pre-inspection. So this pre-inspection is to determine the suitability of the test facility, to undertake a final inspection, to get an idea about the organizational structure, get an idea about the quality system, put in place, and also the types of studies which are conducted by the test facility. After the pre-inspection is done, we give the observations of the inspection to the test facility, where an action is taken by the facility to each of the observation. If the ATR is found satisfactory, we do a final inspection. The final inspection is a detailed inspection, including both facility inspection as well as the study audits. So after the final inspection, if found suitable, the test facility is granted a GLP certificate, which is valid for 3 years. Of course, mid-cycle surveillance takes place 18 months from the date of certification or recertification. During the 3-year cycle, if the test facility wants to extend its scope, it can submit an extension in scope application to NGCMA. At the end of the 3-year cycle, we conduct a recertification inspection. Any test facility which is a part of the National GLP Monitoring Authority can be subject to surprise inspection if there is a complaint about the test facility from any regulator or any other monitoring authority, or if NGCMA has a reason to believe that the data generated at the test facility cannot be relied upon. There is a verification inspection which is conducted by NGCMA to verify the ATR or the action taken report submitted by the facility on site. Of course, we are obliged to conduct inspections at requests of regulatory authorities, be it the Indian regulatory authorities like the DCGI and the Central Insecticides Board, or it is the foreign regulatory authorities like the US FDA, US EPA, MHRA, etc. Next, please. So basically, there are two important.
Dr. Sorry to interrupt. I mean, we are running out of time. Probably next.
I'll just wind up. So there are two important elements of GLP inspection. They include the lab inspection and the study audits. Next, please. So we have tried to conduct an impact analysis study of the national GLP program since we have covered 2 decades of setting off of NGCMA. The objective of this study was to seek inputs from the important stakeholders, namely the sponsors, regulators, GLP inspectors, and members of NGCMA's technical committee on GLP. Next, please. The responses were collected, collated, and the results indicate that so far, Indian test facilities have conducted more than 60,000 non-clinical safety studies, and we have also this has been a major step towards self-reliance in the development of new chemical entities. As on date, we have 50 GLP certified test facilities, which are either CROs or they are conducting these studies to support their own parent manufacturing companies. Next, please. Currently, GLP test facilities in India collectively have the competence to conduct non-clinical health and environmental safety studies on 8 different types of test items and 9 areas of expertise in 28 types of test systems. The facilities have also been able to create job opportunities for various profiles such as scientists, managers, and technical staff. Next slide, please. So I'm just going to conclude the future plans. In the NGCMA has already undergone complete digitization, and impact analysis study has been conducted. As a vice chair of the OECD Working Party, we are there to build a roadmap for and evolving OECD principles and test guidelines, keeping in view the forthcoming challenges in the GLP scenario. For example, the risk-based assessments, data integrity, inspection of cloud-based softwares, etc. We are handholding other OECD member countries, non-member countries with establishing the global standards of GLP. Next, please. the government labs in India to bring them at par with international quality and encouraging them for GLP certification, expanding the scope of GLP program to new research study areas as per mandate of the World Health Organization. And we are there to implement a system of GLP certification of highest commitment to quality, which contributes to the dream of Atmanirbhar Bharat or self-reliant India. Next, please. My last slide. Compliance to both the OECD principles and OECD technical guidelines allows the regulators to assess the safety and carry out the risk assessment of the new chemicals, and the entire system of regulatory toxicology supported by OECD principles of GLP and mutual acceptance of data is geared up to make creditable data available to regulators. To sum up, the GLP is a quality system meant to ensure protection of the public and the environment. Thank you so much for your patience listening. I'll be happy to take up any questions.
Thank you, Dr. Ekta Kapoor. It's a wonderful talk. We have time only for 2 questions. Any questions from the audience? Please.
Myself, Dr. Basha. Why it is only voluntary where such a significant role is playing, but why it is only the voluntary but not the mandatory? GLP. GLP.
I got your question. Thank you so much for this. The reason for GLP being still voluntary is that the regulators have not made it mandatory for all data that is coming to them. The regulators in India per se, the CDSCO has given the new drugs and clinical trial rules where they say that GLP data will be required, but they don't make it mandatory. Similarly, for the CIB, the Central Insecticides Board, the regulator has not made it mandatory that the all data needs to be GLP alone. They say NABL or GLP compliant data will be accepted. So the day the regulators make it mandatory, the GLP certification in India will become mandatory.
Another question.
Please.
Only one question, Hello, due to time constraint. I'm sorry about this. Probably I can share an email ID. You can write an email for any further clarification. Any more questions?
Thank you, Dr. Ekta, for your wonderful brief presentation on GLP, and thank you for accepting our invitation and being with us. Dr. Ekta's presentation is available with the organizing committee. If somebody is interested to collect, they can collect from us. There is one announcement, and sorry for the delayed announcement. There are some important meetings with the speakers like Dr. Ekta Kapoor and Dr. Mohanty. They have the scheduled meetings. So we shifted oral presentation after tea break. Now Dr. Mohanty presentation will be there. Then we'll be breaking for the tea. After tea, we'll be continuing with the oral presentations. Those who are selected for the oral presentation, I request them to be there in the auditorium. Thank you so much. Yeah. Now we are continuing with Dr. Mohanty Gattayamin presentation. Sir, please.
Thank you.
Good morning.
I'm happy to introduce Dr. Montip Gattayamin. She's a senior director for the Southeast Asia at HELAC International, a position she has held for the past 11 years. In this role, she supports HELAC International Accreditation Program to advance research on animal welfare in the region and assist institutions to attain accreditation. Dr. Montip received her veterinary medicine degree from Thailand in 1984. She is a diplomat of the American College of Laboratory Animal Science. She has over 30 years of experience in laboratory animal medicine and previously served as Assistant Chief for Research and Laboratory Animal Medicine and also Senior Research Scientist at the Department of Veterinary Medicine, United States Army Medical Component, Army Forces Research Institute of Medical Science, Bangkok, Thailand. The program has attained HELAC International Accreditation since 1999. She has authored and co-authored more than 50 scientific publications. She translated the guide for the care and use of laboratory animals, National Research Council, the Occupational Health and Safety in the Care and Use of Research Animals, and the Guide for the Care and Use of Agriculture Animals in Research and Testing to Thai. Dr. Montip is a reviewer of the Journal of Visualized Experiments and a member of the editorial board, Animal Models and Experimental Medicine Journal. She is a former member of the ELAC International Council on Accreditation. She has served on International Council for Laboratory Animal Science, ICLAS, governing body, and is a chair of the Asia Regional Committee for 2019 until 2023. I requested Dr. Montip to present on the topic Coronavirus Guidance and Update, ELAC International. Please.
Thank you for the invitation. Good morning, the organizing committee and scientific committee of LASA India. And good morning to all of the attendants in the Hyderabad and virtual attendants who attend online. It's my great honor to participate in, and I hope to see you next time in the LASA India conference next year. I have a committed travel that I cannot be there. So see you next time. So today I will present the coronavirus guidance and update from ELAC International. As we know that coronavirus already spread around the globe, and over 530 million people already been infected. Because of the impacts of the COVID-19 pandemics that we need to follow the social distancing, and there has been some lockdowns or temporary shutdown, and some animal facility operating with only essential staff, and also we have limited resources. So ELAC International understand those impacts very well. So we have act with the resilient action to adapt with that. So we put the latest coronavirus guidance into the website to allow accredited institution to clearly understand what would be the expectation. So we start that since the beginning of 2020. By using that, the guideline, then despite the challenges and disruption during the pandemic, the accredited institution continue to maintain high quality of animal care. ELAC International have frequently asked questions developed for COVID-19. And we have posts on the website about the service postponement if institution accredited by ELAC has been impacted, and they cannot accept the on-site evaluation, that is what we call site visits on their schedule. The system will allow that institution can submit the request, and then the site visit will be postponed and accredited status remain unchanged. During the postponement or during the pandemics, the ELAC International understand that you have limited personnel or access to the resources. So the program review and facility inspection for the animal care and use program can be done at least annually or more often as required. Each institution develop their own criteria how to manage the program review and facility inspection following the local guidance to avoid the social contact. In addition to that, the post-approval monitoring that occur routinely, but during COVID-19, ELAC International accept that the IACUC could accept alternate option to allow the monitoring to continue. Typically, the services schedule start with the entrance briefing and program description review, facility tour, lunch with the IACUC, document review, executive session of the team, and then exit briefing. But during COVID, we have social distancing and especially the travel restriction that we cannot conduct the on-site evaluation because we cannot travel there. So ELAC International developed hybrid site visit schedule during the pandemic by allowing the coordination well with the key personnel of the institution to schedule virtual meeting and also secure document sharing. By doing that, the site visit team can meet with the key personnel of the institution by virtual platform. And then document review can be done also online or sharing document. The on-site facility visit sometimes have to be postponed until facility can be done. And if we conduct site visit on-site, we will follow social distancing and limited personnel as possible. Then the other part also can be done off-site. And these can be scheduled more than one time. It can be a half day of a few days apart. And normally exit briefing must be done within 5 days after the on-site facility visit can be done. So that's what's happening during the pandemic. And currently, there are still some countries that we cannot travel there yet. There's still some restriction. For example, the quarantine requirements still continue in Taiwan, in Hong Kong, in the People's Republic of China. But the rest of the Asian country where accredited institutions are, we can go there. The vaccinated team can go there. For the update of the ELAC International, all we know that the official name of ELAC is ELAC International. So no need to spell out as acronym anymore. ELAC International continue to serve as the only organization that assesses and accredits animal can use program for research, teaching, and testing around the globe. And we are nonprofit, non-governmental. So the standard and policy has been reviewed by the Council on Accreditation, and ELAC International adopt those references as our standard. So for several who are new to ELAC International, we have member organization, including the recent LASA India already applied to become a member organization of ELAC International. So that showed the strong support of LASA India, along with other international organizations such as ICLA, FILASA, AFLAS, along with the Chinese Taipei Society of Lab Animal Science, JCLAM, KCLAM, and TALAS and SALAS. So that's very well support from those organizations. From the member of organization, the representative of member organization vote to select the board of directors. And currently we have 12 member organization delegates serve on the board of directors. And we have also president and vice president of Council on Accreditation serve in the board of directors. So this is the governance body of ELAC International. ELAC International has 3 offices in USA, Spain, and Thailand. So we have the CEO, global director, and 3 senior directors. The pool of international experts, over 500 of them joined ELAC International. There are 73 members serve on the Council on Accreditation and from 16 countries. So those Council on Accreditation members are the group of experts who deliberate on the result of accreditation. We also have assistance from Council member emeriti. The experts also help conducting site visits. In addition to that, we have over 400 ad hoc specialists and consultants from 35 countries. Currently, we have 21 council members from Asia, and we have 80 ad hoc specialists. Amongst the 80, 13 of them are from India. And we welcome the new council member, including Dr. Krashnavin B. Patel from India, Dr. Suying Pan and Dr. Sung Yok Sok from Korea. This time will be effective in July 1st. So welcome the 3 new members from Asia. As of now, we have over 10,059 accredited institutions in 50 countries. If we see the distribution, we know that Asia Pacific or Pacific Rim country, we contribute about 21.7%. And we have the growth rate about 1 to 1.5% each year. Currently 230 units as of May 5th. So the growth of the accredited institution in the Pacific Rim area started in 1995 and continue to expand. We start the Pacific Rim first section in 2009 and then start the second section in 2019. So these members conduct the deliberation separately in the 2 sections. So the distribution in Asia, we have 12 countries total. And in India, we have 29 accredited institutions, and 27 of them are commercial institutions, CRO, pharmaceutical, accredited institution. We also have 2 nonprofit organizations in India become accredited. So the overall commercial institution in the Asia Pacific region is 68%, around that. And from university, it's 14.3%. So this is slightly lower than the global percentage that is about 28%. So we are looking forward to see more university that coming into applying to become accredited by ELAC International. ELAC International use 3 primary standards. The Guide for the Care and Use of Laboratory Animal, that is called the Guide, National Research Council 2011. That is the main primary standard used in Asia. For the program that use agricultural animals in the research and teaching, we have another reference called the Guide for the Care and Use of Agricultural Animals in Research and Teaching, that is called the ACT Guide. The Council on Accreditation recently adopted this ACT Guide, and the effective date for this new guide will be the winter 2023. And some units that have clients from Europe, they also adopt the Council of Europe ETS 123 as their guiding standard. So you can find the ELAC reference resources on the website. You can click here. And currently there is a new reference that is called Biosafety in Microbiological and Biomedical Laboratory, the 6th edition BMBR book, published by the Center for Disease Control and Prevention, National Institute of Health. So we refer to this reference when visited and evaluate the institution using biosafety agents. Also, we adopt the AVMA guideline for euthanasia of animal 2020 edition. In this guideline, the changes from the previous version is that an optimal flow rate for carbon dioxide euthanasia system should displace 30 to 70% of the chamber volume per minute. So that is the new one that we accepted. Previously, the guidelines already emphasized that the carbon dioxide must be supplied from commercially supplied the cylinder or tank, and an appropriate pressure reducing regulator and flow meter must be used. Pre-filled chamber is unacceptable. Oxygen administered together with carbon dioxide is not recommended. If euthanasia cannot be conducted in home cage, chamber must be empty and clean between uses. So if you go to the ALAC website under the FAQ, frequently asked questions, there are some new updates topic in the website. Regarding scientific reproducibility, poultry caging, swine housing system, dairy calf housing and social enrichment, carbon dioxide for rodent euthanasia, HVAC measurements in agricultural settings, and consistency in accreditation. So please seek ALAC website for more information. It's the important new frequently asked question that will come in soon, that is called managing and reporting adverse event. From reporting area is consider inadequate veterinary care, conditions that resulted in unexpected animal deaths, natural disaster, significant animal rights activities, inappropriate euthanasia technique or failure to confirm euthanasia, substantiate compliance reports regarding animal welfare concern. So in the accredited institution, if these things happen, so ALAC expect that the institution will report to ALAC office. In addition, if there is any internal or external reviews or inspection or other similar report that document significant adverse event or non-compliant that resulted in animal harms or death, or investigation by national oversight body, and other serious incident or concern that negatively impact animal well-being, such as failure to follow approved protocol which resulted in compromised animal welfare or death during animal transport. So we expect that the institution will report as well. In addition, significant human health issue directly related to animal care use program. So please stay tuned for the new ALAC FAQ to be posted. But in brief, the guidance will be provided that each institution will develop some criteria and to manage the prompt reporting in those areas of unexpected events that occur in your institution. With that, I conclude my presentation and welcome the questions. Thank you.
Thank you, Dr. Montip, for complying to the timeline for your presentation. And we will take a couple of questions now. Any questions? No more questions? Any questions from the audience? Dr. Montip, you are so lucky enough today morning. No questions from the audience.
Feel free to contact me if you have further questions. I'll be happy to share the information. Thank you.
Thank you. Thank you very much for your wonderful speech and giving insight about the ALAC program and sensitizing how to take care of this pandemic and be preparedness for this pandemic management apart from the disaster plan. Thank you once again.
So I congratulate LASA India for the success of the meeting and Swadika.
Thank you so much, Dr. Monty. Now, I would like to request Dr. Anantha from Sanzyme to felicitate our chairperson, Dr. R. Ravi Krishnan, sir. I would like to request Dr. Durga Bhavani, Virtual Biotech Hyderabad, to come on the dais to felicitate our co-chair, Dr. Irfan, scientist, NARF PR. Thank you so much, Dr. Ravi sir and Dr. Irfan for chair and co-chair for the session. Now I will hand over to the chair.
Now we are breaking for the tea, so I request all that within 10 minutes we can come back. It will be good so that we can continue with the oral presentation. I request Dr. Kalyani to take over for the oral presentations. Kindly try to maintain the time. So 10 minutes, I think it will be good if we can come back. Thank you.
हाँ है सर.
ये वाला यहाँ पे खोल के देख सकते हैं उद्धव HAV ऐसा क्रॉस क्यों आ रहे वैसे
नहीं वो ड्राइव में अपलोड नहीं हो रहा।
इधर नोट्स भी दिखते हैं इधर cannot download information. जाते हुए तो धूप लग रही
लगेच झोपलं नाही तरी पण कॉपी नाही करणार आहे जस्ट वांट टू सी इट Can I see it?
No, sir.
How it looks?
No, it is not. No, after he is not.
He's not. I thought how it not was.
No.
Okay. I think if it's not, then just want to see it, how it looks there. That was the reason. No, don't change the slide.
No, I'm not changing, but I'm just looking how
Hey, what is happening, you know, what I observed from your end, you people are stretching the slide to display over here. That's why the compaction goes. So you have to keep it as the same mode. Don't
But,
scratch. No, but don't scratch. No, put it in between.
gap will be there.
No, that's fine. Gap should be there because what happened, you know, if you stretch it, na, so it distorts. Actually, the slide gets just like just ask him to put it the way it is there. Otherwise, it would have been shared with us that make the slide in the same format. Yeah, it was not. Yeah, ऐसा बोलना चाहिए था. अगर नहीं है तो फिर then it will be difficult. Stretch नहीं होना चाहिए. One or two slides the presentation ना they looks very good because in the same mode whatever is there.
Actually पहले से था ना
हाँ पहले से था. तो उन्होंने PDF नहीं उन्होंने PDF पे रन किया. PDF रन किया ना तो PDF स्ट्रेच नहीं कर पाते.
अच्छा
हाँ.
ये अभी वाले सेशन का कहाँ रखा है आपने ये डेस्कटॉप पे है सारा ऑल इज ऑन डेस्कटॉप हाँ ओके तो ये शायद माय इज फर्स्ट वन इन सीरीज so you will open this Amit
Will give some. Yes. Yes. This is this one. Yeah.
Oh, this is the pointer? Yes. This forward, backward, and point. Okay. Thank you.
ఇది ఓవరాల్ అని ఉందా? ఇంట్లోనే ఉంటాయి అన్ని ఓవరాల్ ప్రజెంట్. ఉంటాయి కాబట్టి ఫస్ట్ ఇలా తర్వాత ఇలా తర్వాత ఫస్ట్ దొరకలేదనుకో ఇంట్లోనే ఉంటాయి కదండి. అబ్స్ట్రాక్ట్ నెంబర్ కొట్టితే అయిపోతుంది. అబ్స్ట్రాక్ట్ నెంబర్
I request everyone to please join and occupy your seats. We'll start the next session. I also request the presenters for this award session for LASA members. Please be ready. So for this session, you will be given 6 minutes, 5 minutes for your talk and 1 minute for discussion. There is a confidential team sitting in the podium who will be judging your presentations. You'll also be judged on your time management, so please stick to your time. Krishna. So we'll start with the next session. I'll just repeat the rules again. This is award session for the best oral presentation for LASA members. You'll be given 6 minutes, 5 minutes for your talk and 1 minute for discussion. There is a confidential team sitting in the podium judging your presentations. Time management will also be judged, so please stick to your time. We'll start with the first presenter, Dr. Amit Kumar, Division of Animal Genetics, ICR IVRI, Bareilly. So the title of his topic is Characterization of Inbred Swiss Albino Mice and Identification of Candidate Genes Related to Fitness Traits. You can start when the time goes to 0.
Thank you for providing opportunity to be here at NAM for presenting our study. We all know that the mice is widely used lab animal species because it is easy to handle and short lifespan with high fecundity. So 53% of the species being used is the mice and 19% followed by the rat. So inbred strain, how it is developed is well known to all of us by brother-sister mating, at least 20 generations consecutive. And there are some lab animal strain developed already by following this way. And the strength of inbred lab animal is to maintain the isogenicity and homozygosity, which actually yield reproducibility of the result in a better way. So we can repeat the experiment the same accuracy, and the prepotency and imprinting ability of the inbred strain of mice is of course better. So rationale of this study was to develop one inbred strain using Swiss albino as a foundation stock, and to estimate effect of inbreeding over the generation on the performance traits as well as the fitness trait. In a study, we also investigated as a part of study the fertility genes, how it is getting affected after inbreeding. So resources where we went for the filial inbred mice of the Swiss albino foundation stock. In this study, we are going to select F4 generation for study the fitness trait, for which 90 full shape meeting were settled, and we obtained 506 progeny out of it. And from where we again in next generation, 156 full SIB meetings were settled for the getting F5 generation. And we recorded the phenotype of the 150 F5 generation. We are going to record here body weight at birth, litter size at birth. Were categorized in the 3 different categories for investigating its effect on the performance and fitness trait. So liter weight at the birth, body weight at the weaning, litter size at the weaning, litter weight at the weaning, adult body weight were our traits. So in this, we can see that effect of inbreeding was significant on the body weight at the birth, where it is decreasing as it is increasing over the generation. Then the litter size at the birth, again it is decreasing over the size over the generation. It is expected that it will decrease from the foundation stock to F5. It decreased, and it is a continuous process. Right now we are going with 11th generation of the inbred meeting. So this decreased, and effect of the litter size at birth on the body weight, body weight at birth, body weight at weaning, adult body weight, body weight at birth, and the live weaning weight. So this is also over the time, according to litter size, decreased. So F5 generation also, we have noticed F4 and F5. The effect of the litter size on birth, we have investigated the effect. We have also used the microsatellite marker for investigating the effect of inbreeding as a molecular marker we have used. And right now we are also going to using the genome, the whole genome sequencing data. We are analyzing over the generation for investigating the homozygosity and reduction in the heterozygosity in those mice. And we have estimated that the overall mean inbreeding coefficient in F4 inbreeding inbred mice population was 69%. And one of the microsatellite was showing just 52%, another 80%. So we have investigated the association of these microsatellite markers, and we found certain markers associated with the litter size at weaning, litter weight at the weaning, and highest homozygosity was obtained for the D7 mid to 323, and the lowest was for the D7. So we went for the transcriptome profiling also. In stress cycle, we selected once there was more than 90% cornified epithelial, and then we went for the sample collection. The criteria were to select high litter size group and low litter size group. So we went for the bioinformatic pipeline by sequencing on Illumina HiSeq 10x. So this is the average statistics what we got by DSEQ analysis. We got 1018 genes using our environment, and 671 genes were found to be downregulated, and 347 were upregulated. This is the volcano plot we drawn for the, and this is the top 10 upregulated and top 10 downregulated genes we recorded after RNA-seq analysis. And we found that gene L has 22 degree of connectedness, and it is linked to the progesterone mediated oocyte maturation. IRS2 has 8 degree of connectedness, which regulates the ovarian steroidogenesis, folliculogenesis, and ovulation in mice. And certain down-regulated gene also we encountered, CDH1, KNG1, ATK3, 13. This is the hot heat map where we found that most of the genes were down-regulated in the high litter size group, where we found one of the important genes.
Your time is up, Dr.
Endocannabinoid receptor plays critical role in the pregnancy. We validated this in the qPCR. And this is the conclusion what we told. Thank you.
Thank you. So any quick questions? Yeah.
Have you ever checked? You said that Swiss albino outbred actually.
Yes.
How you confirm it is an outbred?
Yes.
Then
Yes.
the second question is,
what is the rationale of selecting only this particular microsatellite markers?
Actually, first question that before selecting foundation stock, we ensured that there is no any close breeding.
No.
So,
How you confirm close breeding is other way? What is the how you confirm it is in Swiss Filipino outbred?
So
what is the difference between Swiss Filipino outbred and inbred? Whether there are any changes in the genes or any any mutation occurred or anything? How you confirm rather than selecting close breeding or anything?
we initially in our like lab animal house, what Swiss albino we are maintaining from since long time.
No, that I want to ask because you are maintaining long time and it will automatically become an inbred. Okay, so
no, that for that purpose only. Prior to foundation stock, 3 generations we mated for ensuring that there should be no any close relatives. First, and the second.
Yes, time up. Actually, so I should not. Later we'll discuss it. Okay, thank you.
So microsatellite we selected on the basis of the report available and used in the mice. But now we are moving on the genome. Yes. Okay, thank you.
Okay, thank you.
Done?
So the next presenter is Dr. Shanti Bhushan Senapati from Institute of Life Sciences, Odisha. Dr. Senapati will be joining us online.
Good morning, madam, and good morning all. Should I share my screen or it will be displayed from the team?
You have to share your screen.
Okay, please. Okay.
I hope you have heard the directions before, sir. You will be given 6 minutes, 5 minutes for presentation and 1 minute for discussion. Please stick to your time.
Sure.
Thank you. Thank you.
Thank you. Thank you again. Thank you very much to the organizing team for giving me this opportunity. So before starting discussing about our research work, I just want to mention that at the beginning of this pandemic, my group at ILS took the opportunity to establish the hamster inspection model of SARS-CoV-2. And we collaborated with one of our colleague, Dr. Gulam, which is a virologist. At that time, actually, his group actually established a SARS-CoV-2 isolate from the locally identified COVID patient. And they have gone ahead and characterized its genome and other phenotypes. So using that local isolate, we infected hamsters in our that time existing BSL-3 facility, following all the regulatory procedures. And what we noticed that this isolate also manifested similar kind of pathology and disease manifestation as people have by that time already reported in from other parts of the world. So it and also it mimicked most of the features, critical features of the lung pathology that has been reported in human patients. So based on that preliminary data, actually BIRAC funded us to expand our small BSL-3 facility and establish a standalone exclusive BSL-3 facility for SARS-CoV-2 research. And the mandate was to run it on a service mode. And being the PI, I'm glad that now the facility is full-fledged running and equipped with high-end equipments like micro-CT scanner. So with this, I'll just come to the topic today, that is to evaluate the efficacy of N-acetylcysteine in a hamster model of SARS-CoV-2 infection. I think most of you know that due to the known property of N-acetylcysteine or NAC, many people from the very beginning believed that it will have efficacy in COVID-19 patient, either mostly to reduce the inflammation, because due to its known antioxidant property, anti-inflammatory property as an inhibitor of NF-kappa activation. So it was strongly believed that it can up to some extent control the lung-associated pathology in humans. And also due to its known mucolytic property, many clinicians also started giving NAC for COVID-19 patients. But till then, there was no a control study available in any preclinical model. Really, NAC can give any advantage in a COVID-19 scenario or not. So that's why we just thought about to check it in a hamster model of infection. So this is our study strategy. We tested 2 doses of NAC, a low dose 150 milligram per kg body weight intraperitoneally, and the high dose is 500 milligram per kg body weight intraperitoneally. And in literature, you'll see many people use high dose of NAC, but never mention whether that was pH adjusted or not. I just want, for the sake of audience, I just want to mention that NAC pH is acidic. So high dose of NAC will have high acidic pH. That's why it has to be neutralized. Even in human patient, available drugs are pH neutralized. So we adjusted the pH and went ahead for the study. So as you can see here, we also took remdesivir as a control, and we evaluated NAC remdesivir also. So these are the groups that we had. And you can see the model clearly worked, and the infection showed the lung pathologies, severe lung pathologies. Both grossly it was visible, and microscopically you can see bronchitis, alveolitis, and vasculopathy was also clearly visible. So what we noticed, NAC did not suppress the SARS-CoV-2 associated body weight loss in hamster model. Rather, in some animals, we saw a little bit more decrease in body weight with 500, though it was not statistically significant. Also, we scored lung pathology by taking our 3 parameters: bronchitis, alveolitis, and endothelitis. And what the demarcating observation was that at high dose of NAC only, the lung pathology really got severely reduced. We also scored viral protein by staining with nuclear capsid protein, and also we checked the viral copy number through qPCR analysis. And what we saw that only the remdesivir was was known earlier. Also, it reduced the viral load, but in NAC group, neither in load
Your time is up, Dr. Shanti Bhushan.
Okay, we did not see any difference. And we also saw a reduction in the inflammatory genes expression, both also reduction in the inflammatory molecules presence. It also suppressed the vascular leakage as observed to the albumin leakage to the bronchoalveolar lavage. So with conclusion, we feel that the NAC and remdesivir might be a better therapeutic approach to at least control the lung pathology in COVID-19 patients.
Any
so
quick questions, please?
Thank you. Yeah. Any question, please? I would be happy to take.
Dr. Senapati, nice work actually. I just want to ask, you mean to say that remdesivir alone can control the viral load?
Yes, remdesivir alone in hamster model showed suppression of viral load, but lung pathology didn't get suppressed that much.
We have chosen the that is hamster model. Already you have this, human transgenic agent, transgenic strain.
Yeah, the hamster model we took because in transgenic mice, the lung pathology is not that robust. Though you see a lethality, but the lung pathology is not that robust as you see in hamster. And particularly the vasculopathy is clearly evident in hamster. You will not see that in AC2 transgenic mice.
Okay, thank you.
Thank you.
Yeah. Next up, we have Dr. Aurelie Jory from National Center of Biological Sciences, Tata Institute of Fundamental Research.
Hello, everybody, and welcome, and thank you for the organizers for allowing us to present our work here. And I'm so glad to see everybody again after a long COVID, let's call it a break. So I'll present today a few slides regarding how we generated these mice. And thanks to the previous talks on how to make transgenic models by Satish and this one, I don't have to do too much introduction, so that's great. So first, this is a very highly collaborative project from the different institutes on our campus, TIGS, NCS, and INSTEM. And if you want to know more about our facility, you can scan this QR code if it works. And the BSL-3 work was done in collaboration with Raghavan at ISC and their partner Incubate Mindbat. So like I said, we have set up over the years a very nice transgenic facility on campus. And as you can see, our number of new model generations has increased over the years. And in 2020, there was a big spike related to these COVID models that we have generated. So I won't go into too much details. We've generated different types of models. I'll focus on the first one here, which is the K18ACE2 transgenic model, thanks to the generous donation of the constructs from Paul McRae, who had actually generated a similar model in 2002 from the first SARS pandemic. And then using CRISPR-Cas, we generated some ACE2 knockouts and a knock-in, meaning that we're replacing the mouse ACE2 sequences with the human ACE2 sequences. And this I'll talk a little bit about at the end of the talk. So for the K18 transgenic, here you have Divij and Samya, who were working hard during the pandemic, and we actually generated over 6 founders of these lines. And so first we had to characterize the different copy numbers of transgene in these lines, and they did a lot of molecular biology. And at the end, we isolated these 3 different lines that had a low copy, a medium copy, and a high copy number of this transgene. And then went on to characterize the expression of the transgene in different organs. And we had in the medium copy and a high copy and low copy different variations of organ-specific expression levels. And I'll go, oops, sorry, well, okay, we are here. So here you can see, thanks to the work from Raghavan at ISC and in Mindbat. So what you're looking at here on both panels is the body weight loss after infection. So they use their ABSL3 over there. And after infection, here you have the comparison between the low copy and the high copy and the transgenic net controls. And we can see that the high copy lose fast, body weight very fast, and they end up dying at 6 days. But interestingly, we have also this medium copy, which did have onset acute phenotype after infection, but much later and recovered a bit. So here with these 2 different types of models, we, with the high copy, have a really acute and severe phenotype that we can use for vaccine assays. And the medium copy, which survives and has more prolonged phenotypes, mimics more the long COVID phenotypes, and then can be used more for therapeutics. So this is some staining done by Say and Aditya at INSTEM using these mice that were uninfected and compared to the infected. So in green, you have the viral particles, and in red, you can see that the viral particles were seen both. So this is a lung, by the way, in the secretory cells of the lung, in the alveoli cells of the lungs, and there's other states. You see that they were kind of broadly expressed in different cell types of the lung, but at different levels, interestingly. And here phenotypically, so here the uninfected versus the infected. Again, you see the viral particles in green. And if you high mag, you can see that in the airways, where you usually have these secretory cells that lie on the side of the airways, many of these you have holes, and the cells are actually dying and being raised. So over the last few months, thanks to the hard work of Shubha, who has standardized the production, we've been able to redistribute many of these things in HMAC cohorts to different institutes in India, IIC, ILS, and others. And IIC, they have actually used this to test some of their vaccine formulations, and they show that their vaccine formulation do confer protection against infection, whereas the non-vaccinated have severe phenotype. And I'll leave you with this slide, which is our future perspective, where we actually now want to use these humanized mice to do more precision medicine assays and use the transgenic technology.
Your time is up, Dr. George.
Compared to be able to humanize the genome as well as the immune system to be much more translational in terms of testing different.
Any quick questions, please?
So yeah, this is being done at ISC with Raghavan. So this is ongoing and it's kind of like under a patent and everything with Mindback, so I can't disclose too much, but hopefully I'll be able. No. So when you do random transgenesis, so the insertion site will be different. We have not characterized our insertion sites, but we've made 6 different founders, and so all 6 will be different. And so as I showed, depending on the founder, you have different levels of expression, you have different even organ-to-organ expression levels. So even though the transgene, the construct is the same, these models actually have different properties, which is actually interesting because like I showed, you can use some log copy number that have the animals don't die. So you can use these models for therapeutic approach for testing molecules and things like that. Because if your animals die, then vaccine is fine, but you cannot assay therapy approaches. Yeah, so we received a request from Syngene to make a conditional knock-in. So that's ongoing funds depending.
And we can move to the next presentation.
Next up, we have Dr. Sachin Singh from CCMB Hyderabad.
PPT movement. Shall I start? Thank you, organizing committee, for giving me this opportunity to present my work here. Topic of my work is metabolic syndrome-induced inflammatory conditions, impaired neuronal and astroglial metabolism in different brain regions of the leptin receptor, DBDB mice, popularly known as DBDB mice. You know, the metabolic syndrome is the cluster of the abnormal physiological conditions. In abnormal physiological conditions like increased blood glucose, hyperglycemia, hyperinsulinemia, type 2 diabetes. The appearance, body shape, it becomes the apple shapes, yeah, pear shapes. Or there are the various etiology of the metabolic syndromes like genetic and environmentals. Genetic factors, a lot of genes are responsible for metabolic syndromes. Environmental factors like stress, diabetes, or sedentary lifestyles. Metabolic syndromes induce so many disorders. I'm specifically focused on the cognitive neurological disorders. The hypothesis of my work is to investigate the brain region specific neuronal and astroglial metabolic activity in the DBDB mice. I investigate the potential metabolic and molecular mechanism linked with the depression and anxiety-like phenotypes. So my approach used to 13C NMR spectroscopy and another molecular-based approach. So this is the typical DBDB mice, the obese one. And because of the absence of the leptin receptor in the hypothalamus, the leptin hormones keep secreting, and due to this, they keep feeding and become the obese. So the approach is like that. If first we anesthetize the mice and do the tail vein cannulations, and then take out the blood for serum analysis, and then dissect the brains and make lysates and go for the NMR spectra recordings. So in case of the DBDB mice, as you know that the blood glucose level is quite high, insulin level is quite high, triglyceride level is quite high, GTT, ITT response is poor, and this is the perfect metabolic syndrome models. And then we tested behavior test for anxiety and depressions. For anxiety, we tested elevated plasmage, and you see these mice develop anxiety. For depressions, we test force swim test. So these animals are depressed, and I would like to introduce for for NMR recordings, we infuse beta hydroxybutyrate. You know that beta hydroxybutyrate enter into the TCA cycles and where intermediates of the TCA cycles like alpha ketoglutarate convert into the glutamate, and glutamate efflux in the synaptic cleft, then uptake by the astrocytes where glutamate convert into the glutamine, then again glutamine come into the synaptic cleft, convert into the glutamate, and this glutamate taken by the neuron. This is the typical models to record the spectra. And as you know, in the brain there are two types of majority of the neuron. One is the GABAergic neuron, that is the inhibitory neuron, and other is the glutamatergic neuron, that is the excitatory neurons. There is the balance between the GABAergic neuron and glutamatergic neuron. If there is imbalance, then the disease progress. So I infuse the beta hydroxybutyrate and take out the different brain regions, and I measure that the cerebral metabolic rate of the oxidation of the beta hydroxybutyrate in the cortex, hippocampus, thalamus is quite high in the DBDB mice in comparison to the control mice in both excitatory neurons and inhibitory neurons. And you see the concentration of the glutamate C4, glutamine C4 is quite high in DBDB mice in comparison to the wild type mice. To record the activities in the glia, we also followed the same strategy. We infused the acetate. You see the acetate concentration is quite high. Glutamine concentration is quite high. To further how it is going to happen, we measure certain metabolic enzyme expression of like E2, GAD1, GAD2. These enzymes are involved in the metabolisms of the glutamines. It is quite high in. And similarly, you see the interleukins are quite high in the DBD mice due to the metabolic syndrome-like conditions. And we try to link how it is happening.
Your time is up, Dr.
So this is the conclusions and acknowledgement to all the people who involved in this.
Any quick questions, please?
No, first I used this knockout model, but diet-induced model obesity might be, they will give the same kind of the phenotype, because the ultimately aim is to induce the metabolic syndromes, either genetic basis or diet basis. Once obesity setups, syndrome-like condition setup, then how it is affecting the brain functioning, metabolism, it will give the more or less, I presume, the same kind of the phenotypes.
Did you use any control drug?
Not yet.
For comparison?
No. Because this is very tedious work, so it takes almost 5 years to generate required number of the populations, and DBDB is very poor breeders. So next we can go for resveratrol to see whether the resveratrol can reduce the effect of the metabolic syndromes.
Thank you so much. Next, please.
So the last presentation for in this session will be by Dr. Poonam Arora from SGT University, who will be joining us online. Hope she's. Can you unmute? You are on mute, Dr. Poonam.
Madam, you have to unmute yourself, please. Yeah.
Now, hello. Yes.
So you have 5 minutes, ma'am.
Yes, ma'am. Yes, ma'am. This is Dr. Poonam Arora from SGT College of Pharmacy, SGT University, Gurugram. Thank you, everyone. The team of 10th International Conference by the LASA to allow me to present my work over here. The topic of my title of my work is that autoimmune induced develops allergic asthma by modulating T cells mediated immune response in experimental blister rats. As everyone knows, that allergic bronchial asthma is the chronic inflammatory disorder of the airways with the multiple heterogeneous immunopathologies characterized by the clinical features of bronchial hyperresponsiveness, airway obstruction, and airway inflammation. It is an allergic response due to the exposure of sensitized people to environmental factors or other metabolic or other disease-related factors. As the main target, due to despite large molecular cellular research in this field, the disease, however, has remained underdiagnosed and undertreated, due to which it needs a lot of more research in the particular field, and where in vitro and in vivo models of experimental animals have been already well developed, and in the future also more required in this thing. The objective of my work is was to develop an experimental allergic asthma in the Vista rats that allow induction and quantification of airway hyperresponse with quantitative variables. Rat animal, I have taken the rat animal model of asthma because of its ease in handling, collection of higher amounts of the sample, and easy in mimicking the airway hyperresponsiveness and inflammation. There are a number of species: brown Norway rats, albino Vista rats, Pragdolian, Fisher, and blues are widely used, among which brown Norway and albino Vista rats are specifically used for inducing asthma. And here I have used the albino Vista rat model of during the study, ovalbumin was used as the main allergen. Ovalbumin is the protein, the major protein, about 45% of the total protein present in the chick egg white. As it can be seen from the 3D crystal structure of the ovalbumin, it consists of 386 amino acids among for the IgE binding allergen, for IgE binding with the allergens. However, it has among 386 amino acid sequence, the sequence from the 95 to 108 is responsible for its IgE binding, is the epitope for binding to the IgE in the sensitized people. So, this highlighted area is the IgE binding epitope. It mediates its immune response by IgE hyperresponsiveness, induction of T cells, Th2 polarization, and inflammatory cytokines. The main drawback with the ovalbumin is that it needs frequent injections with the ovalbumin to sustain the Th2 polarization and features of the asthma. Therefore, to overcome this challenge, I have used ovalbumin plus aluminum hydroxide, combination of ovalbumin and aluminum hydroxide model of the asthma to induce asthma in the rats, where for combination with 40 mg of ovalbumin and 2 mg of aluminum hydroxide suspension of this made, and with giving intraperitoneal injection of the day 1 and after from the day 15 twice a week. The exposure only with the ovalbumin to the rat, as it can be seen here, resulted in a production, resulted in the Th2 polarization, sustained polarization, and higher production of interleukins, especially IL-4, IL-5, IL-1 beta, and different distinguished lung pathology that is mainly for the asthmatic individuals. While observing the parameter, I have used ELISA analysis, where I observed that ELISA analysis of bowel fluid I have observed that ELISA analysis of bowel fluid resulted in significant increase the total and over specific IgE in the bowel fluid of over sensitized challenge as compared to the unsensitized control rats. While ELISA analysis of the bowel fluid and serum of both IL-4 and IL-5 also.
Madam, your time is up. You have to conclude now.
Okay, my conclusion is that the model that I have used has resulted in the increased production of all the cytokines, the major IL-4, interleukins 4, 5, and IL-1 beta, and IgE in the bowel fluid, that are the major contributory factors for induction of asthma in the pre-induction of the asthma in the experimental animals as well as in the human beings also. That this model can also be, as histopathological changes can also be observed. I have seen this, a lot of inflammatory cells were observed in the histopathology of.
Thank you, ma'am. Thank you. Any quick questions, please?
These are the models as already established. Actually, it is not a new.
Yeah.
I think what I want to ask you is how different this your model is, and why you didn't choose the mouse model, and what is the rationale for selecting particular strain of rat?
Yes, I have used the rat model of asthma, not sorry, sir, mice. I have not used the mice. It is the rat model of asthma. Yes, you are correct. That albumin plus alum model is already established, but I have used different combinations of OVA, and dose is different, due to which for inducing chronic asthma, large number of free injections are done. For a number of weeks, like 7 to 8 weeks. But in this model, I have used only 4 weeks of induction, 4 weeks of exposure of this. That way, it has reduced, cut down the induction of asthma in the experimental animals. It has cut down the time period required for inducing asthma.
So, ma'am, what symptoms did you observe when you?
Bronchial hyperresponsive, yes, sir. Bronchial hyperresponsive that I had not.
I am asking about the phenotype, about the symptoms. What you observed?
Bronchial obstruction. All the animals were they were having a lot of redness in eyes. They were coughing. Their sneezing was increased.
Thank you, Dr. Poonam.
Yes.
Thank you so much. Thanks for being here. Yeah.
Yes.
Thank you. So we'll proceed with the next session, which is 3 R's and alternatives in laboratory animals and One Health approach. To chair this session, I would like to invite Dr. Vijay Pal Singh, IGIB New Delhi, onto the dais. Dr. Vijay Pal Singh, please. Next session. So the co-chair for this is Dr. Anil Kumar, Assistant Professor, Department of Veterinary Pharmacology, College of Veterinary Science, Rajendra Nagar. So I'd request Dr. Pandey to chair this session.
So in this session, I'm going to invite Dr. Christian Laure. She is going to present on topic laboratory zebrafish history, current status, and future direction for the model.
Okay. I believe I should begin. I will share my screen and then we will go. So hopefully everybody can hear and see me. I would like to first let's see here. Say good morning, or actually good afternoon now to you from Massachusetts here in the United States, where it is very early in the morning, but I am very happy to be with you here today, even if it's only virtually, to talk to you a little bit about zebrafish as it relates to its history, also current developments, and also some implications for the One Health perspective. So I'd like to begin rather than end with an acknowledgement slide, because there are so many people to thank for contributing to the body of understanding that I have, such that I am able to communicate to all of you about this subject with any degree of authority. So I'd like to do that first. I'd also like to especially thank the organizers and sponsors of this conference, and my friend and colleague, Dr. Kahale, for inviting me to speak here today, and then to Bill Budenberg, Chanel Kohler, and Jeff Woodrick from both Xantix and Harvard for sharing, graciously sharing videos that I will use in this presentation. So I'm going to tell you the story about zebrafish. Many of you probably are familiar with it, but perhaps many of you are not. And I think it's a story worth sharing, because the story of how zebrafish became the model that it is today is a fascinating one, but it's also relevant in terms of the reasons why it became the model that it is today are very much still relevant today, and very relevant to the One Health approach, and also to the future application of this model, which is something that I'm very, very interested in. So the zebrafish is a common fish, maybe common to many of you, in fact, because it is from that part of the world. It is a common fish that perhaps many don't pay attention to. But like many other species in this part of the world, is evolved to be amenable to change and tolerant of a wide range of environmental conditions, and in part because of that attribute, it found its way into the aquarium trade, and it's been in the aquarium trade for 100 years. And the aquarium trade is where it first caught the attention of scientists like George Streisinger, that ultimately brought it to the laboratory animal species that it is today. Now, George Streisinger, if you are not familiar with who he is or who he was, he's unfortunately passed away at an early age, but not before he made significant contributions to basic science, in particular through the development of the zebrafish as a research model. But in the late 1970s, he was a scientist with a particular problem. And his problem was that he really wanted to understand what genes control embryonic development, ultimately in humans. And obviously we have a whole host of animal models to study this process in, but he was particularly ambitious, in particular the mouse, which is the gold standard. However, what he wanted to do was particularly ambitious in the sense that he wanted to essentially knock out every single gene in the genome of a model organism, and then dissect each of those genes' contribution to that particular process. Not a very easy thing, and certainly logistically difficult in the classic model, which would be the mouse. However, Streisinger had done his early work in very simple model organisms. In fact, viruses is what he did his early postdoctoral work in. But what he wanted to do was find a model organism that would allow him the greater flexibility of a lower model organism, so that he could do things at scale and with speed, like knock out every single gene in the genome and quickly dissect its contribution. The particular interesting feature of this story is that while he was a scientist with this particular problem, he also happened to be a fish hobbyist. So he was always fascinated with fish, and he was one of those people that kept fish, hundreds, if not thousands of tanks of fish in his house, in his garage. And so he was always interested in fish, and he made the connection that perhaps fish were the answer to his particular problem. And so he went essentially to the local pet store and chose the zebrafish to be that model organism that he was actually looking for. And he chose it for a number of different reasons, many of which you're probably familiar with. But again, it's worth spending the time to go over those basic things because I think it illustrates some important lessons. Number one, and the most important, is the genetic similarity to humans. Here you have a simple graphic that I've used for years to show some of the most important model organisms that contribute to our body of understanding of how human biology and disease works, starting from the nematode worm, C. elegans, Drosophila, mouse, non-human primates. And the degree of genetic similarity is shown below. The zebrafish is 70 to 75% of the same genes doing the same things in the fish that they do in humans. So that high degree of genetic conservation is a hallmark feature. External rapid fertilization. This is just a video. If you've never seen this, this is a common attribute of the zebrafish is in its spawning behavior. External fertilization of embryos, which again, if you think back to what Streisinger wanted to do, is he wanted to watch and control and understand what genes were impacting the formation of the vertebrate embryo. Easy to see from the moment of fertilization because it happens outside the mother, no parental care. And you can see those embryos are being fertilized and deposited to the bottom of that tank. This is also something that happens with regularity. A sexually mature zebrafish can reproduce on an almost daily basis, and they can produce hundreds, if not thousands, of embryos per individual female over their reproductive lifespan per spawning event. So many iterations of embryos that are immediately available to study from the moment of fertilization on. And they're also very easy to handle, very sort of amenable to handling. You can put them in a flow sorter. You can do many things with this robust embryo, which again can be produced at high volumes. It's also transparent. This is a video of the first 24 hours of zebrafish development sped up for your viewing pleasure, but it illustrates a number of other features. Number one, it's optically tractable. So from that moment of fertilization, with very basic microscopy, you can observe and ultimately manipulate the embryo as it develops, and it does it very quickly and in a way that's very easy for you to observe. This again is first 24 hours of development. So this is happening again at scale, rapidly, easily observable for scientific manipulation. This transparency continues. This is just a head of a 4-day-old or 5-day-old zebrafish larvae. You can see, even though the resolution of this video is not tremendous, you can still observe the transparency of the organism and sort of your ability to assess the function of the animal and the anatomy of the animal through the skin. You can see the 2-chambered heart that's beating there. If you look a little bit carefully, you can see movement there on what's left of the yolk. Those are red blood cells that are in circulation, but you can really see almost every process happening in the animal in real time. And again, this is 4 days post-fertilization, so it's very rapid assembly. Again, very easy to observe and manipulate with minimal disruption. And this transparency actually can last throughout the lifespan. So we have transparent lines, the genetic lines of fish that are stable mutant lines that, if do not produce all the pigment types, with the result being that you can see into the body of an adult fish throughout its lifespan, which is, in terms of a vertebrate model, it's an unmatched feature in terms of your ability to do imaging experiments. It's also very small. So this is a typical zebrafish facility. I know some of you have seen and are very familiar with this, but perhaps some of you are not. But this is an incredibly, 1 to 2 inch, 4 to 5 centimeter fish at max. You can fit many thousands of them in a small space. So in square footage footprints at facilities at Boston Children's, we have more fish in one small room than we do all other animal species on 2 floors of a vivarium combined, all in that one small space. That space efficiency is really powerful as well. They have incredible regenerative capacity, so you can damage a number of tissues and they will regenerate fully, including the brain, retina, heart, liver, pancreas, spinal cord, and of course the tail and caudal tail, all fin types. So this is an incredibly powerful system. And for all of these reasons, Streisinger ultimately chose the zebrafish to be his model of choice. And he and members of his lab set sort of the stage and built the basic infrastructure from which all subsequent work in the zebrafish has followed. And for many reasons, it really is the model that combines features of lower model organisms, the tractability of lower model organisms like flies and worms, but combines them with features that are more, comparable, obviously, to the gold standard, which was just the rodent, and gives us a very, very powerful organism that has very high fecundity, low cost, lower regulatory burden, lower genome size, a high degree of genetic similarity, a lower ethical and public concern. That those are not sort of absent, certainly, but there is less pressure on the model from that standpoint, fast generation time, small size, as we said, and of course that external development, regenerative capacity, and transparency all made this choice by Streisinger really a prescient one. And today, 50 years later, we have fully sequenced genome and an arsenal of genetic tools that allow us to manipulate that genome really in any cell, any tissue, at any time, in any way, particularly with the advent and developments in the CRISPR-Cas9 technology system. We have just incredible ability to manipulate an animal, a vertebrate model organism, to provide valuable insights in a short amount of time and for a relatively small cost as compared to other model organisms. So again, the vision of Streisinger, which was the features of a lower model organism that combine with a higher model organism closer to human really have been realized. And we have thousands of transgenic lines, particularly that have been engineered with fluorescent reporters so we can visualize function. So you're just looking at videos of an organism, of a 5-day-old zebrafish that has been, that is expressing green fluorescent protein in the blood vessels, red fluorescent protein in the erythrocytes. And so this is just illustrating sort of the type of tools that you have that combine all of these features and really allow for a unique ability to visualize biological function in a vertebrate model. Obviously, given all of these technologies and these developments, we have libraries of thousands of models, zebrafish models of human disease. These are just 3 different examples of these that have been developed in labs at Boston Children's and Harvard over the years. but these are some of the more important diseases that impact humans today, and we have really wonderful models of them in zebrafish that have been used to both elucidate function and the biology of these diseases, but also to look for new clinical solutions to these problems. We have, over time, have developed ability to phenotype animals in a way that is comparable to what you would see in other model organisms, particularly mice and rats. We have the tools and technology to do that now in zebrafish with increasing prevalence. And so this is just video of a swim tunnel that will allow us to visualize and assess various physiological function in zebrafish that in some cases have been genetically modified, some cases they have not, but this just illustrates one of the phenotyping tools that we have. Here we also have videos of both larvae on the left and then adult animals and behavioral apparatus, which tracks movement and behaviors over time. We are developing every week. There are new behavioral paradigms, behavioral tests that are being developed in conjunction with technology that is used to assess and visualize and track this data. So again, providing us with an ability to phenotype these animals with increasing accuracy and scale. And for these reasons, the zebrafish is emerging in drug discovery, in part because you're combining all of these features, right? So you're able to, with increasing regularity and complexity and honestly lower cost and time, you're able to generate models of zebrafish models of human disease. That plus their features, the high fecundity, the ease with which you can handle the embryos. You can essentially screen thousands of compounds and look for their effect on zebrafish larval stages or adults, particularly with respect to high throughput larval stages. And again, using this increasing ability to phenotype the animal, you can really generate rich, meaningful data in short periods of time that really, in terms of drug discovery and drug development, shortens the time and cost that it would take to make really critical decisions in this pathway. And this is really extending now into co-clinical trials. So we have a number of different examples from labs around the world where we are actually generating zebrafish avatars for human disease, where patient tumor cells are transplanted into zebrafish avatars, and potential therapies are sort of tested in real time for their efficacy. And that has been an endeavor that has been generate a lot of excitement, in part because it yielded a number of promising results in a number of different instances. But it's an illustration of the power of the system that was maybe not visualized exactly by Streisinger 50 years ago, but certainly sort of his contributions and the people in his lab and their contributions all those years ago have gotten us to the point where we are now, where this kind of work can be done. And again, this is just a slide or a figure that shows you the contribution of the zebrafish to the drug development process at this particular time, where it's increasingly important for discovery and development, preclinical research in terms of safety and toxicology, and again, even some of the co-clinical trials that I just highlighted before with zebrafish avatars for human patients. For all of these reasons, the zebrafish is still rapidly growing. If you look at the number of publications that are generated each year in indexes like PubMed, this is a clear trend that is only accelerating over the last decade. So you can really see that it's growing to the point where the zebrafish in most countries where basic research is done, it's the second most used organism in basic research. And this is just some usage statistics that come from the UK, which is one of the countries that has the best statistics of this type, and it's fish, but really this is driven by zebrafish, and of course it still trails the mouse, which is natural and still obvious, but it highlights the prevalence of this particular organism in science today, and I think also highlights the fact that it's not necessarily an alternative to mice, but it is a complement. And helps us sort of inform studies in mice and other models like rats and other higher vertebrates. So in terms of the One Health approach, to try to link it for that, the zebrafish is honestly, it's a wonderful model for the One Health approach because it has applications in animal health, human health. And
to wind up, please.
Yes, I'm finishing up right within a few minutes here. And so it's a wonderful model for this initiative, and this is indicated by some of the growth that you see in some of the key areas of the One Health endeavor, both in terms of climate change, food safety, infectious disease, ecotoxicology, drug development, and antibiotic resistance. These are just fold changes in the number of publications over the last 10 years. And you can see that it's matching that growth that I showed you earlier in the general total growth of the model, and in many cases even exceeding it. So it's really there's a lot of potential here that at the end of the day is going to be limited by things, and this is where I'll close for this audience, the laboratory animal scientists. These are the things that limit the zebrafish in its utility as a model going forward. It's obviously great potential, but in all of these areas, in welfare, the physical environment, nutrition, housing, genetics, and pathogens, all of these things that laboratory animal scientists are concerned about, these are the things where we need to have breakthroughs and movement from the lab animal science community to help drive improvement so that the model can be realized. And in closing, what I'll do is just highlight one quick thing, one critical need in each area. So for physical environment, we have to characterize and define water quality variables, nutrition, define diets, housing, we need to redesign to support and meet science. Genetics, we need to find genetic lines. Pathogens, we need to develop SPF stocks and ways to maintain them. And then we need to use data-driven approaches to improve well-being of animals. So with that, I will end and thank you for your time.
Your time is up, Chris, please.
Yes.
I request you.
So thank you.
So thanks, Chris. That was a wonderful talk and overview about the zebrafish model. So we are running short of time, so maybe two quick questions from the audience. Are we audible, Chris?
I don't hear the audience, but if there are questions, I'm happy to answer them.
Yeah, so we have one question from the audience.
How many models are acceptable to FDA? Say the data, suppose you generate data from these models, will it be accepted by FDA?
FDA, how about FDA acceptance of the data? The question is.
Yeah, how accepting of the FDA is the FDA to ecotox data and zebrafish? Is that the question essentially?
Yeah, that's correct.
Yeah, so there is growing acceptance, and there is for instance, a consortium of companies in pharma space that are developing a paradigm for this that is growing. That I don't know where it is in terms of its formal acceptance by the FDA, but there is progress being made, and there are a number of papers that summarize where we are with respect to that. But there is a growing acceptance by the FDA of the zebrafish as a preclinical safety toxicology model that has been validated by this multi-agency group.
So
And I'm happy to talk offline as well. You can always contact me by email, and I can send you information that's a little bit clearer than what I'm saying here and now, in the interest of time.
Okay, so if there are no more questions, let's thank Chris for his time. Thank you.
Thank you. Thank you, everybody.
Joining us.
Thank you so much. I wish the best to all of you.
So we are moving to the next and the final talk of this session. And the topic is Beyond the 3 Rs, Human Relevant Replacement Methods. And the speaker is Professor Muhammad Abdul Qadir Akbar Shah. Oh, sorry.
About 5 minutes.
So I don't know how I introduced you. You're such a senior,
You have
quite
something like
experienced
that, no?
person. So I saw that 50 and more years of experience. You don't need any topic. You can just, So Professor Muhammad is a faculty of zoology at Zamal Muhammad College in Tiruchirappalli. Has more than 52 years in teaching and research. His area of research includes animal science, biomedical science, and biotechnology. So he has published more than 250 research and review publications, 2 books on medicinal plants, and of course edited Laboratory Animal Science. He is currently the editor-in-chief of the Journal of Endocrinology and Reproduction and associate section editor for Frontiers in Toxicology. He also served as president of the Society for Reproductive Biology and Comparative Endocrinology, India. One of his important contributions is he established the Mahatma Gandhi Camp Center for Alternatives at Bharatidasan University, Tiruchirappalli, with the funds received from, I'm sorry, I can pronounce some foundation in Switzerland, and has been heading the alternative movement in India and elsewhere. So he is also the founder of Society for Alternatives for Animal Experiments in India, and it will be very exciting to hear because this is totally a new avenue for me at least. And presently he is the General Secretary, member working group of supporting adoption for MPS for COVID research under NICE ATM. I can read ATM very well, and NC3Rs. So, sir, I think I hope I have done fair enough job to introduce, but your 52 years experience is good enough. It's a big introduction. So please. So it is a formal request, but as a chair, I have to do it. So you have to stick to the time.
It's really a
floor is all
I'll try
yours.
to do that. So very good afternoon. At the outset, I would like to thank the chairperson for the very profuse introduction. But my CV is so huge, it will be really tough for one to handle it in 2 minutes time. Well, anyway, I thank Gerald and Dr. Vijaypal and all friends in the LASA for inviting me to this conference. And this is maybe 4th or 5th time I am addressing the LASA association in its meetings. Today, not to lose time, I will quickly move into the topic. What I intend to do over these 18-19 minutes is to very briefly tell you about the 3 Rs: yesterday, today, and tomorrow. You may ask me why, what is very funny about it, yesterday, today, tomorrow. Well, I have been in this alternatives domain for well over 20 years, and I have understood the 3 R's over the years, not to begin with, over the years, looking at reading through the 3 R's book of Russell and Burke, I have realized that the way things have been presented and published, we have not rightly understood what Russell and Burke really had in mind and real translated in their writing in the 3 R's book. Well, I have to necessarily, for the interest of the students and even to a certain extent the senior scientists, start with the beginning, because I am going to tell you about the alternatives yesterday, today, and tomorrow I mentioned. You look at the yesterday, the way Russell and Burke really did, and look at today and tomorrow, the way Russell and Burke expected to happen. So I think I would like you to keep this in mind, to tell you briefly what Russell and Burke did yesterday, really did yesterday, and to look at what they intended us to know, us to do today and tomorrow. So 1954, the Universities Federation of Laboratory Animals in the UK thought about bringing out a monograph on laboratory animal science material, and they initially nominated William Russell to do this endeavor. It was actually a society and organization. So Russell was asked to do it, and Russell later felt he would like to have support of another person who would go around and collect data. So the society put Rex Burke into the team. So it was Russell and Burke. And in 1959, they came out with their product, the publication, The Principles of Humane Experimental Technique. The book, as it was published, it is shown on the right. Well, the 3 R's were originally proposed by I mentioned that originally. So there are 3 major recommendations, in the sense that the whole book speaks about the 3 R's, the replacement, reduction, and refinement. We have understood replacement, reduction, and refinement the way many reviewers have, authors have written in the course of time. But what I'm giving here is the quote. What Russell and Burke meant as replacement is, within quote, substitution for conscious living higher animals of insentient material. Not use live animal, animals who feel the pain. Use insentient animals. Material. Reduction in the number of animals used to obtain information of given amount and precision. Which is very particular about this. Reduction in the number of animals used to obtain information of given amount and precision. Refinement, any decrease in the severity of inhuman procedures applied to those animals which still have to be used. If at all you are going to use, intend to use, important to use animal, use it the way severity of humane procedures applied. So Russell and Burke described them in this order. The order is very important. Why didn't they not put reduction first? Why didn't they not put refinement first? Why should they put replacement first? So there is their intention. Now Russell and Burke described them in this order, reflecting the order in which they should be addressed. That sentient animals should not be used if non-sentient alternatives are available. They started that. Sentient animals are not to be used if non-sentient material is available. If animals do have to be used, then the project's design and analysis should be such that the minimum number of sentient animals are used. Minimum number is always adapted, but there's more than that. The project's design and analysis should be such that we use the minimum number of animals. That appropriate measures should be taken to mitigate any pain, suffering, or distress that the animals might feel. This is practically the replacement. Well, little elaborately, to tell you what Russell and Burke had as knowledge in that time, as these 3 alternatives, replacement, reduction, and refinement. So Russell and Burke, they had a vision like in vitro studies. That was the time when just cell culture and tissue culture just starting, so they thought the alternative, the replacement alternative would be in the form of in vitro studies and the relative replacement using invertebrate models. The earlier speakers spoke about the zebrafish, that is practically relative replacement. Replace the hair sentient animals with the zebrafish or C. elegans or Drosophila. Different approaches are going on. And then for reduction, you apply statistical methodology, do power calculations, fix the absolute number of animals minimally required to conduct experiment, accurate design of experimental plan, harmonization of experimental procedures, reuse of animals. And a refinement, use of genetic models, consideration of individual variability, choice of the behavioral test, skilled experiment, and most importantly, improvement of husbandry. This is very conveniently forgotten in our current work. Animal husbandry, of course, I am aware that some of our friends here are highly conscious about it. That's one of the reasons why we established the society to discuss about it. But look at it very, very widely, not much concern about the animal husbandry. Well, why should Russell and Buck put replacement first, reduction second, and refinement last? So prioritizing replacement in the year of 21st century technology is simply an extension of Russell and Buck's thinking that reduction and refinement are interim steps. For Russell and Buck, reduction and refinement are interim steps. They were ultimately aiming at reaching a target of total replacement. So I repeat, prioritizing replacement as we envision today, the year of 21st century technology, is simply an extension of Russell and Buck's thinking that reduction and refinement are interim steps on the path towards replacement. They wrote, refinement is never enough, and we should always seek further for reduction and, if possible, replacement. Still more generally, replacement is always a satisfactory. That is what Russell and Buck said. They wanted to disclose. Ultimately, we should lead into total replacement. That was their aim and objective. They refer to the distress the animals would experience in the laboratory. Whatever we do, modality to adapt, reduction and refinement, well, there is still inhumanity. They said there are two kinds of inhumanity. They mentioned direct inhumanity could result from unavoidable consequence of the procedures employed, no matter how well defined, and contingent inhumanity that could result as incidental and inadvertent byproduct of use of these procedures, which is not necessary for its successes. So, in their view, it was better to avoid both types of inhumanity by using replacement methods when available. So having said about it, coming to today, but looking at the use of animals, status of animal use since 3 hours, are now the present. The more than 127 million vertebrate animals were used worldwide for scientific purposes in 2004. More than 127 million vertebrate animals were used worldwide. I mean, that too from about 24 countries only, but still we claim it is worldwide. The number has increased further since then, estimated 192.1 million in 2015, again referred to the authors. So this function of animals are still used in spite of the 3 hours that came up in 1999. This is the data for 2015 and 2005 and 2015. So animals continue to be used not only in high numbers, but more than the number of animals that were used in the past. So you need to have novel, stringent, straightforward genetic modification techniques such as Crispr that likely lead to further increase in the number. This is not including the genetically modified vacuum mice that are used in experimentation, that are otherwise the wild, the animals, laboratory animals that are routinely used. So basic and applied research accounts for most of the animals used in science. Basic applied research. So the production and quality control of devices and products used in human medicine and dentistry. There is one major use of animals, one avenue of use of animals, and toxicological and other safety tests, among others. If you look at the data, the number of, the relative percentage of use of animals in biomedical research is about 90%, and in the toxicological research is about 10%. More details, but I don't have time to explain that. The high number of animals used in biomedical research warrants additional emphasis to replace their use in these fields. In drug discovery, in instrumental discovery, and so on, in testing. So we are using enormous number of animals in biomedical research, but equally, a good number of animals are used in the toxicology, also 10%. So I make a comparison of the use in the biomedical research and toxicology, which is skip. So what would be the replacement techniques envisaged by the principles? That going back to 1959, when Russell and Clark wrote the book and published it, they should have had something in their mind and even written something about what would be the kind of replacement material that would arrive. They are the insentient material. So they said, Russell and Clark, mammalian tissue cultures have become, since the Second World War, one of the most important replacement technique, and indeed one of the most important developments in biology. So they envisaged tissue culture. The movement is 1959. We are in 2022, and the developments since then has been exactly ending with tissue culture, cell culture, in its different dimensions and its perceptions. So then they mentioned about homogeneous two-dimensional arrays, short-lived in vitro preparations, and a relatively improved reconstructed tissue, all in vitro. So that is what they envisaged in 1959, and this is what we are heading towards. So, what did they envision? This is the most critical statement I am going to disclose to you. As new fields of biology open in the future, please try to memorize this statement. As new fields of biology open in the future, it may become a matter of routine to apply the lessons of the past and turn as soon as possible the techniques of replacement. They were ultimately very clearly directed towards replacement and open up. As new fields open, there will be new technologies coming up, new methodologies coming up to make it possible the replacement to happen. So this is what I started off saying, that to tell you what Russell and Clark wanted to say, intended to say in their language, and this is what you should also be able to endure. Well, what is that? Now I'm linking the previous slide and this slide. What the technology will bring up in the course of time, can we take the replacement material? Okay, what are all the materials that are available? We have now the scientific revolution in every field of science, in biological sciences, and in alternative science also, there's been a real revolution. So you have now organs on chip. You can see the pictures there. Organ on chip, the Wise Institute in Harvard Medical School brought up with a very novel technology of in a microchip in a hydrophile. Get replaced with microscope, and you can look at 4 or 5 organs are captured in wells inside this model organs on chip. That is a real revolution. And then we get the organoids, 3-dimensional organoids. That's what I have been involved in research in over the period of a few years. So the next picture is there, what you call as the organoids, 3-dimensional organoids. Earlier it was all 2-dimensional ones, flat cultures. And the time came when we started off doing work with the 3-dimensional models. We call it 3D spheroids. In monotypic or heterotypic, one cell type, you can all put together into one small sphere, microsphere, that would represent one particular liver or a lung or a brain or an intestine or kidney. So there is organoids, and the latest entry into this field is organoids. You can put together organs on chip, different organs on chip, and there's an organ on chip, the spheroid is there, and you can put several such spheroids of liver, lung, and intestine and kidney in different wells in the plate, and that becomes an organoid, which is the I am taking to the microphysiological systems. So high content imaging technologies that have come into use, we can combine it with this DNA and RNA microarray screening, add it to that, put together all. So And you have more additional scientific development contributing to the alternative scenario, computational tools and approaches. And you have now machine learning and artificial intelligence with big data. So every development, technological development sense over the period of time have all been brought into some contribution to the alternative scenario. And last one is to have what we call as the automation. So the machine can do all the things that are required of analyzing toxicological effects or pharmacological applications. So that's again from Weiss Institute, the automation. I have now the 21st century technologies offer a number of advantages of 20th century. So you have increased speed through all techniques, throughput, which is showed in the machine, and biological relevance. So you have 3D architecture. Earlier it was all 2D. I did a lot of 2D, and now I changed it to 3D. So you have now change of to 3D architecture, fluid exchange, and enable the modes of function more like human tissues or organs, and to survive in culture for long time of periods. Earlier, keeping the cells in the lab for 2 days, 3 days was a big issue. Now you can keep it for months and years together because science and technology has improved so fast that we have so many modalities available to keep the cells and tissues and organs and chips and organoids to remain alive for years. So owing to the development of induced, this is the most important contribution, important development that contributed for victory replacement, the induced pluripotent stem cells. So this is a clear panacea. With the iPSCs, you can develop kidney, you can develop lung, any of these organs, iPSCs. So you can say contributing factor to the alternative scenario, particularly the replacement. Thus, different organs can be connected with one another. You have the illustrations there. You put different organs in different wells, and then there is micro-canalization. So you have connection, circulation is established. So fluid and material can move between organs, and then in exchange. When I started in 2020 years back, I never thought I'm really happy that things have moved so fast, and I'm able to adapt these technologies in my research. And it means I'm very close to that. These are very important milestones, regulatory decisions that came up in USA, the toxicity testing in the 21st century, vision and strategy. These are all protection maps. And in EU, Directive 2010 on the protection of animals used for scientific purposes. So fantastic. These are two areas and two nations or countries which do fantastically towards alternatives, particularly replacement alternatives. So coming back to the title, prioritizing biomedical research and the target of non-animal methods. So the major goals, these are the major goals I skip in. We have these are again two important milestones that Biomed 21 is the policy paper that came up, which envisions the human-based pathway approach to human disease research. And then subsequently, IAMS and information is driven from observational, prospective, epidemiological, and interventional studies conducted in the human patient cohorts. I'm spend of time. I'll have to quickly pass through. This is the point. When you can proteomics and genomics, they will have overlap. You can adapt those technologies, and the expression analysis will be conducted so that organ on chip or the steroids, they can be ultimately put to toxicological and pharmacological investigations. They can look at the expression, the pharmacoproteins, and the genes or the mRNA. Well, this is a critical. Again, I don't have time. That you have now plenty of material available. Non-sentient material obtained from human. So if your interest is to find application of with pharmacology and toxicology. This is for human applications. Use human material. And human material was not available earlier, but we have a lot of human material, different kinds of human material are available. And if anybody is interested, you can copy this PowerPoint because I'm not finishing because full. This is nothing more than one of my own papers. Microdosing is another important development. I'll come to sum up with the, increasing awareness. You can put these things in the courses, teaching, and enlighten the students about the avenues that are available to attitude to change. These are all the models which technology has brought to us. I mean, these are companies. Many companies have started up making these material, and you can acquire this material and conduct the investigations. These are the beautiful organ on chip, the 3-dimensional model. So, coming to Russell and Buck, the emphasis in the 21st century is likely to be on expanding and exploring the capabilities of these non-animal methods further and using them to better understand the human biological pathways and the better patients to these pathways that can cause diseases. That is, these methods will come to comprise the role. These methods in 10 years, 15 years, 20 years, there will be an occasion when we will not be able to, we will not be using animal experiments. Without your option to you, there will be a development in the scientific community that there would be no need to use animals in experimentation. Everything to that is possible. Human nature. That is what is expected. Envisage looking at what Russell and Buck said. As things have happened over these years, tremendous, miraculous changes have taken place, and therefore we are very positive. So we have, just to tell you that I'm not speaking based on reading literature, but based on my own experience, not a few of my publications using these technologies, and my contribution. This is contribution to the 3D model in a cost affordable manner. Anybody can have access to it. I'm finishing it. And this is the 3D model which we developed. Okay. I mean, this is to be patented. And the applications. And this is another important message when it comes to community argument, that the way the science is moving so aggressively, this May 29th to June 3rd, there was a 4-5-day World Conference on microphysiological systems alone. The topic was microphysiological systems. World Conference on microphysiology happened on 29th May to 3rd June in USA. This is the proceedings, and I had to say that I am party to cannot make all claims. These are companies which make the products available when useful for this scenario. Infura is doing a lot of contribution in this field. These are funding resources available now. And finally, I would like to read this particular clause where organ-on-chip technology has been proven to predict human responses better than other testing methods. With human-relevant science now available, why do we continue to rely on animal testing and in vitro models that fail to perfect human response, predict human response? Shouldn't we be doing everything possible to relieve human suffering caused by diseases, animal models? Having said that, I want to introduce the Society for Alternate Animal Experiments, subjectives, office bearers, and I invite you, members of this society. Thank you so much. I little overstepped the time, but I also stress to complete it within the time.
Thank you, sir, for a very futuristic and informative talk. And we all hope that all the IAC, CPCA becomes irrelevant soon and switch to whatever new technology that is coming up. So with that, I think the questions, if anybody has, we can ask during the lunchtime. So in the interest of time,
Even you can contact me over email ID. It is there in the last slide. You can get it. They're all, but I'll be available to answer your questions. Guide you, help you, support you in the endeavor of alternatives. I thank the organizer, thank the chairpersons, and thank colleagues for working to organize the lab coverage. Thank you so much.
Thank you, sir. I request the chairperson of this session to felicitate our speaker. Dr. S.K. Datta from Animal Welfare Board is here. We welcome you, sir. And to felicitate the chairperson of this session, I would like to invite Dr. Balaji from LASACON. I would like to invite Dr. Asad from Perkil and Elmer to give a memento as a token of gratitude to our co-chairperson. Thank you. I have a small request. I know everyone's hungry, but we have a speaker waiting from Japan. So we'll initiate the next session. We'll just have this one talk by Dr. Atsushi Riki, and then we'll break for lunch. So to initiate this session on animal model for non-infectious diseases, I would like to invite Dr. Vikas Dige as the chair for this session. Onto the dais, please. A small correction, Dr. Satyadev onto the dais, please, to chair this session. And to co-chair, Dr. Pradeep Batu Patil, please come on to the stage. So we'll have this one talk by Dr. Iriki, and then we'll break for lunch, and we'll continue with the rest of the two talks. I'll hand over the stage to the chairperson now. Thank you.
Good afternoon. The next speaker is Dr. Hiriki. He will be speaking from Japan. So he is a PhD in neuroscience from Tokyo Medical and Dental University, 1986. He has held the research and associate positions in Tokyo Medical and Dental University, and then at the Rockefeller University, USA was there. He joined the faculty of Toho University Medical School as an assistant professor and then an associate professor in physiology. In 1999, he returned to Tokyo Medical and Dental University as a full professor and chairman of cognitive neurobiology. In 2004, Dr. Iriki was appointed as a head of the laboratory for the symbolic cognitive development at Riken Institute. And he is a visiting professor in the University of College of London. And he is a vice president of High Class International. In the interest of the time, I request Dr. Iriki to go over your presentation now.
Okay, thank you very much for this invitation. And can you hear me?
Yes, please.
Yes. In sake of your lunchtime, I'll go directly to my talk. So I was asked to talk about the neuropsychiatric disorders and primate model for that. Myself being a cognitive neuroscientist, so I have been studying the primate mental disorder connected to human mental disorders. And my talk is going to be why we need primate models. And this kind of primate models for mental disorders can be slightly different from conventional, the rodent physical models. And I would like to propose a new mindset or a new sets of laboratory, which has been published recently in these two papers, although one of them is under review still. So, sliders. Okay, so we are now, the biology are now ready to study the mental disorders, which is a slightly different from physical disorders or physical functions. And we need primate models because we are interested in human mental disorders, and mental functions are a function of the brain. And the human brain is part of the primate brain, and it is included in the animal cognition or in a more broader sense. And for the physical functions or biomedical research, we can use animal models because this is based on the bodily structures and function. It is common with humans in most cases, with few exceptions. But the mental functions of humans are peculiar, and this is different from simpler animals. And as being the primate brain, our mental function have some commonality with primate brain. And therefore, it is slightly difficult to directly extrapolate from rodent model or a non-primate model into the human mental function. Therefore, we need primate model for that. But the primate model, it's so why I emphasize the primate brain, I'm going to tell you in the next slides. So this is a mammalian evolutionary tree from back to 250 million years ago. And here primates and here rodents. And after the mammals have bifurcated in many species, there are these three orders are most successful orders in the mammals. These numbers indicate the number of the species in these orders. And as you can see, primate and rodents are very common or similar, or the closest ancestor among the mammals, However, the brain structure and function is very much different based on the different environment they live in. Primates live in visual world. Rodents live in auditory, in olfactory and somatosensory world, and bats live in auditory world. Therefore, the brain structure design is different. If you compare the big rodents and small monkeys, they are very different size of the brains among different species in same order. If you look at the rodent brain, smaller rodents and big rodents like capybara, they have a brain of like macaque monkeys, and their structures are quite similar. These colored areas are primary sensory areas, and these are just an expansion of or inflation of the smaller brain. But in the primate, when the primate brain becomes bigger, these colored areas become big as well, but more expansion are induced in white area, which are association area. So the principal design of the brain is different between orders. And this is the numbers in different species. These are the primates. If the primate's brain becomes bigger, the number of the neuron becomes much larger. But if you compare the capybara brain, similar to macaque brain, the number of the neuron much smaller. And if you compare with much larger brain of elephants, but it's like 3 times larger than the humans, but the number of the neurons are like 1/3. So this is a reason why to study the mental function of the humans, we need primate models. But as I said, I told you in the previous slide, primates, well, in the rodents of biomedical studies, it has to be studied in well-controlled, reduced laboratory environment. But in the primate, for the mental disorders and mental functions, we have to study them not in the reduced and controlled laboratory condition, but in more naturalistic condition. But since we cannot study mental function in the nature because of many reasons, we have to have some compromise to create a new mindset or paradigm to study the primary mental function. So this is a proposal in my talk, is that, as I told you, there are two gaps. If you compare primate model and rodent model, there's a gap because of the design of the brain. And among the primate, we have to study the mental function in laboratory, but more naturalistic, like wildlife-like conditions. But we cannot do the experiment in wildlife. So there are the gaps here. So what we actually need to study the mental function in the primate model is something very novel. I call this natural lab or in situ eco lab, or in other words, lab in nature and nature in lab. So these new sectors can bridge these gaps and probably brings us to the next higher level of the biomedical research studying human mental function. So how can we do that? So to create such a completely novel paradigm, there are many challenges, like technical challenges or ethical challenges. And to overcome those challenges, we need a new mindset. So therefore, I'm proposing here two things: a paradigm shift by dimension expansion. What I mean by dimension expansion, I'll tell you later. And also to realize that we have to propose a novel paradigm of the novel laboratory conditions. So first, about a paradigm shift by dimension expansion. So to shift from the reduced laboratory condition physical biomedicine, we have to change from hypothesis-driven experimentation to data-driven one. And we have to care about the emergence of complexity or complex phenomena. And also we have to deal with accumulation of extensive heterogeneous data. I'll tell you more in detail from the next slides. So thinking about a biomedical research we have been very familiar with. Important thing is reproducibility of data, and it has to be hypothesis-driven, very solid hypothesis we have to have. And we have to strictly define the boundary condition of the experiments. And we need a large number of data for make a statistical significance with that. And conduct such a biomedical research using animals, we have to care a lot about harm-benefit assessment of 3 R's, which we have heard a lot in the previous talk. And there are some discrepancies or gaps between these requirements. And this is very important for biomedical research. But for the mindset, when we think about a more naturalistic condition, which resemble the wildlife conservation, and there becomes a different gap between here. And in contrast to the important factor that I just talked, reproducibility, hypothesis, and boundary condition and statistics, in these wild or naturalistic conditions, we have to care about the uniqueness of the conditions. And also has to be data-driven accumulation of emergence of data, and have to deal with extra boundary complexity and emergent properties of the things. And this is quite different mindset or concept of experimentation. And in between these wild naturalistic condition and biomedical laboratory reduced condition, there already exist conditions for animal care. It's like domestication, like agriculture or pet animals. And here we have to deal with the 3 R's have to be built in these conditions, rather than we have to care about 3 R's in design of the experiments. And psychobiology or neuropsychiatric disorders experimentation somehow resemble this condition, and which is installed very much as a new component in this overall scheme. So this interception brings this structure slightly differently, like this. This is pushed this way, and here the dimension becomes a little bit more complex because we really care about these factors And this have similarity on the characteristics of data, like here, and benefit of animals and human benefit are like cross-cutting. So like this is an expansion of the dimension of the ethical space of the experimentation and become two-dimensional. And we have to think about this complex control of animal experimentation to study the complexity of human mental disorders. And now to sum up with the talk so far, we have to care about 3 R's in all the experimentation, but particularly in this neuropsychiatric disorders, we have to think about overlap of this trade-offs. And these 3 R's have to be built in this expanded dimension area to the areas of the ethical plane. And this has to be carried out by novel paradigm and new technology, which also we have heard in the previous talk. So to realize this dimension expanded paradigm, we have to propose the new mindset or novel paradigm to study multidimensional data collection and potential improvement of the primate welfare. And we have to think seriously about more ecological settings and data-driven approach. And to propose that, I am now proposing a completely novel and open field primate research experimental conditions, which in short, I call it natural park or eco zoo or eco lab or remote laboratory, which I'm going to tell you in the next slides. So in short, since the primates are tropical animals that lives in tropical area, like in India, you see many primates around yourself, but not in the northern or southern bands of the climate bands on the globe. And many laboratories are in the areas which do not have the natural primates. So with the modern remote technologies, we can share the data collected locally in these conditions. And we can set up the global laboratory conditions using modern internet technology and our transportation technologies. So I'll introduce you something I have been studied for past few years. We have been studying a kind of eco lab in Thailand before the COVID. Because of this COVID pandemic, this has been suspended for these 2 years or so, but we have studied before that. And what we have been trying to do is to establish a natural lab in this area where the monkeys using tools lives in this area. In macaque monkeys, in the previous belief is that they never use tools. But as yourselves are Indians, I think you sometimes seldom observe the monkeys using a sort of tools. And this is some representation of the human-like higher cognitive functions. And if we can find the monkeys that extensively express these unusual behaviors as a macaques, we can make as a platform of experimentation. And what we have been doing is that in this area, the other side of the Indian Ocean, in the coast of Thailand near Phuket, there are many islands that this kind of monkeys live. What kind of monkeys is that? Here are the video clips of the monkeys. They come in the family on the shore, and they use stone tools to crack oysters. And they come with the families, and small kids like this learn from the parents how to use tools. He will start using tools, but it's not good. So there are some kind of cultural learning and cultural tradition and higher cognitive function expressed in this lab, in these natural settings. So we have decided to study these monkeys as a subject of the animal experimentation. But since these areas are in the national park, we cannot do invasive study there. We have to think about a completely non-invasive animal settings, but we have to collect reliable data. To do that, the local governors, we have made a collaboration with local governors and also the national park chief and park staff and rangers. And these are the local Thailand professors or the primatologists studying this. And we have, the government have provided us with, with this building, and we are trying to create a lab here. And since this lab is at this point, it's a part of the continent, and these monkeys live around here, the other side of this small channels, and we can videotape these monkeys and also install the camera in these shores. And we cannot develop directly the modern technologies in natural settings. So what we have planned is between this laboratory settings, which I'm not going to tell you in detail here, but this is the house cage with this automatic measure of the behaviors and with the fully computer control laboratory settings. But this is completely laboratory conditions. And in between these natural settings, we created something intermediate with enriched environment. We call it the nature in the lab conditions. And by combining this modern technology, we call it, later I will introduce about the digital zoo condition, we can link laboratory in the nature. So I call it lab in the nature condition. And together with intermediate new paradigm, we decided to develop the new technology to study these primate behavior and complex cognitive function in the naturalistic settings. And since this, the nature in the lab, we have only limited space in Japan. It's not like in India. So we introduce the smaller monkey like marmoset, and combined with the modern the behavior measuring technology recently developed like a few years ago. It's a novel motion capturing technology. And this is just a simple video clip taken from the YouTube. And applying this deep learning technology, we can almost at a real time measure the movement of these video clips. So if this is online, we can measure the animal's movement in online. So, and this technology had been published like 3 years. And this is my collaborator developing the technology in EPFL in Switzerland, Professor Alexander Mattis. And their group have published some multi-animal tracking technology just a few weeks ago. And applying this, and these are small monkeys called marmosets in this nature in lab, we can monitor the behavior of these multiple animals in real time. And by the digitized animal behavior, we are now, Dr. Mattis is developing the online behavior segmentation technology, automatics, and also applying deep learning technology. We can transfer and label. This is a preliminary video clip, so it's slightly, it's not very well done, but you can see the animals moving here, and here are the behavior segmented kind of behavior detected automatically by this programs. And by this way, we can automatically compose the dynamics of the behavior transition in naturalistic settings. And we have now laser scanned this naturalistic environment, and we can fully digitize the environment. And by combining multi-camera, then we can 3D reconstruct animal behavior. And in this way, we can fully digitize the naturalistic animal conditions in the primate conditions. And this digitized, the data can be transferred via internet technology, by the cloud technology, to all over the world. And the labs in anywhere in the globe can do the experiment of observation in the natural setting on site in the natural habitat of the monkeys.
I think your
yes,
time
I'm
is
running
up.
short of the time. Okay, so I'm introducing this natural lab here with a modern technology, and we can collaborate with the local collaborators. And most important thing is that this is a multi-sector collaboration with mutual understanding and respect with each other, especially with husbandry people and scientists using that. The experiment is part of husbandry condition, and husbandry is part of the scientific work. And this probably is a common concept with the One Health application of the biomedical study. And thank you very much for your attention.
Thank you, Dr. Ireki, for the insightful talk on the non-human primate and the natural settings as well as the laboratory. So I request the audience if you have questions. Any questions from the audience? If there are no questions, I think there is a question. Yes, please.
Hello? Hello, can you hear me?
Yes, yes, I do.
Are you applying a chip on the brain, or how you are doing the behavior means
tracking?
Behavior tracking.
What do you mean by chips in the brain? You mean the brain, like a brain machine interface to record the neural responses?
Yes,
or chips to identify the animal ID?
Identify to animal ID plus in suppose that in situ if different animals shows different kind of the behavior, then how you record in that condition?
Well, in the nature in the lab condition, the animal already have the chips. So we can have animal identification. And also with this, the video tracking system, by the visual appearance, the program can identify the individual animal as well.
If the different animals shows different kind of the behavior in the same setup, then how you interpreted those data?
So that's another thing that I didn't explain this time. So we can apply the complexity dynamics technology to study the dynamical transition patterns of the behavior. And so in this way, it is called in the complexity in the complexity study field, it is called the tipping point. So we can predict the transition of behavior before it is expressed. It's like a pre-symptomatic diagnosis of the behavior patterns. And this is another subject of the science, to how to deal with this data. So today I have told about the settings of the national setups. And from these setups, there are the way to interpret those data. And these are completely different from conventional statistical significant kind of the paradigm. Does this answer your question?
Yeah, thank you.
Okay. Thank you, Dr. Ireki, for your presentation.
Thank you very much.
Thank you very much. I'll hand over the mic to the next.
Thank you. We'll break for lunch. Quick lunch, 30 minutes. Thank you, sir. Chair and co-chair, we'll resume after lunch. Thank you. Let's.
I request all, please assemble again for the next session within half an hour, please. 30 minutes. Put the timer there, Chandu.
ఫోటో తీసుకున్నామా రీసెంట్ తీసుకున్నాను బాగా ప్రెజెంట్ చేయి అవార్డు వచ్చి
Good afternoon, all of you. Once again, we'll have this session 9, animal model for non-infectious diseases. I'll request Dr. Vikas Dige to come on to the dais, chair the session, and Dr. Pradeep Bhattu Patil. Pradeep. Thank you.
Good afternoon. So let's begin the session. So I invite Dr. J. Kwang Sang, who will be, he's a professor at College of Veterinary Medicine at the Seoul National University, as well as the director of the Korea Mouse Phenotyping Center. So let's hear him about his experiences. Yes, Dr. J, you can start the session.
Well, can I start? Okay, thank you for your
Yes,
inviting.
Dr. Singh, you have 18 minutes to complete the presentation, please.
Okay, thank you. Yes, today's my talk title is, mouse metabolic phenotyping and a mouse model. Briefly, I introduce my background and I want to share my experience and knowledge in this field. Also, very great appreciation for inviting me to this session. I also have a very good memory with you because I was already invited by your organization. Also, we have some AFLAS meeting in 2017. Also, I remember your team already wonderfully organized the AFLAS meeting in 2018. Also, I was invited at the time. So briefly, before I start my tour, I'd like to share this information. Next AFLAS Congress will be held in September 2023. Please join and enjoy your great fun with us. Recently, the entire genome information has now been published and whole genome sequencing is becoming very quick and cheap to complete. However, still, despite this advancement, still the function of the majority of genes remains unknown. So we really need a mouse model for elucidating gene function in vivo level. This is one of the examples. Left side mouse is a little bit better because this mouse is just a leptin mutated mouse. We are interested in mouse phenotyping instead of just gene sequencing data. So even though this mouse was already introduced in 1952, however, the leptin gene was, fully sequenced in 1994. That means, even after we understand the genome sequencing data, still we needed to understand the gene function in vivo model, in vivo level. This is one of the examples for making this, realized by several, core organism meeting. This is IMPC, International Mouse Phenotyping Consortium. The concept of this approach was already introduced in Nature 2010. And we wanted to make a single gene knockout mouse, and based on broad spectrum mouse phenotyping data set, maybe we can share the encyclopedia of gene function in mouse level. Also, this data was already released to the community and everybody can easily access. So IMPC already, we make more than 8,500 genes phenotyping data. Also, based on this basic phenotyping data, several publication highlighted is rapidly increased, including Nature 2016, and also Nature Genetics in 2017. And more recently, 2021, we published a resource of targeted mutant mouse lines for more than 5,000 genes. After that, we just focused, for example, in several organs, for example, sexual dimorphism, also just bone mineral density, or also large-scale hearing loss, and also just circadian mismatching, and also just to disease gene discovery genes, also including metabolism. So that means this kind of approach is very powerful for catch up the gene function in vivo level. Nowadays, second, issues was already studied as, in 2021. That's a function of human genetic variation. That means in this period, the key goal is to deliver broad-based genomic insight into the function of human genetic variation. Because even though we can use, just a non-mutation in mouse models, however, in human, there are several types of genetic variation. So if we wanted to understand each variations, meaning, we have to make each gene types of genetic variation in same of mouse model. Also, this kind of information will be spread out just the internet. Okay, let's start, just metabolic regulation. Simply, we want to compare disease and normal. And we wanted to make this kind of knowledge for developing drug. However, based on our experience, we already know, even though in normal variations, there are much larger variation compared to the disease status itself. We can call them Schuffer-Hertz. Schuffer-Hertz can be reached by just exercise or low-calorie diet. However, we wanted to know just the physiology, pathophysiology, from Schuffer-Hertz to leading to disease is quite different to the normal to disease levels. This is simply, understood by many researchers like this. In disease status, that means metabolic dysregulation. In Schuffer-Hertz, that means, just resistance to metabolic stress. The main organ, is, determining resistance to metabolic stress is, just to fat tissues. There are several fat tissues. Just one is, white adipocyte. Much larger than the other ones. Also, brown adipocyte and a beige adipocyte is just between white adipocyte and brown adipocyte. To determine the metabolic stress, several organs have some crosstalk, for inter-organ crosstalk we call. So to understand the main pathogenesis of metabolic stress, we have to measure inter-organs, communications, and how can they work each other. So in my center, this is a Korea mouse phenotyping center. We provide just a generation of knockout mouse and a systemic mouse phenotyping pipelines. And furthermore, in metabolic syndrome, we provide in-depth phenotyping. In-depth phenotyping will be achieved by energy balance and exercise, immune behavior, and several kinds of phenotype parameter. Especially in my laboratory, we can measure metabolic calorimetry in jump-free condition, also in treadmill and cold stresses. We published several data, STK3 and 4 knockout mice in Nature Metabolism, also TM4SFX5 knockout mouse in diabetes, also TMEM6A, this is under revision in Nature Communication. Also, we report here, some CDR reporter mice. This is a kind of just measuring thermogenic drug screen. Also, let's start with this one. This is just a metabolic phenotyping platform, used by IMPC members, IMPC institutes, among them insulin level, and calorimetry, and IPGTT, and a body composition is closely related with the metabolic syndrome itself. And furthermore, the other ones, for example, grip strength, and just the clinical blood chemistry, including leptin, cytokine, and adipokine. Also, calorie whole body, challenge whole body plasmography is very deeply involved in respiratory exchange rate. Also, just organ weight and a history of pancreas and fat tissue is involved in metabolic stress. So we wanted to make a determination for prioritization of disease-related genes from IMPC mouse phenotyping data. Simply, we make just a pheno heat map. We measured, just more than 5,000 mouse models. Among them, we selected 119 pheno heat map. Red means just abnormal heat issues, blue is just normal. You can see easily weight curves issues and body composition issues, and some mouse shows also glucose tolerance issues. And some mouse showed, they do not show, just issues in heart rate and non-invasive blood pressure. However, they showed some weight curves issues. Also, we wanted to make a summarize like this. This is total almost 5,000 mouse data. Among them, 120, almost 120 is showed something problems. However, among them, 65 is a day, I mean, just previous data was already existed. However, 54 genes, I mean, these are really normal genes, I mean, related with the metabolic syndrome. So we summarized this data. That means we just focus on body composition and a body weight and a glucose tolerance and a calorimetry. Among them, MYL96 or GRXC1, that kind of, I mean, gene mutations, I mean, cause body composition issues, body weight issues, glucose issues, and calorimetry issues. However, ACIL1 issues, just they are involved in body weight, however, not related with deeply body composition, glucose, and calorimetry. So we want to make just a prioritization of obesity and diabetes related genes. So among them is just, this is one of example, this is ANAC. ANAC is the, I mean, just the largest protein in our body. And we already published this data. ANAC knockout mouse showed just resistance to obesity in due to impaired adipogenesis. They are very slim. Even after we fed just a high-fat diet, however, they are very slim compared with, I mean, normal wild-type mouse, high-fat feeding. So also they showed some issues. I mean, they are very advanced, I mean, brown adipogenesis and lipolysis via responsive to beta-adrenergic signaling. You can easily see this is a high-fat diet wild-type. However, even after we fed high-fat diet in knockout mouse, their RNA sequencing data is almost the same with, I mean, normal mice and with CHO diet. So that means ANAC mouse showed very different, I mean, pathway in adipogenesis even after high-fat diet feeding. Among them, okay, right side is just a wild-type mice. SEMA4 gene is highly expressed after high-fat diet feeding. This is, I mean, normal cases. However, in ANAC knockout mouse, they showed non-obese phenotype. SEMA4 gene level is almost the same. So that means in ANAC knockout mouse, SEMA4 gene regulation is quite different with the normal issues. So we assumed maybe SEMA4 is closely related with, I mean, just insulin resistance and high-fat diet induced obesity in mouse models. And we check up the, I mean, phenotype data. And SEMA4 knockout mouse showed, I mean, just something problems in homeostasis and metabolism issues. Also, that means, I mean, this is a good chance for us focus on how they can work with the other issues in SEMA4 knockout mouse. However, we have to remember this one. Even though we measured, I mean, gene expression of SEMA4 in adipose tissue, however, still, adipose tissue is composed of adipocyte and the other cells. That means, I mean, mainly immune cells, and we call them SVFs, stromal vascular fraction. However, SEMA4 is expressed mainly in immune cell fraction, not in adipocyte itself. Therefore, I mean, some immune-related genes in the adipose tissue can great effect on, I mean, adipogenesis by high-fat diet feeding. This is one of the examples. SEMA4 expression is, I mean, increased after high-fat diet feeding, even in spleen. Also, this is, I mean, just metabolic improvement in diet feeding in SEMA4 mice, because after high-fat diet feeding, red means knockout and high-fat diet feeding, there is some difference after 11 to 12 weeks. Since 11 to 12 weeks after feeding, body weight is, I mean, changed even after high-fat diet feeding in knockout mouse only. So we can easily compare just the fat mass is decreased in knockout mouse after high-fat diet feeding. Also, body composition is, I mean, decreased in high-fat diet feeding. You can easily see the fat size in high-fat diet feeding after, I mean, just epidermal or WAT size is smaller. Also, the fat cell size is smaller. How can they work? This is one of the example. This is, I mean, metabolic improvement, because you can see easily several kinds of mTOR signaling or PIFA gamma signaling in after high-fat diet feeding in knockout mouse is decreased in protein level, in just mRNA level. Also, this is a direct, I mean, evidence. Metabolic improvement in diet-induced obese SEMA4 mice, because IVGTT, I mean, area under curve is significantly, decrease in knockout mice after hepatitis feeding. This is very interesting things because semaphore is mainly expressed not in adipocyte, but they strongly expressed in just B cell fraction. However, they have a great effect on body weight control, including insulin resistance and sensitivity. So we wanted to also check out, after GTT, because this is an ITT. ITT is a much more sensitive parameter, determining insulin resistance. So in ITT, insulin tolerance test, area under cover is also decreased here. Also after that, we also check the adipose tissue inflammation. Adipose tissue inflammation is, decreased in semaphore knockout mouse. After hepatitis feeding, you can easily see the IL-1 beta and interferon gamma is decreased in mRNA level, also protein level in PK, IK beta 1. Also, this is just histological data. You can easily see the CCL4 or CCL2 is increased in hepatitis feeding. However, in knockout mouse after hepatitis feeding, not significantly increased. They are decreased compared with the wild type cell. So you can easily see the hepatitis after hepatitis feeding, just the inflammation signaling here. However, knockout mouse very significantly decreased. Also, pro-inflammatory, adipose tissue macrophage were decreased. We can easily check up the just surface staining marker in M1 and M2. Also, M1 M2 ratio is highly significantly decreased in this mouse model. However, still, we wanted to understand the exact mechanism, how it can work here. So we did single cell analysis. This is wild type knockout and knockout mice. This is wild type hepatitis feeding. You can easily see the TGF beta 1 and HER-PAX and kappa TNF for many inflammation is highly increased in after hepatitis feeding. However, in knockout mouse, not significantly increased. How can they work? This is, one of the example. This is macrophage, fraction. You can see the here. We compare the, knockout hepatitis diet with a wild type NCD mouse models. So just the good answer is, in single cell level, the interaction between macrophage and the other cell is highly decreased by semaphore knockout mice. So this is another example. Phagocytosis was decreased in hepatitis diet. Phagocytosis is very important for increased inflammation and adipogenesis in adipose tissue. However, phagocyte itself is decreased in semaphore knockout mouse. Also in spleen, inflammation is highly reduced. Also, just to class switching. That means B cell has a working for plasma cells. This is class switching molecules, immunoglobulin G2C and immunoglobulin A. You can easily see that immunoglobulin concentration in serum is decreased in this kind of mouse models. Also, this is another data, and we compare the lymphoid tissues. You can easily see this one. You can seriously compare in wild type after hepatitis feeding, total inflammation signaling by lymphoid tissues is significantly decreased. This is normal. However, after hepatitis feeding in knockout mouse, the inflammation signal is anti-inflammatory signal is almost equal to knockout mouse and wild type with non-normal chow diet. So this is one of the key issues leading to improved adipogenesis inflammation signaling in semaphore knockout mouse with hepatitis feeding. Just to summarize this example. I really have a great appreciation for inviting this chance and also share my experience, even though this is very short time, so I don't have enough time to share my experience. However, if you have any kind of mutant mice, please mail to me or let us know. We can share the mouse phenotyping platform with you. Also, thanks again, and also we wanted to see you in next Plus meeting in Seoul, Korea. Thank you.
Thank you, Sam, for a nice presentation. So it's quite exhaustive for the most of the students. So this time for 2 quick questions because we're already running up the time. Anybody from audience? Yes, please provide him the mic.
Hello, Jay. It's a nice presentation.
Yeah.
Yeah. So any in vitro data for this study? Do you have? Do you have any in vitro data like before to
Pardon?
go to the in vitro data? In vitro.
In vitro data. Okay. Due to limited time, I will just skip up. I mean, we already, make a semaphore knockout mouse Cas9 designed, some inflammation cells, including, B cells. However, as you know, as they already, B cell issues, very not easy and very handling is very difficult. So instead of that, we make a knockout macrophage and we make a knockout lymphoid cells. With the Cas9 designed semaphore, their inflammation signal is very lower than the others.
Okay, thank you.
Any other question? Yeah, if not, thank you, Dr. Sain.
Okay, thank you. See you in Seoul in next year. See you.
Bye. So now I invite the next speaker, Dr. Uddhav Chaudhary. Dr. Uddhav is a scientist E at NIRH Mumbai, who is pursuing research in the primate biology and physiology. So today he's going to enlighten us about the global epidemic obesity, the lesson learned from the spontaneous obese non-human primate model for prevention of human obesity. And I would like to mention that Dr. Uddhav is already running a healthy diet plan at NIRH campus, and he's actually showing these all this by examples. And his work is whatever he is going to speak is his own experience with work at the US Primate Center when done his postdoc time. So let's listen to him.
So thank you very much, Chair. And I would like to take this opportunity to thank the organizer for inviting me for this important meeting. So let me begin with acknowledging my sincere thanks to Dr. Barbara Hansen, where I did my postdoc fellowship under DHR fellowship, and whatever learning I have in the obesity and diabetes in non-human parameters because of her lab experience. I would like to also thank Department of Health and Research for providing me human resource development fellowship, so that I connected with Dr. Barbara Hansen in her lab in 2016 and 17. And from the last 5 years, we are collaborating and understanding the different aspects of non-human primate obesity and diabetes. And also I would like to thank Dr. Gitanjali Sachdeva, Director of NIRSC. So let me begin with what is the obesity has important role in One Health. So we know obesity is a risk factor for many metabolic diseases, but what it has in the One Health. So the COVID pandemic has taught us that obesity increases the susceptibility to infectious diseases. And we know the pathophysiology that there is increased pro-inflammatory cytokine, there is increased M1 cytokine in case of obesity, and that make obese people are more vulnerable to the infectious diseases. And it has been seen both in the influenza as well as COVID pandemic. Then obesity increases the antimicrobial resistance because we don't know what rate or how much right dose of the antimicrobial in case of the obese individual, and we also don't know what is the biodistribution and what is the interaction of my antimicrobial with different metabolites in case of the obesity. Also, during the obesity, there will be insulin resistance and leptin resistance, and leptin resistance makes us satiety very lower. So we always need high calorie food, and high calorie foods is causing deforestation, and we are going more closer to the new viruses because we know in Indonesia there are lot many deforestation has done to grow the palm oil, and palm oil is one of the major exporter there, and then due to the deforestation we are going more closer to the viruses. And also studies have shown that those owner who are obese, their pet animal are also more likely to, they will be obese, because it will reflect the lifestyle of the disease owner into his companion animal. And that is also one of the major factor for the One Health obesity contributes. So one good news is that obesity is preventable and aging related condition can be delayed. So obesity is nothing but the excess calorie intake exceed over the calorie output. So it is an individual behavior and that can be preventable, but it is not so easy because the individual behavior is influenced by so many cultural, political, and other biological determinants which influence the individual behavior, and it's make very challenging to prevent the obesity. And also based on the different clinical and preclinical model, whatever evidences we are generating that can help in the prevention of obesity, and animal model play a very important role. Many milestone has been achieved because of the animal model to understand pathophysiology of obesity and diabetes. So we have discussed so many animal model, which are starting from the non-vertebrate to small rodent model, which are genetic monogenic or polygenic or diet induced. Then we have large animal model and more relevant animal model like non-human primates. So does size matter in the obesity research? So as per the rate of living theory, faster is the metabolism, shorter is the lifespan. And compared to our body size, human has larger or longer longevity as compared to the other animal. And this is because the whatever medical innovation we have done, due to that in the last 100 years, the human lifespan has been increased almost double. So to study the long span life course trajectories of obesity and diabetes, we require relevant animal model, and non-human primate, due to their physiological similarity and larger lifespan, are the proper animal model. So do spontaneous obesity develop in non-human primate? So in wild, it is non-likely to develop obesity because of the kind of exercise this non-human primate do, and also most of the energy is used for the thermoregulation. But when these non-human primates come in close contact with the human and if they are getting excess food, mostly in the case of temple, and they are high calorie food, so they do develop spontaneous obesity. And under laboratory condition, the median life of non-human primates is 26 years, and almost 10% non-human primate can survive up to 35 years. So 25 years of the non-human primate is equivalent to the 75 years of the human life. So if you can study the longitudinally 25 years of the non-human primate, then we can study almost 75 years human being, and we can track the longitudinal trajectory. And during the laboratory condition, even in the healthy diet, almost 10 to 20% non-human primate develop spontaneous obesity. And these are the classical signs of the metabolic syndrome, which can be seen in the non-human primate model too. So non-human primate exhibit greater similarity to human being and susceptibility to middle age onset of the obesity. So most of the obesity which develop in the human life is during the middle stage between 40 to 45 years because of the sedentary lifestyle and hormonal changes occurring during this middle age. So this is the colony which we studied at the University of South Florida. They were having almost 262, 73% were male, and almost 28% they developed spontaneous obesity. Their age range between 4 to 40, and their weight was 4 to 28. So we studied the longitudinal trajectories of body weight, and you can see, so it is not possible that every monkey will follow the same trajectory. So we have grouped this non-human primate into different phases. So 1 to 2 phase represent the normal body weight, and 3, 4, 5, 6, they are like metabolic syndrome. So there will be increased levels of whatever the markers of metabolic syndrome. 7 phase is like pre-diabetes phase. So before development of diabetes. 8 phase is a diabetic, and 9 is the insulin treated diabetes. So if you see the insulin response during the body weight change, we can see dramatically changes and bumping of the insulin or hyperinsulinemia. And this hyperinsulinemia is mostly responsible for most of the diabetic and cardiovascular complications. So insulin levels were significantly higher during the metabolic phase, and also the insulin response to the glucose 75 gram glucose intravenous tolerance test, there will be almost 3 to 4 times increase in the insulin level. And if you see the insulin sensitivity toward the glucose, it's get decreasing. So in the body, glucose will stay for the longer time, and that will convert it into the lipid because of the insulin resistance. Similarly, blood glucose level is tightly controlled throughout the phases, but during the pre-diabetes phase, as the beta cell compensation of insulin response decreases, the increased level of the glucose will be seen, and there will also increase level of other markers like triglyceride and cholesterol. So if you see, obesity is like very complex. There are almost 300 networks and hundreds of determinants which interconnect each other and which are mostly responsible for the obesity. But the summary is that obesity is nothing but the energy imbalance. So, there are different researchers, they ask question, can calorie restriction prevent obesity and its long-term complication? So calorie restriction is not new to us. So calorie restriction was calorie restriction studies was conducted by Clean Mackay. He was the animal physiologist and nutritionist. So in 1935, he studied the effect of calorie restriction on rat, and he found that if we will restrict the 25% of calorie in the rat, their lifespan will increase to almost 30%. So these are the male rats and these are the female rats. So this shows the ad libitum film, and these two are showing the restricted film. So you can see under the calorie restriction regime, the lifespan of rats was increased almost 30%, both in male and female. But this research was done almost 75 years back, and that time longevity or lifespan was the not major issue. That time communicable diseases was the major, so it was not got that much attention because penicillin was not discovered before this experiment, and that time overall lifespan of human was almost 50 to 53. So that time it was not that much recognized, this research, but eventually when the prevalence of obesity started increase, the recognition for this work was increased, and several studies were conducted. So researchers asked question, does calorie restriction phenomenon seen in the mice, that we have already seen that if 25% calories are restricted, then the lifespan is increased to 30%. So can same phenomenon seen in the highly complex organism like human and non-human primate? So so when I was doing my postdoc, so in that lab already they have done the calorie restriction study almost 20 to 30 years back. So what approach they use? They use weight clamp approach, means they were knowing the normal weight of the monkey, then they feed it the normal, suppose if they require 15 biscuit to maintain the 10 kg of body weight, so they give an equal quantity of that biscuit so that their body weight can be maintained. If their body weight is increasing, then they will reduce one biscuit, and if their body weight is decreasing, then they will give one extra biscuit. So like that, they just clamp their body weight throughout their lifespan to the normal body weight. And what they observe after doing the calorie restriction during the mid age of the life, because obesity is mostly seen during the middle age, and they observe that average lifespan of the monkey, those who are calorie restricted, were increased. Their maximum lifespan were also increased, and all the calorie restriction monkey, they do not develop diabetes mellitus. So this was the major conclusion. And when they did the survival curve, so you can see the calorie restricted monkey 50% survival was almost 32 years, and we know that in the laboratory condition, the average survival is of the non-human primate is just 26 years. So compared to the dietary restricted monkey, ad libitum monkey had 2.5-fold increased risk of death, and hyperinsulinemia leads to 3.7-fold increase this risk death. So the hyperinsulinemia was the major culprit for the this death in the calorie ad libitum food. So they studied the insulin sensitivity throughout the lifespan, and they observed that the calorie restricted monkey, their insulin sensitivity was low, was the minimum throughout the lifespan. Also, the glucose, plasma glucose response was maintained. So their insulin sensitivity was better throughout their lifespan as compared to the ad libitum monkey. You can see how much the spike of insulinemia or hyperinsulinemia is there during the middle age onset of the obesity, and also there is a decrease in glucose disappearance rate, and this ultimately leads to the development of cardiovascular diseases and diabetes. And we are known that hyperinsulinemia is one of the major contributors for the comorbidities or non-communicable diseases. So under the calorie restriction regime, we can lower the hyperinsulinemia and we can maintain the healthy lifestyle for the longer duration, and it will increase the longevity and healthy aging. Ultimately, we want that quality of life should not be suffer at the end of the life. So calorie restriction improve the insulin sensitivity throughout the lifespan of non-human primate. And similarly, another two study which were published in 2017. So one study was done at the University of Wisconsin Primate Center, and another was at National Institute of Aging. So here they were almost used 50-50 calorie restricted monkeys, and they were studied throughout almost 30 to 40 years. And still these studies are continue. If you see the University of Wisconsin study, they have not found that much difference in the overall longevity, but their study regimes were and their calorie restriction protocol were different as compared to the National Institute of Aging studies. And if you see overall prevalence of the cancer, insulin resistance, and the cardiovascular disorder, there was significantly decrease in the incidence of the all cancer, diabetes, and cardiovascular diseases in calorie restricted non-human primate as compared to the ad-limited non-human primates. So calorie restriction can conserve in monkey, means whatever the finding which we have seen almost 75 years back in the rat were similarly found in the human, and this can be translated to the human health. So that day, doctor, we have to see some simple approach. We are searching so many drugs which can improve the obesity, but this is the simple approach. Just we have to cut our calories and we can improve our lifespan as well as our longevity. So where are we are now? So there are several studies from the invertebrate to the vertebrate they have shown, and there are different regimes now available, like calorie restriction, intermittent fasting. Then we have calorie restricted mimetic like drugs, and also we have time restricted feeding. So there are different protocols are available to cut down our calorie, and we can improve our lifespan. And there have been several evidences from yeast to the monkey, but we don't know whether it will happen in human or not. But definitely, if you have studied in the most animal like non-human primate, it is going to translate into the human also. Improve overall survival and longevity. So it reduces the overall inflammation, then it improves the insulin sensitivity, then the glucose levels will be less, and ultimately all these parameters will help to reduce the incidence of cancer, type 2 diabetes, and cardiovascular diseases, and it will improve the health and longevity of the human population, and it will be less burden on the healthcare system throughout the world. So now coming, what I have learned from this calorie restriction study. So incidentally or accidentally, in 2019, I was also diagnosed with pre-diabetes, means my glycated hemoglobin level was 6.2. And that time I thought, oh, if it is going to 6.5, then I will become diabetic. And whatever I have learned from the animal model or whatever I have studied, sacrifice their life, and they have shown clear-cut evidence that if you can restrict your calories to 25%, most of your complications can be avoided in your life. So I followed, I did my nutrition course, and then I just did some calorie restriction initially for 2-3 days to reduce, because my BMI was 28. For Asian, if BMI is more than 27, then we consider ourselves as obese. So first I want to reduce my BMI to the normal range between 25 to 26. So initially I did some calorie restriction, only time restricted feeding. So evening I just, I have not taken any calories in the evening, only salad. So within the 3, 4 to 5, 6 months, I could reduce my weight. And after that, I just continue on the 20% calorie reduction. And over the period of time, almost 3 to 4 years, I am maintaining my body weight. And we have to just, I am sharing this personal experience because we have to sensitize more people. And we know some of our colleagues like Dr. Rahul Thorat, he is doing bicycle to healthy, adopt a healthy lifestyle. Then Dr. Vijay Pal Singh, his eat right movement, he is taking forward. So all these, whatever experience we are getting from the animal, that we have to translate into our real life so that society can be helpful, and the burden of the animal experiment should be, it will be lower. So that can be diverted to other conditions. So there are also similar studies has been now initiated in the human. They are like randomized controlled trials, and where also because it is not possible, because in monkey in the laboratory condition it is possible to track them up to the 40 year, but in case of human it is very difficult. You just 1 year maintaining on the 25% reduced calorie, it is not possible. But there are studies. So this is the study which is conducted for 2 years, and they have found significantly reduction in the cardiometabolic risk factors, like reducing the body weight, then reducing the systolic blood pressure, then increase HDL levels, and others parameters. So calorie restriction over the period of time helps in significantly reducing the cardiometabolic risk factor in young and non-obese adults. So there are evidences coming based on the animal study. Now human trials are going on, and it is ultimately our behavior that will help to reduce the global burden of obesity. So thank you, Mary. This is the food pyramids, means how we should eat. So we should eat more of the plant-based food and less of the high-calorie food. So it has inversely effect, means if you consume more high-calorie food, it will have more environmental impact on the society, as well as, because we know beef production, how much carbon emission is there, and ultimately it will help in the environmental health. So ultimately food is I think the center of the One Health program, and if we can reduce this food to the minimum level, we can solve many problems of world health. So thank you very much for your attention. I'm happy to answer any question.
Yes, the session is open for questions. Yeah, please, Dr. Vijay. Yeah, Mike, please.
What was the their exercise regime, and when you are saying restriction of the diet, which portion of the diet you manipulate, or you just manipulated as the whole diet?
No. That I am sorry, I didn't mention calorie restriction without malnutrition. So we have to only the calorie restrict the calorie portion of this, but we have to give excess micronutrient like minerals as well as vitamins. So calorie restriction only in the form of carbohydrate.
Okay. And what about the exercise?
For whom?
This your monkeys.
Monkey, no, they were not. I think they were maintained in the normal caging system.
Okay. I want to also, this is extremely good work, and this should be translated into the human. But humans are complicated because we take a lot of junk diet and because of our, taste and everything, and that we have discussed. But another important point I want to ask: recently there is a systematic review come up that if you are obese, you start exercising and restriction diet, you become normal. Then there are increased chances of your cardiac arrest and everything. What's your comment on that?
So when we say calorie restriction, so if you see Indians calories in the form of carbohydrate is almost 80 to 90%. We are protein deficient as well as micro deficient. So what they say, minimum calories we should take from the carbohydrate is between 60 to 65%. But if you will reduce significantly calories between 45 to 50%, then it will be like having a different effect, and their cardiovascular complications. We should not extremely restrict your calories from the carbohydrate, because carbohydrate is required. It is not like that without carbohydrate we can survive, but the proportion of the carbohydrate which is required is between 60 to 65%. You should not be below the 60%. But as an Indian, we have like 80 to 90% carbohydrate, and that is the major risk for the obesity.
Achha, sir, one more question. This calories, suppose we may take 2000 calories in a day. Is it only the number of calories we are taking in a 24-hour time period, or the timing of calories also? Suppose nowadays this intermittent fasting concept is coming up. We should take all our calories, finish it before 7 o'clock or something.
Yes,
So does timing of the calories also matter, or it is the only number of calories taken during a 24-hour duration? What is your viewpoint on this?
So of course, intermittent fasting help. Means if you will take almost 12 hours, if you will do fasting, and if you will consume your 12 means almost 200 calories in one day, and if you are fasted for another half day, so it will work. But if you want to take throughout your day calorie, then we just restrict 200 kilocalories. Suppose you are having 200 kilocalorie requirement, if you will restrict it to at least 200 kilocalories, so over the period of time you will get effect of weight reduction. So it is both way. Either you do intermittent fasting, or you can do the calorie restriction.
I'm asking, suppose in intermittent fasting also I am taking 2000 calories, and in suppose someone is not doing intermittent fasting, and
so it will depend upon how much your calorie requirement.
Means suppose both two persons, two groups, they are taking, one is taking 2000 calories, another one is taking again 2000 calories, but with intermittent fasting. So is that intermittent fasting beneficial, because number of calories in both the groups are same? So what is your viewpoint on this?
So recently, I think New England Journal of Medicine has published one article on intermittent fasting versus calorie restriction, and they have not found any difference. There was reduction in the body weight, but there was not significant difference between the calorie restriction versus time restricted feeding. So it is like same.
Now we have some time restriction as well for to conduct this particular session. So maybe we can talk for the tea time.
नहीं,
Please,
ये
yeah.
एक रिक्वेस्ट है कि लासा मीटिंग में कैलोरी रिस्ट्रिक्शन नहीं होना चाहिए.
Yeah,
ये
that
थोड़ा
is the
ध्यान दे दो.
cultural, yeah.
तो नेक्स्ट ईयर से नहीं तो पूरा सलाड पे रख देंगे 3 दिन.
No, we should have packed food with calculated calories and higher.
Depend upon the BMI. So we have to give the weight of the person while I'm joining the conference, and then accordingly the weight plan will be given.
I worked at ONRC. ONRC means Obesity Nutritional Research Center at NIH. I worked as a postdoctoral fellow. Earlier I worked with NIN, Dr. Giridharan with Obesity.
Sir, please speak in a mic, please.
Hello.
Yeah.
Yeah. Okay, sir. So earlier I used to work with Dr. Giridharan with obese rat. When I was in NIN, I used to weigh around 110 kgs. After joining ONRC, I reduced it to around 65 kgs. It was very difficult for me to maintain 65 kgs. Then slowly increased. Now for the last 10 years, I'm like this. So what I did, my experience is, we have a different programs, calorie restriction programs. We did alternate day dietary restriction, alternate day calorie restriction. We do. Like earlier, in the beginning day, we used to eat around 1500 to 2000 calories. Next day, we used to have 1200 calories. Like that, we used to maintain. I used to do that exercise. And then
So self-awareness is most important for the maintaining the body weight. And whatever knowledge we have, that we have to translate, and we have to control our mind that this is only I require. I don't want extra.
Okay, yeah. Yes, sir. Thank you, Dr. Uddhav, for a nice presentation. So now we close the session.
I request the chair of this session to felicitate the speaker with the memento, please. Go for this. I request Mr. P.Y. Guru to come and give a token of appreciation to the chair of this session.
It's okay.
I request Dr. Mahesh Sahare, NCBS Bangalore, to please come on to the dais and give a token of appreciation to the coaching. Okay. And start the next session. Thank you. We'll proceed to the next session, which is the award session for the best oral presentation by students. I hope the students are ready on their presentations. So we'll start with the first presenter, Dr. P. Veeresh Babu, Department of Pharmacology, Kokaraju Rangaraju College of Pharmacy, Hyderabad. Are you here? I don't see. Go to the next one then. Not the what? So we'll go as planned before. We'll go with the oral presentations. Okay, let's wait. Let them decide. Okay, so this is final. We are going with the award session for the best oral presentation by students. So I'll quickly give the rules. You'll have 5 minutes for presentation and 1 minute for discussion. So the first one is by Dr. P. Veeresh Babu. I think he's joining us online. You have to project your slides yourself, Dr. P. Veeresh Babu.
Hello, madam.
Am I clear?
Madam, I am getting some disturbances. Can I get some time so that I can give second presentation like that?
Okay. You please get it ready for the next presentation in the next 5 minutes.
Okay,
We'll
thank you, madam.
We'll go with Dr. Venkat Ramireddy now. Is he joining online or offline?
Yeah, online, madam. I'm ready.
Okay. So you please, I hope you've heard my the rules, right? Yeah. You have 5 minutes. You please project your slides.
Okay. So thank you. Very good afternoon to one and all. I am Venkat from Kadapa, Yogavyamana University. My topic is biogenic silver nanoparticles ameliorate dimethyl hydrazine-induced colorectal cancer in rat model. So these biogenic nanoparticles we prepared using eucalyptus globulus plant because it contains several phytophenols, anti-cancer agents and anti-inflammatory agents for treatment of colorectal cancer. So in the colorectal cancer, as you see, the major role that plays are COX-2 and NF-kappa B. So they are mainly responsible for the translation of inflammation to the colon cancer. So there are, even though there are several chemotherapeutic radiations therapies that are really not having good effects in the treatment. So hence there is a need for the promising therapies. One of the promised therapies recently has been discovered is the nanoparticles. So in this case, we have selected the silver nanoparticles because these silver nanoparticles have been tested in vitro for different cancer cells, including lung and colorectal and carcinoma cells. And the main objective of this study is to prepare the silver nanoparticles using eucalyptus globulus and then induce the animal model with dihydrazine dimethyl hydrazine. Here we used 5 groups. One is control and second one is just disease model, and the other 4 are different combinations of silver nanoparticles prepared with and without 5-fluoroaerosol. That is a combination drug. This protocol has been approved by the Institutional Animal Ethics Committee of the university. And then after actually this is the 15 weeks model. After 15 weeks, initial weight and after final weight has been obtained, there was no significant difference. And then colon sections was collected from the different groups, and we found several tumors, that is a foci, aberrant foci was present in DMH groups, whereas in treated groups, we seen few in 5-fluorouracil, and in combined treatment with 5-fluorouracil, that is known drug, and with the silver nanoparticles, almost nil or one number of the tumors was observed per rat. So that indicates that the incidence of the tumors was clearly reduced in case of the silver nanoparticle treatments with the combination. And then we did the histology for this the rat samples, where we found in the colon there is a inflammation surrounding these tissues, and also it was gradually reduced with the combination treatments. And you don't see any infiltration of the inflammatory cells, and foci, aberrant foci also not observed in these sections. Also, we wanted to look in the other tissues like lung and liver, where also you see a lot of inflammation and infiltration of the cells was present in the diseased model, whereas with the different groups of treatment, it is significantly reduced in the combination therapy, suggesting that it is also interfering with the induction of the inflammation. Both we can see same results in the liver also. And also because the oxidative stress also caused because of this DMH, we also studied these the enzymes responsible for the antioxidants, SOD and catalase. We did the SOD activity in the serum samples and as well as in the colon samples, like also catalase also we did in the similar way. In all the cases, this enzyme activity was reduced in the DMH induced samples, whereas it is gradually increased with the combination treatment in all the cases, suggesting that these molecules are inducing the antioxidant pathways so that it may having a interfering with the ROS production and other intermediates. That may be one of the pathways. And also we studied the inflammatory cytokines in the serum as well as in the colon tissue. That is mainly the IL-6 and TNF-alpha responsible for the pro-inflammatory cytokines that induce the inflammation that leads to the initially the cancers. Here also the combined treatment as well as individual treatment with the silver nitrate significantly reduced the levels of inflammatory cytokines both in the serum as well as in the colon sample, suggesting that our biogenic silver nanoparticles have effective in reducing the production of inflammatory cytokines. And also we wanted to see the other inflammatory mediators, cyclooxygenase and one of the transcription factor involved in the inflammatory things. And in this case also it was clearly reduced. And also we looked at the some of the stem cell markers that is there in the colon cancer stem cells. These stem cells are responsible for induction of this, and these also reduced. In vitro cells also reduced the cytotoxicity, and suggesting that these colon cancers cells and the colon cancer in rats is inhibited by our biogenic silver nanoparticles. So this is the first report demonstrating that efficiency of combination of biogenic nanoparticles with the fibrinolysis treating colon cancer cells. Thank you very much.
Thank you. Anybody have any quick questions?
I have so much other in vitro data, but because of the time constraint, I could not present.
So have you checked this toxicity of these nanoparticles?
Yes, sir. Toxicity has checked in both normal cells, HEC29 and HT cells. In HEC2 cells, there was no toxicity.
In vivo model. In animal model, have you checked the toxicity?
Acute toxicity, sir.
Yeah, acute toxicity, also chronic toxicity. And
because yeah, sir. Actually,
this is a huge problem with
in this case, we have not tested because these silver nanoparticles has been reported in several models. So based on that, we go ahead with the in vitro and as well as in vivo models. And also the eucalyptus has no toxicity, sorry, eucalyptus phytophenols. That's why we did not study these things. Based on the literature, we have selected this concentration and other things.
If there are no more questions, we'll go for the next presenter.
Thank you. Thank you for giving me this opportunity.
Thank you. Veeresh Babu, are you ready?
Yes, madam. Shall I start my presentation?
Yeah, please. We'll start the timer for 5 minutes.
Is my presentation visible, ma'am?
No. No, it's not.
Okay, ma'am. Now?
No.
Not yet. 1 minute, madam. Now, ma'am?
Give it a minute.
Okay.
Okay.
Okay, ma'am?
No, it's not. I think you have very slow connection.
It might be. 1 minute, madam. It is open on my side.
Ask him to wait and go for the next presentation.
You please check your connection, please. We'll go
Okay.
for the next one. We'll come back to you.
Okay.
Anusha, are you there?
Yes, ma'am.
Veeresh, please stop your presentation so that the
Okay,
other one can.
Sure, sure.
Can you email it to you? Can you
She's
email
there,
it to
she's
you?
there, she's there.
Anusha is there. Okay,
Yes,
Anusha, you
ma'am.
please start projecting your slides.
Yes, ma'am. Good evening, everyone. I'm Manusha Goindula, research scholar from Department of Pharmacology, Manipal College of Pharmaceutical Sciences. I'm here to present my research work that is early life stress exaggerates behavioral phenotype in post-traumatic stress disorder. First, I would like to
Hold
give
on,
you
we
an-
can't see your presentation.
Oh, is it? From my side, it is sharing, ma'am.
Ma'am, you have not shared your screen.
No, I have shared.
No, it is not shared.
We
Anusha?
can't see anything.
Just a second. Once again, I'll share.
Skip and share it again.
Yeah.
Yeah, we can see it now.
Now is it visible?
It's okay.
Yeah, go ahead.
Yeah. So I'm Anusha. Once again, I would like to introduce myself. I'm Anusha, a research scholar from Department of Pharmacology, Manipal College of Pharmaceutical Sciences, Manipal. I'm here to present my research work on early life stress exaggerate behavioral phenotype in post-traumatic stress disorder. So first, I would like to give you an overview on some imperative facts. According to the Diagnostic and Statistical Manual of Mental Disorder version 5 proposed by American Psychiatric Association, post-traumatic stress disorder is a debilitating condition that affects an individual aftermath of a traumatic event that may be a terror attack or a physical or sexual abuse or a context of war or any maltreatment during a childhood. So emerging evidence suggests that early life stress appears to be a substantial risk factor for persistence of PTSD. According to a national epidemiological survey on alcohol and related conditions, traumatic events experienced during childhood could engender severe constellations in DSM-5 symptomology, and then they can surrogate for increasing in the complexity of this disorder. And not only that, in Indian context, a cross-sectional study conducted in Kerala in public and private schools revealed that 34.9% of children had probable PTSD, and in that 31% of public school kids and 38.8% of private school kids had intrusive memories. So PTSD has majorly 4 symptom clusters. One is intrusive memories, avoidance of stimuli related to the traumatic event, negative mood and cognition, and hypervigilance or hyperarousal. So taking these facts into consideration, we have modeled an early life stress in 7-day-old male mouse pups. And this study was approved by Institutional Animal Ethical Committee, MAHE. So during this postnatal day, from day 7 to day 21, the pups were separated from their dams for daily 3 hours till the age of weaning. And then on day 22nd, a physical stressor was induced using single prolonged stress protocol, which includes 3 stressors, 2 hours restraint followed by 10 minutes of FST and inhalational anesthesia. Then they were kept under solitary confinement and a quiescent period of 1 week was followed. And on day 29, a psychological stressor was given by exposing the animals to the predator scent, which is in this case is a cat litter and bedding for 10 minutes. Then 16 animals which were gone through these stressors were randomized into 2 groups. One is PTSD group and treatment group, whereas the control group, they were under the normal housing conditions and they hadn't exposed to any kind of stressors. The control group received 10 ml per kg per day of the vehicle, that is in this case 0.25% weight by volume of carboxymethyl cellulose, and treatment group received the PTSD group even received the vehicle. The treatment group received fluoxetine 10 mg per kg per day as a suspension in the CMC. The treatment were given per oral daily for daily from day 30 to day 60. After that, from day 44, a battery of behavioral tests were followed. These are the behavioral parameters which were assessed to establish the DSM-5 symptomology. For intrusive memories, contextual and acute fear condition test was followed. For avoidance, open field test, elevated test, maze test, and light dark box test was followed. For negative mood and cognition, Morris water maze test, 3-chambered sociability test, which is also known as Crawley test, was followed. And for the fourth cluster, that is hyperarousal, marble bearing test was followed. So for during the intrusive memories, we followed this contextual and pure conditioning test, wherein which we have assessed the freezing behavior. On day 1, peer conditioning test was followed with a context A, and where we have given 3 pairings of auditory cue and a shock with an inter-trial interval of 90 seconds. And on day 2nd, we have changed the context where only we have given the auditory cue. And on day 3, the animals were introduced into the novel context where we have only given the auditory cue. And on day 4, the similar conditions, we have used the similar conditions as on day 1 with the same context. So in this, we have assessed the freezing behavior as well as the freezing episodes as well as the time of freezing, where the PTSD group have shown significant difference with the normal control and treatment group, indicating that the PTSD animals able to remember.
Anusha, you are done with your time. You have to conclude quickly.
In a nutshell, I can say that this model significantly established the 4 symptom clusters. Along with we have also assessed the HPA axis stress hormones, CRH and corticosterone, and concomitant with the behavioral data, with the elevated stress hormones, this model can be used as a translational tool for understanding the behavioral anomalies in the childhood trauma.
Any quick questions?
These are the references, and I would like to thank the organizers and sponsors for giving me this opportunity. And I would like to also thank Griffith University for collaborating in this project, and Mahe for providing the resources.
Anusha?
Yes, sir.
How many cats were used to collect the litter?
No, single, sir.
Single. In case the cat had to be, listen, in case the cat had to be injected and the blood has to be collected to check some of the parameters, how was the IAC concerned about, sanction? I'm asking the question to you.
We have only used the cat litter.
I'm giving a condition wherein, suppose you, instead of collecting only urine, you are collecting blood from these cats and for some analysis, wherein that is part of the experiment. Does it attract the IAC?
Sir, actually, we are inducing a psychological stressor, but the blood exposing. Yeah, thank you, sir.
Yeah, next presentation, please.
Thank you, sir.
Veeresh, you there? Hello.
Go to next.
Veeresh.
Hello.
Yeah. We'll project your slides from here.
Okay. Thank you, madam.
Okay.
Thank you. Am I audible?
Yes. You have 5 minutes. Go.
Okay, madam. Thank you. Myself, Dr. P. Veeresh Babu. Today I am going to present a brief talk on my topic entitled Development and Evaluation of Polyherbal Syrup for Anti-Asthmatic Activity. Next slide. The contents of presentation include introduction, aim and objectives, materials and methods, results and discussion, conclusion and references. Next slide. Hello. Introduction. One of the most
5 minutes.
Okay, sir. Sure. One of the most important respiratory disorder or disease is the asthma, which may be defined as an obstructive disease of the airways that is due to the spasm of airway smooth muscle, increased mucus secretion, or inflammation. Next slide. This is the how the airway smooth muscles or airway bronchioles appear during the asthma. Next slide. And asthma affects almost 5 to 10% of the population in industrialized countries, and it is increasing in prevalence and severity. Next slide. And there are many causes like smoking, nicotine dependence, allergic rhinitis, COPD, upper airway inflammation, which ultimately results in the asthma. These are some of the etiological factors. Next slide. And signs and symptoms of this asthma include poor tolerance to the exercise, irregular sleep, panting, abnormal palpitations, fatigue, runny nose, etc. Next slide. And pathophysiology of this asthma includes due to the exposure to the allergen, dendritic cells gets activated, which in turn activates CD4 cells, which causes the activation of plasma cells and eosinophils, which ultimately results in the release of histamine, leukotrienes, prostaglandins, which causes the airway obstruction and precipitates the asthma. Next slide. So usually we do the diagnosis of asthma using spirometry, bronchodilator response test, inhalation challenge test, and imaging tests, etc. Next slide. And the drug treatment include bronchodilators, leukotriene antagonists, mast cell stabilizers, corticosteroids, and anti-IgE antibody. But these treatments are supported with many of the side effects. So which helped us, or which made us to think of the alternative or complementary medicine. Next slide. Next slide. For our study, we selected 3 plant species, namely Majorana hortensis. Next slide. Next slide. Bauhinia purpurea, which contains beta-sitosterol as major phytoconstituent. Next slide. Next slide, please. And Pizzellobium dulci, which contains the major constituents as squalene, dulcitol, azipilin, etc. Next slide. The aim of this work is to develop and evaluate polyherbal syrup for anti-asthmatic activity using the 3 plants which we discussed earlier, like Majorana hortensis, Bauhinia purpurea, Pizzellobium dulci, and to perform the pre-formulation and preliminary phytochemical investigation of these extracts. Next slide. Formulate polyherbal syrup, characterize and carry out acute toxicity studies in mice according to the OECD guidelines, and to carry out in vitro antioxidant and in vitro and in vivo anti-asthmatic activity. Next slide. So we collected, dried and pulverized the plant species and performed pre-formulation studies by using the standard techniques like determination of foreign matter, moisture content. Next slide. pH analysis, determination of total ash. Next slide. Determination of acid insoluble ash, extraction of crude drug by using oxalation. Next slide. Preparation of liquid oral syrup by using the standard method. Next slide. And we carried out evaluation or characterization studies by standard methods. Next slide. And then we evaluated physicochemical parameters and then carried out acute toxicity studies using the guideline number 425. Next slide. And in vitro antioxidant activity was performed using reducing power assay. And the next slide. Hydrogen peroxide radical scavenging activity. Next slide. And we carried out in vitro antioxidant.
Viresh, you are done with your time. You have to summarize quickly.
Okay. So please come to the last slide, sir. Conclusion, please. So we prepared the oral herbal syrup by standard methods, and we carried out its pre-formulation studies and characterization studies, which revealed that our extract falls within the standard conditions. And then we carried out antioxidant activity by 2 in vitro methods, which stated that our extracts along with the formulation has very good antioxidant activity. And we tested the anti-asthmatic activity of this polyherbal syrup using in vitro and in vivo methods in
Thank you, Viresh.
Okay.
Any quick questions, please? Quick questions. Thank you so much. Thank you, Viresh.
Okay,
We'll go to
thank you,
next
sir.
presentation. Yeah, thank you so much.
Question hai na? Doctor, I have a question.
Sorry, Viresh.
Babu.
Viresh, are you there?
Yes, sir.
Have you checked the AHR airway hyperresponsiveness? Because that is very pathognomic to the asthma.
Yes, sir.
Did you check?
Sir, actually, we checked about the other biochemical parameters related to the asthmatic activity, but not the airway hyper-responsiveness. So the studies are still in response. These are the preliminary studies, sir. We are carrying out the further studies on this activity.
That is the must and only the very first activity which shows about the antihistaminic. Thank you.
Okay,
Thank
sir,
you.
thank
Thank
you.
you. Thank you, Viresh.
Thank you, sir.
Thanks, Viresh.
Thank you, madam.
Next, if there are no more questions, I'll go for the next presenter, which is Dr. Deepali Rawat from NIAB. तो मैं इससे यहाँ से कर लूंगी. So shall I start?
Very good afternoon to all. I am Deepali Rawat, PhD student from NIAB Hyderabad, and today I'll be presenting on monoclonal antibody generation, large-scale production in mouse model for the development of elapid venom detection kit. So most of the molecular diagnostic kits are based on antibodies. These antibodies can be polyclonal and monoclonal. Polyclonal antibodies being easy to produce and in large scale amount, but they have some drawbacks, like they have reduced specificity, they show variation, reproducibility is an issue with polyclonal antibodies, they have a finite supply, and large scale production is tough in polyclonal antibodies. So these drawbacks of polyclonal antibodies are easily overcome when we come to monoclonal antibodies, as they have single epitope specificity, constant supply, no batch-to-batch variation, and less animals are required to be sacrificed for the production of monoclonal antibodies. Plus, they have an advantage of sequencing and documentation that can be done for further experimental processes. So that is why polyclonal antibody-based venom detection kits are not very popular. So here we aim to develop one such venom detection kit using monoclonal antibodies. So for this, we have selected cytotoxin 7. It is a protein it is one of the protein which is differentially expressed in the venom. That is, it is found in the elapid venom, which is cobra and crate venom, and it is not found in the viper venom. So what we did, we actually we expressed this protein in prokaryotic system, purified, and later on characterized this protein using CD spectroscopy, which depicts that this protein majorly have beta-pleated sheet structure. Next, using this protein recombinant cytotoxin 7, we generated monoclonal antibody by a technique known as hybridoma, where we choose mouse model, which is BALB-C, and it was undergone and it was given the injection, or I can say it was immunized with this recombinant protein. The spleen was collected and the splenocytes were isolated from this mouse. It was fused with the SP2O myeloma cells, which are basically the cancer cells. After the fusion, the hybridoma or the fused cell was selected for positive clone. All the clones that were producing antibodies which were reactive to the protein cytotoxin 7. So here, as you can see, we come up with only 16 such clones that were producing antibody that were reactive to cytotoxin 7 protein. So the antibody so the clone producing antibody AB1 was the clone which showed the maximum reactivity towards the cytotoxin 7 protein, and we then went forward with this clone for large scale antibody production. Next, antibody production. So here we employed two approaches, where first we isolated antibodies from cell culture supernatant, and in second we used the same mouse model, which is BALB-C, for the large scale production of antibody. In the first method, from cell culture supernatant, the antibody producing clone, hybridoma clone, was grown into the cell culture medium, and later on the antibody was procured from the column purification method. Whereas in the second method, using ascitic fluid, the antibody producing clone cells was intraperitoneally injected into the mouse, and then after the abdominal distension, the ascitic fluid was collected, and from that the antibody was procured. Here you can see it is a 10% SDS page gel which is showing antibody produced from both the cell culture supernatant and ascitic fluids. There is no compromise in the purity of the antibodies. So where the difference lies among both the methods? The difference is in the yield and the cost of antibody production. As you can see in cell supernatant method, the antibody is 3 mg which is produced per liter of cell culture supernatant, whereas in ascitic fluid the same amount of antibody was produced per ml of the ascitic fluid, and it depicts and it reflects into the cost of antibody for the production of LFA, which is 60 to 70 rupees for 1 LFA strip when it was procured from cell culture supernatant, whereas it was rupees 10 in case of ascitic fluid production. So here I can say that producing monoclonal antibody using mouse model at laboratory level is not just efficient but also cost effective. This is a schematic I've shown here to show how the LFA kit for the detection of venom was made, and I would like to focus on these two lines over the LFA. This is the venom line and safe line. As you can see, when there is no venom in the sample, there will be presence of both the line. But as the venom concentration increases, so that was the LFA, and this is the case reports which we have got by we have made the antibody detection kit, and we have given it to nearby primary health centers, and we have got the same result. As you can see here, this was
with your time, you have to summarize.
here this was the case of a child who was bitten by cobra, and this was a mild case. So same results was obtained when the physician used the antibody kit which we developed for this. And in this case, it was a severe case. Here you can see no safe line. That means yes. So I would like to say that animal models as small as mice can be used not just for the human welfare but also for the welfare of animals. I would like to thank DVT, NIAB, Small Animal Facility of NIAB, and CSIR and BIRAC. Thank you.
Like there.
What you see this detection kit which you have developed, what sample from the human being you are taking to actually check?
Sir, this is the serum sample, human serum sample.
Non-specificity, did you observe?
No, there is no non-specificity observed. Yes, sir. This antibody detection kit is right now just for crate and cobra. It will detect only those two envenomations.
It is bitten, and after how much time do you expect this to give a positive result?
Sir, if the person is bitten at time 0 and at that time only the serum is collected, the blood is drawn from the person, at that time only if serum is collected, this will show the result at that time only. There is no need to wait for time.
We can move to the next presentation, please. Thank you. Thanks a lot.
No, I'll just. Sir.
Next, we have Dr. Nasiha Begum from National Institute of Unani Medicine, Bangalore. Nasiha. Excuse me. Down.
Very good afternoon, all. I'm Dr. Nasiha Begum from NIUM. I'm a PG scholar. This is a part of my MD work. The study topic was the antihypertensive effect of seed of Coriandrum sativum lean in adrenaline-induced hypertensive model. As we know, the hypertensive is a major cause for the cardiovascular diseases and death in the current scenario. There are several drugs available in the market for the reducing the hypertension, even though the prevalence is increasing in the population. There's one expectation it will be double up in the 2025 compared to 2000. So here the need of study was to develop a new drug for the hypertension, which should be patient friendly and it should be very low side effect. On the basis of that need, the objective was to evaluate the antihypertensive effect of the Unani drug, that is Tuhmekashni's Coriandrum sativum in the adrenaline-induced hypertensive rat model. This is the Coriandrum sativum under the Apiaceae family, well-known plant that is known as coriander. For the study, we use an adrenaline-induced hypertensive rat model in the method of tail cuff non-invasive method by using the NIBP controller. As start, the drug is procured and it's cleaned, and after that, it's identified and authenticated, and finally, its extract is done by the Sox-like apparatus, and the extractive percentage was observed 24%. From that, the oral administration is prepared in the form of suspension in water. For the statistic, GPAD is used for final calculation. The study design was, the animal was used all healthy vista rat, male vista rat in the weight of 150 to 250 grams. So there was three groups. One is disease control, other one is standard control, third one is test group. For the standard control, those animals receive adrenaline in the dose of 0.5 milligram per kilogram, and the standard control drug was propranolol, and the test drug was Coriandrum sativum. The detailed study plan, the study is divided into 4 parts. First one is hypotensive effect of the drug. Second one is antihypertensive effect of single dose of treatment. And third one is antihypertensive effect of multiple dose treatment. And fourth one is curative effect of the drug. Now I'm coming to results and discussion. In the first step, we check the hypertensive effect. For that, before starting the work, all the BP of the all animals were recorded, and that BP was considered as a baseline BP of the animals. And after that, the respective drug is treated for the respective group. For this step, only 2 groups were utilized, that is standard and test group. After the respective drug administration, 1 hour after the drug administration, again BP was recorded. And the difference between the baseline and after drug administration was calculated, and that was used for the evaluate the hypotensive effect. And the second step, after the first step finish, the same animal utilized for the second step, but in the second step, all the 3 groups were utilized. Here, the adrenaline was injected to the disease control, and after that, after 5 minutes of adrenaline injection, again the BP was recorded. And in the other propranolol and test group treated groups, after the adrenaline injection, 5 minutes later, the BP was recorded. Here, after the adrenaline BP value after the adrenaline injection and the baseline was calculated for the evaluation of the antihypertensive effect of the single dose of treatment. Here, the first steps and second step followed in same day. And third step, the treatment was continued for 4 days, and the treatment was continuous for 4 days. And after 4 days, the same method was conducted in the 4th day and 5th day. After that, and from 5th day, the adrenaline was stopped and treatment was continued for another 6 days. Here, in the conclusion, in the first step, we observed the BP reduction in the both test and standard group, but those reduction was not significant, statistically significant.
You're done with your time.
Conclusion. Here I can conclude the hypotensive effect observed in the both group are not significant, and the antihypertensive effect was observed in single and multiple dose of treatment was observed, that is statistically significant, and there was it was observed the similar effect in the both standard and test group. And the curative effect also observed, it was very significant in the statistically. So, however, to design dosage form of hypertension for the patient, this must be proven by different model and clinical trial. Thank you.
Any quick questions, please? No question? So we can move to next presenters.
Next is Durgavira Hanuman from College of Veterinary Science, Rajendra.
Good afternoon. My topic is note-ketone amyloid cerulin-induced chronic pancreatitis through modulation of NF-kappa B or NRF2 pathway in Swiss albuminomyces. Chronic pancreatitis. Chronic pancreatitis is the chronic inflammation of the SNR cells, which leads to the integrity loss of these cells, which mainly results in the destruction of the exocrine functions of the pancreas. There is an increasing prevalence, morbidity and mortality over the last few years. Generally, it is primarily represented by premature activation of the digestive enzymes, inflammatory cell aggregations, pancreatic edema, etc. Cerulin. It is an induced compound. It is 10 amino acid oligopeptide that stimulates the smooth muscle and increases digestive secretions. It has a 2 receptors, which is an analog of cholecystokinin (CCK), which has 2 receptors, CCK1 and CCK2. CCK1 receptor, which mainly stimulates the gastric and biliary and pancreatic secretions and certain smooth muscles. Chemical action of the cerulin. Cerulin, when it acts upon the phospholipase C, which converts the inositol triphosphate, which there is a sufficient release of calcium. It will enter into the mitochondria, and there is a production of the reactive oxygen species, which means to release IKB release. So there is a NF-kappa B activation and transcription the factors which release in the inflammatory cytokines, and also there will be a self digestion of the released from the granule gynogen from trypsin and digestion of the proteins, which results in the increase of the amylase and the lipase. Phytochemical is a note ketone, which is a ketone terpenoid, which is a major component of the grapefruit and belongs to the biggest subclass of sequiterpenes. It is also present in the citrus, crispinogen, and other generic essential oils. It is used mainly in anti-cancer, dyslipidemia, antifibrotic, etc. Results. The cerulin induced in the disease control group, there is a change in the relative pancreatic weight percentage, which is used, which is compared with the body weight of the pancreas by body weight of the animal, which are expressed in the percentage. And serum analysis. There is an increased amount of the amylase and the lipase, which indicates the pancreatic injury, which mainly tells about the cerulin is induced chronic pancreatitis. Now, antioxidants. There is an increase in the oxidative stress and nitrative stress by comparing the MDA levels and nitrate levels, and also decreased in the intercellular reduced GSH and SOD values, and also catalase values, which helps, which means there is a more important prominence of the oxidative stress and nitrate induction. And cytokine profile. Generally, TNF alpha and IL-1 beta and IL-6 are the pro-inflammatory cytokines. When cerulin induced, these pro-inflammatory cytokines are generally increased, and these are significantly increased in diseased animals when compared to normal control animals. And inflammatory cytokine IL-10, which is reduced in the DSE, and it is normal in the normal control animals. Hematology. Total erythrocyte count, it is reduced and hemoglobin is reduced, but total leukocyte is increased in the diseased animals. In histopathology, in diseased animal, there is a congestion of blood vessels, dissolution of the SNR cells, and interstitial space is increased. And also SNR cell space is also increased. In note-ketone low dose and also high dose, the moderate amount is absorbed, but they are recovered from the infection. immunohistochemistry, we observed the pro-inflammatory markers TNF-alpha, NF-kappa B, and COX-2. The expression of TNF-alpha and NF-kappa B and COX-2 are observed in the diseased animals when compared to normal animals. In diseased animals, the expression is more showing the expression of brown color, and it is significantly reduced in the high dose and slightly reduced in the low dose. Conclusion. Nutcaton was found to be possess the ameliorating action against cerulin-induced experimental chronic pancreatitis. The results showed more significant amelioration in the group high dose, which was evident in this study by reducing the pro-inflammatory cytokines and restoration of antioxidant enzymes, possibly by inhibiting the activating of the NF-kappa B pathway. Hence, Nutcaton can be used as a prophylactic strategy for pancreatitis with its overall beneficial effects attributed to anti-inflammatory and antioxidant property. Thank you. Thank you to my major guide also, Kala Kumar sir, who given this opportunity.
Durga, you must get the award for presenting your finishing your talk within the time. Thank you. Any quick question, please?
I cannot hear the question once.
Why you use the cerulin other than the L-arginine, other parameters?
Cerulin is a short inducer within the time, and L-arginine and ethanol will take the long time exposure to induce the chronic pancreatitis. And also when compared to mortality, in cerulin induced it is less, and in L-arginine and ethanol, the mortality is somewhat more when compared to cerulin.
Any quick question, please? One more. No questions? We can move to next presentation.
Last one in this session is by Dr. Biru B. Biru from Department of Pharmaceutical Sciences, Nagpur University.
Scan.
We'll go with Dr. Vikas Dige's presentation next. Just wait.
Good afternoon, all. I am Birudud Bhate from Department of Pharmaceutical Sciences, Nagpur University. Here I am presenting my research topic, neurobiological understanding on anger-like behavior using novel anger paradigm. So anger is a basic emotion, but when it is exceeds, so it converted into the symptoms of several neuropsychiatric disorders such as addiction, ADHD, schizophrenia, PTSD, and mania. So about 20% psychiatric condition associated with this aggression, and several cues associated in society that may generate angers in humans. So this anger is converted into violence, and this is major affecting symptoms in total society. These are some reports I have presented. So there is a crucial need. This is basic emotion, but it exceeds, so it is converted into the one of the pathophysiologic symptoms. So because of that, there is a need to understand this anger-like behavior. For that, we have developed anger-like models. We can easily assess angers in humans, but it cannot happen in rodents model. So for that, some models are aggressive models, aggressive behavior assessment models are available, but they have lacunae like electric shock model, resident intruder model, and maternal separation models. So in that, there is in electric shock, there is a ethically restricted model because of pain, and resident intruder model, it also generates injury to the rat. Maternal separation, it generates anxiety in the litters. Because of that, there is a need of novel animals model. For that, we have developed this anger model. So this is model have two components, means it is separated in it is one chamber separated by one plate, and that one plate attached with a roller, a rotating roller, and in that, and in one side we have kept food, and this rat are fasted for 24 hours, and rats rotating that rollers, and this number of rotation have counted by presenting IR beams. So this indicates struggles, and this indicates reward motivation. In second, we have developed a biting plate, and this roller is replaced with that biting plate. This biting plate developed by two wires metals and separated by insulating materials. When a rat bites to that biting plate, it shows anger behavior. So this biting have counted on by light. Here I have video. Sorry, it is not working. Yes, ma'am. Small. Okay. So by using these anger models, we have generated data. We check heart rate, blood pressure, and in anger-induced model, there is an increase in the heart rate and blood pressure. We check CPOS expression in different brain regions, in that PVT, PVN, ARC, and all brain regions. So there is an increase in CPOS in the 24-hour fasted food in feed sack model, means anger-induced models. So this data we have presented in Journal of Neuroscience Methods. And after that, we move towards the understanding the basic neurobiology. So for that, we cannulated rats in CA, because amygdala is a nucleus responsible for the generation of anger-like behavior. So we bilaterally cannulated this amygdala, and we injected drugs that is a 5-HT agonist and antagonist and fluoxetines in central amygdala. And after that, we check immunohistochemistry of CFOS in specifically subregions of central amygdala, and also collected the samples from central amygdala by using microdialysis, and we have quantified the serotonin by using the HPLC ECG. So these are plan of work. We have fasted animals for 24-hour, and we kept that animals in the anger models. So number of bitings we have checked, and this express anger behaviors. And we injected drugs, that 5-HT agonist, antagonist, and fluoxetine prior to 10 minutes. So we found there is a by treating serotonin agonist, there is a decrease in the anger behaviors, and in specifically amygdala region, there is central amygdala is more expressed CPOS, so it is involved in the anger like behaviors. After that, we check the expression of 5HT1A and 5HT2C in central amygdala, so there is an increase in the CPOS immune reactivity in central amygdala regions, and we quantified the serotonin in anger induced animals. There is a decrease in the serotonin. So these anger models easily used for the neurobiological understanding of anger-like behaviors, and it is used for the screening of anger-like emotions as well as aggressive drugs. So this is the models we have established in our lab.
Any quick questions? Yes, Mike there.
So first of all, this 24-hour fasting, why did you do this 24-hour fasting? In laboratory animal, this 24-hour is actually a long period. And second thing, do you think this behavior was related to anger, or it was related to its urge to get the food after 24 hours?
Yes, sir. Actually, main things is in our papers, we have did experiment with 6 hour, 12 hour, 24 hours, 48 hours. And after that, we check blood glucose level also. Means we found there is no significant reductions in blood glucose in 24 hours animals, and in 24-hour animals showing more aggressive behaviors, numbers of bites. So this data we have presented in our paper, you can see. This is validity things. And second thing is there is 24-hour fasted is not parameters, but condition we have generated means we are prohibiting to rat to reach that food. Rat can see and smell food, but it can't reach. Means this common behavior to get that food, he is continuously biting to that plate, and this is the anger like
short, Dr. Biru.
Yeah.
There is one more question, please, quickly.
Emotion is a complex phenomenon that is always associated with so many other physiological parameters.
Yes, sir.
Which one you can say and relate to the human one in your model?
Yes, sir. Actually, we have directly stated anger-like behaviors, not like completely. We are showing anger. Its main advantages is single animals models. Means we can assess these aggressive behaviors in single animal. Means there is no injury and no anything for that. So we can easily correlate these models for understanding neurobiology and screening of aggressive behavior drugs.
Thank you, Dr. Biru.
Yes, sir. Thank you, sir. Direct.
That ends the award session for the best oral presentation by students. So I have a quick announcement. We'll start the valedictory at exactly 4:30. So there is no dedicated tea break. So those who want can go and have tea while the next session is going on. Not everybody at once, but just plan it. So the next session is oral presentations, non-award category. We have 6 presentations in this. The first one will be by Dr. Anupama Kumari from Institute of Liver and Biliary Sciences.
Dr. Anupama, please.
So the rules are the same for this as well. You'll have 5 minutes for presentation and 1 minute for discussion. Please try to stick to the time given.
So good afternoon, everyone. Thanks organizers for giving me this opportunity. So topic for my presentation is a common bile duct induced mouse model of hepatopulmonary syndrome. So what is hepatopulmonary syndrome? This is a condition which occurs during chronic liver disease. It's a progressive condition which occurs during the chronic liver disease. And this is the most common cause of respiratory insufficiency and dyspnea in case of cirrhotic patient. So what happens when there is a cirrhosis? When there is a cirrhosis, there is an excess fibrous tissue accumulation within the liver, and this leads to the increased in portal pressure, and this condition is called portal hypertension. So this portal hypertension, because of this portal hypertension, what happens? There is an increase in intestinal permeability, and this condition called as leaky gut condition. And in this condition, there is a bacterial translocation, and there is a chronic inflammatory state is there. So this condition leads to increased in release of pro-inflammatory cytokines like TNF-alpha, and also vasoactive mediators like endothelin-1. And these two important mediators cause activation of eNOS pathway and also increased of VEGF. And further, this TNF-alpha leads monocyte activation and release of iNOS. So these eNOS and iNOS is basically increase the nitric oxide production. So because of the nitric oxide, there is a vasodilation. And this VEGF, what it does, it causes the angiogenesis. So within the lungs, there is a formation of intrapulmonary shunting. So these two conditions, vasodilation as well as the intrapulmonary shunting, there is a ventilation perfusion match within the lungs. So overall, this condition leads to low saturation of oxygen within the blood, and dyspnea condition, more lethargy, these kind of condition will be there in the chronic liver disease. So coming to the S1P, actually this sphingosine 1-phosphate, it is a sphingolipid bioactive mediator, and it has role in vascular tone, maintaining the vascular tone via two signaling, this S1P-R1 receptor and S1P-R3 receptor. So through the S1P-R1, basically it cause production of nitric oxide and vasodilation when there is an increased flow. So it maintains the normotension condition. And when there is a low flow, it binds with the S1P-R3 receptor, and it increases the vascular tone by vasoconstriction in vascular smooth muscle cells. So however, in this study, we tested this FTY F70 720 compound, which is an agonist of this S1P, and this specifically works with S1P-R1 receptor. And further, there is a more S1P in the circulation, and in the downstream pathway will be impaired. So there is a less nitric oxide production. So the goal of the study was to investigate the role of sphingosine 1-phosphate in the severity of HPS in cirrhosis patient and its therapeutic implication in CBDL-induced HPS mouse model. So first, we tested our hypothesis in the human study. So we enrolled 3 groups of patients: cirrhosis with HPS, and then cirrhosis without HPS, and healthy control. So these 3 groups of patients enrolled by the clinician. And in this group of patients, we collected the blood, and we measured the S1P level. And further, we also categorized the patient in 3 groups: mild HPS, moderate HPS, and severe HPS, based on their saturation of oxygen in the blood. So when we measured the S1P, we found there is a significant drop in the S1P level, whereas also, and this S1P level was correlated with the severity of HPS. So we further investigated the vasodilatory mediators of HPS, such as eNOS, iNOS, and these were increased in the HPS patient. So further, we did our hypothesis in the animal model of HPS. For this, we ligated the common bile duct in the mouse. And
you are done with your time, Dr. Anupam.
Yeah, 1 minute more I require, please. So We did treatment at this 2 week of stage, and further we euthanized the animal at post 2 week of treatment, and we analyzed the lung permeability and his liver pathology. So in the treated group, you can see there is improvement in the liver pathology, and also you can see here the trans albumin influx is lower. So, and also improved the liver body weight ratio. There is less hemosiderin macrophage was there, and the nitric oxide of bile fluid was also less in case of treated group, whereas the S1P level was higher.
Thank you. Thank you. So we'll move to the
in
next
conclusion, I
presenter.
can say
time is done. No, we have to see there are another 6 presenters of them. No, we are running short of time. Please.
Okay,
Yeah, thank you.
thanks.
Next, we have Dr. Vikas Dige from ICMR National Institute for Research in Reproductive and Child Health.
पूरा ही कर दो उसको, पूरा कर दो उसको. Okay, so it's a tough task to finish the presentation in 5 minutes. So maybe I will just keep some slides. So today we'll be discussing about, I'll be sharing some data about the animal model for endocrine disruption. So over the 2 days, we have heard about all these topics, but nobody has touched the endocrine disruption. It's one of the very important area, which is also been considered as a responsible or maybe the causative agent for non-communicable diseases. And it's also been considered as a One Health program very importantly, because recently we observed that the endocrine disruption leads to any metabolic syndrome diseases and ultimately leads to non-communicable diseases such as obesity, cardiovascular diseases, and diabetes, etc. So exactly what is the endocrine disruption? The disruption is a normal response, like from a circulating hormone. This normally it is almost considered like it is going from normal circulation to the hormone, and this is a normal response. And the endocrine disruptions take the place of the particular hormone receptors and then give the abnormal response. And sometimes they actually mitigate the particular binding, and there is a no response been given. The response has been hampered there. So this particular response has to be assessed in different way, because this particular response of endocrine disruption is especially latency from the exposure. At what time it is been exposed, age of exposure, it's a critical window period also be important. Mixtures of the exposures, the importance of the mixture, that is whether it is given or exposed in combination in isolation, and then also the endocrine disruptors follow the non-traditional dose response curve at the low doses. I just explained it, and then also the transgenerational effects also been modulated and documented very well. And recently, last 3 to 4 years, the endocrine disruptors also been studied for their epigenetic effects and transgeneration effects. So as I was telling, the ADCs and non-response response curve, wherein even at the low doses, it shows some response. At the high doses, they don't show the response. That is a monotonic response. Non-monotonic response, you can say the high dose, there is a response, but in the coming to the slightly bigger dose, there is no response being shown. And again, at the very high dose, you can see the response. So this is the characteristics of most of the endocrine disruptors. That's why there is always an ambiguity at what doses this particular response is being created or been exerted by these endocrine disruptors in the human body. That need to be assessed very well. So as I told you, that these are the key characteristics of the different ADCs. They usually target the receptor ligand as maybe act as an agonist and maybe act as an antagonist. They may be modulate the expression of the receptors. There will be disturbance in the signal transduction. There will be epigenetic alterations. There are some hormone biosynthesis has been synthesized. Then you can say sometimes disturb the hormone transport also. So there are different endocrine disruptors like BPA, PCBs, then the phthalates are also exerting their ways. So I'm going to walk through one or two studies where how exactly the risk assessment of these ADCs needs to be done.
So, already I told you the EDCs and their exposure was very much documented with the wildlife studies, but it's a high time to, and this is the now there is a mandatory to undertake these studies how exactly the EDCs are exerting their effect by phenotypic way or physiologically or maybe behavioral way, and molecular levels also need to be understood so that it will put more light so that the non-communicable disease can be tackled. As I told you, the exposure of ADCs, particularly during the critical window of time, organ development, contributing rising to the multiple disorders like chronic premature female puberty, compromised fertility also there. Obesity is a very important area. Then cardiovascular disease and the disorders of neurodevelopment also been one of the causative agents. So to study this type of behavior or the studies, one has to use the animal models with the, and then as I told, everybody is watching for last 2 days that how animal models are useful. You need to tell more about it. So what we are, I'm just giving example of one of the endocrine disruptor triclosan, which is readily being used everywhere, every day as antimicrobial agents. Most of the time, the all the soaps and the even we are using it in the some cosmetics also there, toothpaste it has been used, and this particular triclosan has been documented because it falls in the environment. So these are the perinatal model we have used. So I'm just coming to the conclusion. So what exactly has happened? See, this is the way we have given this particular compound perinatally to the animal at the age of, I say, in the pregnancy time, and then we followed these particular things from first generation to second generation. There was a delay in the descent. Testicular descent was delayed. There was alteration in the histological condition. There was the pre-spermatogenesis, and then developmental defects were noticed. Same thing we observed for the another molecules like atrazine also it been done, but parabens also it has been done. And so to conclude, what I say is that the animals models are very useful to understand the effects of the endocrine disruptors, and which is a very important area need to be addressed very significantly and very meticulously. And animal models are definitely can be used very successfully.
Thank you.
And we have published all the results, and I'm happy to share that the NTP, the National Toxicology Program, and the ETP also taken the note of our publications, and they all have been included in the policy document of endocrine disruptor prevention.
Thank you, sir. Thank you so much. Thank you.
Thank you.
Move to next presentation.
Next is Dr. Saurabh Chawla from National Institute of Science Education and Research, Bhubaneswar. You have to load it.
Yes. So, good evening, everyone. I've been working with this animal models of wound healing since last 5 to 6 years. So I'll be summarizing my experience of this using this model and how we can refine the ways with which we are using this model. And this presentation will be specifically targeted for this newly budding vets. Animal models of wound healing, it's very commonly used when we study the therapeutic effect of some substances, some chemicals, some reagents, some drugs, which actually accelerate the wound healing. So wound healing, as we know, is a very complex process. Yesterday, Director Madam from NIAB, she also gave some talk regarding wound healing. So it's a complex process. There are different phases which overlap with each other. What we actually see is actually the tip of the iceberg. As a clinician, those who are clinicians, they very well know sometimes wound healing can be very much complicated. And you know, when you have to convince your institutional animal ethics committee, because this is a very pain giving model, a very painful model for the laboratory animals. So sometimes you have to convince your institutional animal ethics committee. So you can elaborate these points why you are doing this study. And why do we need these animal models? Just to give you a small example, every 30 seconds a leg is being amputated due to diabetes somewhere in the world. So that is the importance why do we need to study more and more of these wound healing strategies and come up with a therapeutic regime. Choosing a right animal model, that is a complex task, because what we see is pig is one of the most closest to that of a human being. Skin anatomy of skin of a pig is very much close to that of a human being. And what we usually do is we use this rat and mice model, which have a this muscular layer beneath the skin, panniculus carnosus, which immediately contracts as soon as a wound is inflicted. If you see these animals, they have evolved like that. They live in small spaces. They live in crowded environment, infectious environments. So nature has given them this power. If they don't heal their wound immediately as early as possible, they may die due to sepsis. So we have no other choice but to use this rat and mice due to managemental problems. But how we can overcome this problem of contraction? Because in humans, wound majorly heals by epithelialization and not by contraction. What we do, suturing, thereby the wound heals by contraction when we bring opposed two sides of the wound, as done in clinical cases. So using rodent models worldwide, mice and rat are one of the most commonly used models. That is what I got from a PubMed search. And there is difference between human and pigs, human and mice skin, but somehow we have to have a model which can studies on which can be later translated into human beings. So we can use different models which can be translated to different kind of human conditions. We can use sepsis model, we can use diabetic model, DBDB mice can be used. In case of immunocompromised individuals also, wound healing is a big problem, so immunodeficient mice can be used. And in aging humans also, this wound healing is a big problem, so aged mice can be used to actually as a model for wound healing. And here I'll show you my tell you my experiences. So this presentation is about refining this animal models. So sometimes small things, they have a big implication, like shaving. If you see shaving, it leads to more of skin complications. So if you are shaving a mice, it may lead to skin complication, increased bacterial load. Rather, you should use a trimmer. And how do you find a good trimmer? You go to a market and you search, you will find a good trimmer which gives you a close shave. So that also sometimes makes a big impact. And choosing a right anesthesia, when you inflict a wound, using xylazine ketamine is one option, but xylazine ketamine leads to increased hepatic circulation of hepatic enzymes. So you have to do it continuously. So it's better always to use isoflurane, which have a very less biotransformation. And that is how we create a wound. I made a small video. I think so, that video, I don't know if we can play
Saurabh,
that
your
video.
time is up, please.
Okay, thank you.
sir.
So I made a small 50-second video. I think so. Okay, so it cannot be played now. So thank you very much.
Thank you, Saurabh.
Next, we have Dr. Pratap Reddy from ICMR NIN, National Institute of Nutrition. 77.
Myself, Dr. Pratap, I am a technical officer at NIN. Today, I will be discussing about my work, which shows obesity associated with pre-diabetes increases the risk of breast cancer development. It's an animal study using an obese rat available at NIN. So coming to the introduction of obesity, I think after Chaudhary sir presentation, everybody has know it's a tripling from 1975. It's tripled And most importantly, along with the other than the obesity, there was increased cases of diabetes. Apart from diabetes also, there is increased cases of impaired glucose tolerance, which are considered as a pre-diabetic. And the chances of pre-diabetic becoming diabetic is 3 to 10 folds when compared to the normal persons. And the other problem is breast cancer. The prevalence of breast cancer is the most, every year 11.7% of new cases were reported, and it's the most cause for the deaths in the women worldwide. And there were several reports that shows obesity or diabetes or pre-diabetes is associated with increased risk of breast cancer. All are clinical studies, but there is not, and there is only one study which shows the underlying mechanism, molecular mechanism between obesity or pre-diabetes, which increases the breast cancer. So we are interested to see what is the underlying mechanism that links the obesity or pre-diabetes with impaired glucose tolerance and increase of the breast cancer. For that, we had chosen a rat which is available with us. It's a WN9-GROB rat. It's an obese rat with impaired glucose tolerance and hyperlipidemic condition. And this, it has also, we have the counter lean parts of this rat also. Coming to the experimental part, so in order to know that, we had chosen both obese rats and lean rats, and we had given DMB at the rate of 40 mg per kg body weight to induce breast mammary cancer in the animal, and followed by to know the mechanistic details of the tumor parameters like incidence percentage, latency period, and HND and IHC, and even Western blots were done for this experimentation. And coming to this, this image shows that the development of breast cancer in the lean rats, and this image shows the breast cancer development in the obese rat. And coming to the incidence percentage, the onset of breast cancer in obese animal is much earlier when compared to the lean rats. It's the obese animals has developed breast cancer somewhere in the, it started from 9th week onwards after administration of DMBA, whereas it took 26 weeks to develop in the case of lean rats, and 62% of the animals had got breast cancer in obese rats, whereas it was only 22% in the lean rats. And not only this, coming to even the latency period, the average period that taken for the animals to develop breast cancer was almost double in the lean rats, whereas in obese rats it's 119 days only. And even the number of tumors when compared to the lean rats, it was higher in obese rats. And further, it was confirmed with the histology, HND staining. And most of the studies has given a clues that in case of obesity or pre-diabetes or diabetes, there will be an alteration in the IGF, insulin growth factor 1, which ultimately leads to some activation of cancer pathways. So we are more interested in that. So we want to see what will happen to the IGF-1 and subsequent pathways that activated by the IGF-1. In that case, we are more interested in PI3K pathway. So the signaling molecules majorly involved in IGF-mediated PI3-AKT pathway, which leads to cancer in general, has been studied by using IGF. And it was showed that IGF levels and the subsequent pathway molecules like phospho-IRS and PI3 and phospho-AKT were higher in obese rats when compared to the lean tumor also. And not only that, this activation of AKT further leads to inactivation of GSK and activation of phospho-GSK. This active state of GSK will regulate the beta-catenin, whereas inactive state cannot regulate the beta-catenin. As a result, beta-catenin levels will be increased in the cell, and subsequently it will be transported to the nucleus, and it further beta-catenin has an ability to activate the transcription factors. So further, the studies also confirm, Western blots also confirm the similar results. And finally, coming to the conclusion, so whenever there is a alteration or sometimes elevated levels of IGF, it activates the IRS phosphate, and further it activates the PI3K and activate the AKT, and AKT leads to phosphorylation of serine GSK3, which leads to inactivation of GSK3, and which cannot bind to the beta-catenin. As a result, beta-catenin accumulation occurs, and beta-catenin further.
Dr. Pratap, your time is up.
Yes,
Please
I'm
conclude.
done. I am done. So most of my results were my conclusions. So basic point is this model can be used for the obesity associated breast cancer studies. And not only that, it clearly shows that AKT activates the PI3 AKT pathway GSK signaling, and through that it's it's developing early breast cancer in the obese rats when compared to the lean rats. That's all. Thank you. Thank you very much.
Thank you so
I would
much.
like to thank director and
Next presentation, please.
Next, we have Dr. P. Krishnamurthy from ICR Nivedi.
Dr.
Nine.
Krishnamurthy,
9.
online. Move on to next.
Okay, we'll go for the last presenter then, Dr. Sriharsh from Novartis
Sriharsh.
Healthcare. 23.
Good evening to all of you. I am presenting this is the collaborative work from Innovative Medicine Innovations. This is a public-private collaborative work which is going on in Europe. It's a 5-year project, and a part of the project is to develop predictive model for predicting the drug transfer to infants through mother's milk. So, introduction, you all know that breastfeeding is important, but when a lactating mother needs medication, there is a compromise she has to go through. That is, either she has to avoid breastfeeding, she has to avoid medication, or she has to look for the safest alternative for the condition where she requires medication. So why this is like this? Because the current regulatory setup from the preclinical side and the clinical side, the information what we have before the drug goes into market is not sufficient to prove the safety of the drug for pregnant and lactating. This was also a point which our keynote speaker made. So from preclinical, we just have PPND studies, and it is mostly done in rats. When required, it is done in other species, like for large molecules or something like that. And in clinical, if you see, mostly inclusion of women of childbearing potential is avoided because we don't have preclinical data on that. And the information which goes into the prescriber label that comes from the post-marketing surveillance data. So there is a clear gap, and the gap is because you don't have a proper, a model, animal model to predict safety for pregnant and lactating women. So with this background, the conception was formed. This is with the intention to disseminate reliable evidence-based information regarding effect of medication used during pregnancy and breastfeeding to women and the healthcare professionals. How we achieve this? This will be achieved by generating, cataloging, linking, collecting, and analyzing data from pharmacovigilance, modeling, routine healthcare, breast milk samples from the large network or the biobanks. Among this consortium, there are 8 working parties or working groups, and work package 3 is related with non-clinical model development, in which we are trying to develop, characterize, validate and apply a non-clinical platform, not only model, the entire platform for a reliable prediction. So basically the intention is to use cell culture models, animal models, along with the information what we are getting from human, from the normal population, to combine them and have a PBPK model. I'll go to the details in next. For the animal models, what we did, we looked into the current animal models which we have, that is mice, rat, rabbit, dog, pig, and NHPs. So we compared physiological factors, milk composition, anatomy of mammary gland, and feasibility of data collection and milk collection. Also ethics related to these models. So physiological consideration, we looked into the process of lactation, which is typically secretory differentiation, secretory activation, and continuous lactation. These are the 3 processes. And when we look into the physiology of it, the physiology was mostly conserved among the different species. There were no much differences, but large difference was there in the duration of lactation in different species, ranging from 3 weeks to 1 year. So considering this and the hormonal variation was one of the things which we considered in physiological factor. In milk composition, if you see, human were very much closely related to NHPs, but next better model was pig or mini pig. Then we also compared the other criteria which I mentioned. Here I'm showing the cartoon of human mammary nipple, and it has multiple ducts opening, that is from 10 to 15. Now you compare with the other animals like mice, rat, rabbits, there is a difference in anatomy as well as collection of milk. Considering all this, pig was considered as a better model. Then we also looked into data from transporters. If you see, this is just a representative data where we compared, and there are different transporters. Again, the population of the transporter, expression, everything differs. This is just giving for the idea. So in conclusion, we are trying to have a, right now we are trying
you
to,
are done with your time. Please
so this is
continue.
the conclusion where we want to use cell culture with PBPK model and animal models to predict in human how the milk drug is transferred through to infants. Thank you. So these are the 47 scientists who are involved in this, activity. Thank you.
So I'll call again for Dr. P. Krishnamurthy, ICR Nivedi, if he's here. No. So that ends the session for oral presentations under non-award category. So we'll proceed to, I request Dr. Satyavani to initiate the valedictory ceremony in a couple of minutes. We'll start it in a couple of minutes.
What is the sequence of seating? Sequence है? नहीं ये लगाना है ना, वो पता है दूसरा
Ashok. You made those slides for those award winners. You're playing from there? Okay, then this is. ఇది దగ్గర పట్టుకో లాస్ట్ ఎవరెవరికి ఏమి ఇవ్వాలి ఆర్డర్
నాకు ఫోన్ దొరికింది ఇక్కడ మళ్ళీ అడగలేదు. ఆ పిల్ల సెల్ఫీలు దిగే దానిలో ఉంది కనీసం
వదిలే రామచంద్ర సార్
వాళ్ళు అనౌన్స్ చేస్తారు సర్టిఫికేట్ మీద తర్వాత ఇస్తాము డబ్బులు ఇస్తారు సర్టిఫికేట్ ఇది నాది అంటే నాకు సంబంధం లేదు.
అందరికి ఇవ్వాల్సింది నాన్ లాసా మెంబర్స్ కి డబ్బులు ఇవ్వరు
ఇస్తారు మరి ఆయన డబ్బులు ఇవ్వని చెప్పిందా
డబ్బులు
ఇవ్వమని చెప్పారా? చెప్పారా?
కొంచెం అనౌన్స్ చేసినప్పుడు వచ్చి క్రౌడ్ అయిపోతే జాగ్రత్త
ఇంకా 5 నిమిషాల్లో పోతాం. ఎవరెవరు నాకు తెలియదు.
अब ठीक है।
Good evening and warm welcome for this valedictory function. Dignitaries and participants, we have come to this last session of valedictory session. After having many more presentations by eminent scientists, young scientists, students, and many more deliberations on the occasion of animal models of One Health program and challenges and future perspectives of this conference on LASA conference. Now I request our chief guest, Dr. S.K. Datta sir, on this occasion, Member Secretary CPCSA, to come on to the dais. Dr. S.K. Datta sir from CPCSA. May I request Dr. S.G. Ramachandra, Director, North BR, to come on to the dais. Dr. Arvind Ingle, President of LASA, requested to come on to the dais. I request Dr. Gerald Mahesh, Co-Organizing Secretary of LASA 2022, and Dr. Jayant Hole, Co-Organizing Secretary LASA 2022, and Dr. Irfan Ahmed, Mir, Treasurer of this LASA 2022, onto the dais. Now I request Co-Organizing Secretary of LASA, Dr. Gerald Mahesh, to report the 10th International Conference of LASA 2022.
Friends, we have gathered here for the valedictory function. A nice memory we are taking away, but just to remind you that two of our very eminent scientists, one a person who was like a very father figure, Dr. S.N. Naik, from Cancer Research Institute, where the institute started, passed away last year, and another Dr. K.R. Bhardwaj, who was also founder of the Laboratory Animal Science Association of India, PDRI. So in their memory, I request everyone to raise and silence of 1 minute is requested. Thank you.
Good evening, friends. Good evening, friends. I want to update them the LASA conference activity of last 3 days. First day free conference workshop, second day main conference, 2 days, last 3 days exhausted with many lectures. But what is the real status of this conference status that need to be announced? What is the status? As we are displaying here, you can see them. This is the status. There's totally 622 people registered both online and offline. And what you are seeing there, 15 pending registration. That means we are our colleague people, they are helping us. We are unable to register them. We are keeping as a pending registration. Okay. And And another is 190 people, the pre-conference workshop offline they have participated. They have 190 people, they came here registered for offline participation and 427 for main conference offline mode. And total 88 abstracts are approved. That means, and again 20 abstracts are rejected. This is the conditions. And among this, we have best oral presentation is there, best poster presentation is there, even oral also. We are dividing them two category. One is LASA member category, another one is student award. And we are again poster also, we are dividing them best poster presentation and best e-poster presentation. All these awards are, we have carried out exhaustively and very transparent manner. In case of every judges have 3 judges, they are watching inside them, they are giving the marks, we are accumulating them. Which one is higher, we are selecting them in this way. In this way, we are going to announce the result period to that. This is the status. And coming to these recommendations of LASACON, so I probably announced that Indian College of Laboratory Animal Medicine is established on par with ECLAM, IECLAM, and JECLAM. The second one is LASA EC has decided and nominated Dr. Ramachandra, Director NRPR, as a returning officer to elect new LASA body. And third recommendation is LASA EC body has nominated Dr. Nagarajan as editor for Indian Journal of Lab Animal. Thank you.
Thank you, sir. Updating the report of 10th International Conference of LASA 2022. It's really success of this many number of registrations, so successful completion and organization of this conference. We are very much happy to announce this once again. Thank you once again. Now I request our chief guest of this function, valedictory function, to address the gathering. Dr. S.K. Datta, Member Secretary, CPCSA. So.
President LASA, Dr. Ingle, and our CPCSA committee member and director NARBAR, Dr. S.C. Ramachandran, and other dignitaries. I will be very short because I don't want to give lecture on this Caucasian because I feel myself amongst you. I am not beyond you, but we are a co-worker. And we are working for the development of the science and establishment of the scientific ethos in our countries. So that's why I feel proud to be here in the LASA conference. Last time I came in Pune, and because of the Corona, we could not arrange this program. And when I came to Pune, I committed that I will my CPCSA will contribute to this program because I spoke with Dr. Ingle and we have done it. So we are the CPCSA is the partner of this this program, and we feel proud of having this within the scientific community. I was listening some of the very good presentations, this young scientist presentations, and really it's a very good innovative kind of ideas that exactly needed for our country, because you know this there are researches, enormous researches going on in all over the world, and now India is really going ahead in the world, and India we think this India should go beyond this beyond the India should be great in the scientific research, and India is now going this, our children and our scientists are really working great work right from the space technology and biological sciences technology everywhere in the world. So the scientific ethos and scientific knowledges, it should prevail in our country. We should not go for the pseudoscience, whatever is going, that is this politicians' mandate. So we should concentrate on our science for, and science always prevails. I always believe that if a wind is blowing, it is a science. My daughter is studying this astrophysics and says, the Papa, nothing is in the universe and nothing is in the environment. It's go without science. Everything is science. Everywhere is science. Once you invent and how do you explain this scientific knowledge in your records? Now with this, when this CPCACS subject was came to this our animal husbandry department, then there was lot of complaints about CPCCA. These protocols, these are not getting approved, and it was lying pending for long period of time. And we started working towards this. What we can do, because I am not that much of learned people. That's what we can facilitate you to fast track all these applications and whatever your requirement is from the regulatory point of view. Then we started working, and really we have entirely modified our CPCSA working systems. Now we are going to notify the time frame of all the activities of the CPCCA. For this, we have already published the draft administrative rules, although some of the peoples, they have objection on that. So we'll do it because we have a commitment to the highest office in our country, and everybody is monitoring. Now, what is happening is we are getting appreciation from our highest office because we have modified all our working system. You know, these protocols are getting approved in a time-bound manner, and every month we are clearing all these protocols. So whatever studies, still there are some reforms needs to be done. And now we have taken a task of the two major initiative. One, Dr. Ramchandra has taken. So we want to create a database from the animal model studies so that no duplication cannot be done. Actually, if we stop the duplication of the animal studies, then actually really we can do for the animal welfare, because number of animals sacrificed and number of animal used in the experiments, so that will come down. Another thing we have taken that we have a demand from this, I am looking after the animal welfare board also, because I have to listen this others side also. They have a demand that CPCCA is doing a wonderful job, but the CPCCA should maintain the database on the non-animal experimentation wherever it is available. So I was discussing with Dr. Ramchandra and with all your people, we try to maintain one database, because I was listening is very good presentation on the non-animals methodology available to reduce the animal experimentation. So we'll maintain that database also. So these are some of the activities, and I don't think this I am beyond you. I don't consider myself as a big officer and like that. So I am a mere veterinarian, and so always I am available. And if you people are facing any problem, I am here and my team is here. And if you are facing any kind of difficulties for animal welfare, animal house registration, renewal, everything, we have a time frame, and we have now fixed office. So now CPCSA has his own office in DMS complex of New Delhi. So always we are available, and this online portals, it is now getting integrated with the buyer portals. You might be might have heard this. Government of India is developing one portal that is called buyer for submitting application and in a single window systems. Although this regulatory requirement will be there for that. So we are also revamping our website and portal, and will be integrated with the buyer portals. So these are some of the things, and I congratulate LASA for organizing such a wonderful worldwide conference here, and I will be participating till I continue with these assignments. Thank you very much, LASA, for inviting me here, and thank you all the delegates for being here, and thank you all for, thank you.
Thank you, sir, for your valuable valedictory remarks. Also, to enhance the need of concentration of science and reforms to be done in the field of animal sciences. Once again, thank you very much on behalf of organizing committee. Thank you, sir. Now I request Dr. Mahesh to declare the oral presentation award LASA member category 2022.
Yeah. So best oral presentation award under LASA member category. The first prize goes to Dr. Aruli Jori, NCPS, Bangalore.
Certificate.
Coming. Don't worry.
Purpose of the present, sir.
Give you no problem. Listen to me. Organize first properly. One by one. Yeah, second prize goes to Dr. Sachin Singh. Yeah. It's online. The third prize goes to Santi Bhushan Senadipati. He is from Institute of Life Science, Bhubaneswar. He participated online. It's very well presentations. He will get the certificate digitally. He can download them. First thing. Second thing, will award money will be transferred to electronically.
Thank you. Thank you very much. That's the Indian Gazette. Thank you.
I will request Dr. Jain to declare Best Oral Presentation Award Student Category of LASA 2022.
Thank you, Satyavani Madam. I request our chief guest, Dr. SK Sir, to present this award. Any guess for Best Oral Presentation Award Student Category? Any guess? So first presentation goes to Dipali Rawat, NIB Hyderabad. ठीक है. Dr. Ramachandra sir also will associate with Dr. SK sir. Dipali Rawat, please. Yeah, thank you. I request our chief guest and Dr. Ramachandra sir will present the award. Thank you so much. Thank you, Dipali, and congratulations for your future endeavors. Good luck. Any guess for second prize? Oh, sorry, I have not seen. Yeah, the second prize, Dr. Donga Durga Veera Hanuman, College of Veterinary Science, Rajendra Nagar. Please come on the dais, receive your prize. I request Dr. SK sir and Dr. Ramachandra sir to present the award. Congratulations, Dr. Donga Durga Veera Hanuman, and best luck for your future career endeavors. Third prize goes to Dr. Biru B. Dudabhate, RT MNU, Nagpur University. I request you to come on the dais and receive your award. I request Dr. Ramchandra sir and Dr. SK sir to present the award. Congratulations to the winners and good luck to the all participants. Thank you very much.
Now I request Dr. Arvind Ingle, President LASA, to declare the Poster Presentation Award 2022, LASA 2022. Please, sir.
Yes, yes. I will not ask you to judge because it may be displayed here and it will be like you will be telling me who is the first prize. First prize goes to Gaurav Kansagara. The award. No, you should be telling this one. I should be presenting the award. Okay, no problem. Yeah, do it. No problem. I request Member Secretary to present.
You want to speak for a minute? Yeah, please.
Hello, everyone. Sorry if I'm too loud because I'm very excited here. First of all, I would like to thank the organizers for putting together an awesome conference and poster judges for the critical feedback. And I would like to dedicate this award to the laboratory animals that I have used to support my studies, as well as the National NCBS and INSTEM SCRC team, Animal Care and Resource Center. We get very few opportunities to acknowledge the support we receive from them. And I will take this opportunity to say that you guys are doing an amazing job. Thank you so much.
Second prize, Inderjeet. I request Member Secretary, please confer the award. Third prize goes to Arpana. NCBS Bangalore. Please.
Hello. Sorry, I will not take too much of your time, but I would like to dedicate this award to Lily for doing the initial works, and then to Dr. Mahesh Sahare, who has always encouraged me that I have the capability to do this. Our studies are still in initial stages, but this prize is a huge encouragement to go forward, and hopefully we'll be able to succeed in what we have started. Thank you so much.
Now I request Dr. Irfan Ahmed Mir from North BR to announce Best Tea Poster Award declaration.
Hello, everyone. Good evening. Thank you for showering your love for this conference. Let's not forget, this conference is a hybrid conference. There are many researchers who have presented their work through online mode. So I'm here to announce the Best e-Poster Award category awardees. So the best award for the first prize goes to Dr. Yogita Pawar. She's from Radiation Medicine Center, Baba Atomic Research Center. The second prize goes to Dr. Nanda from Kasturba Medical College, Manipal Academy. The third prize goes to Dr. Anson S. Maroki from Sri Datta Institute of Pharmacy. All these awardees will get their certification online, and their prize award will be remunerated through electronically. Please give a huge round of applause for all the awardees. I congratulate them on behalf of LASA. Thank you for your contribution to the category, online poster category.
Thank you, Dr. Irfan. Congratulations to all the winners, award winners. Once again from organizing committee, we congratulations, all the best to you in your future and yours. Now, this is the time to present the mementos of our sponsors by Dr. S.K. Datta Saab, Member Secretary, CPCSC. Now, Dr. Thomas will come and announce the sponsor to receive the mementos. They're the supporters to conduct this conference. Please, Dr. Thomas will announce their names.
Good evening, everybody. The sponsors always have a special place in our heart. So we'll be applauding them and giving them the most desired respect for supporting us. To begin with, the first, we'll be taking through the different levels of sponsorship. First, we'll be starting with the platinum levels. We have in the beginning itself the Committee for the Purpose of Control and Supervision of Experiment on Animals. So CPCCA.
Yes. Then you call.
Thank you, Dr. Vivek Tyagi. The next is Hylascope Biotechnology. Dr. Balaji. Okay. Orchid Scientific and Innovation India Private Limited. Vivo Biotech.
We announce one more to be ready. Citizen Industries.
Citizen Industries. They will pass it around here.
No, at least they'll be ready. Just make announcement, be ready.
MS
Ask them whether they are coming or not. Vivo?
Vivo Biotech. Sorry.
Okay.
Announce loudly.
Citizen Industries,
There.
Palamur Biosciences. Ready रहता ना, वही रहता ना.
Citizen Industries.
Mark that. No, he is not allowed. No, he is giving instructions. It's not correct.
Palamur Biosciences.
Next.
दूसरा बोल के रखिए वो विल बी रेडी बहुत लंबा बट वो टाइम लगता है ना आने को किसके नाम पता नहीं है बोलो
AT&T Laboratories in collaboration with Jackson Laboratories. AT&T Laboratories.
Be ready, buddy.
It's coming. Satish is here. Vishnu Traders.
Again, they're getting the dose because of this.
Samitech Instruments. Techniplast. Collaborating with Techniplast. The first in the diamond category, Livion Biolabs Private Limited. Livion Biolabs.
Will go.
No, please. Sai Life Sciences. The gold category, first one is VRK solution, VRK nutritional solutions. VRK nutritional solutions. Agile Life Sciences Private Limited. Advinas. Agile Life Sciences Private Limited, Advinas. Integrated Clean Room Technologies Private Limited, iClean. Vimta Labs. Vimta Labs. Perkin Elmer. Perkin Elmer. Research Diet Biotechnology Labs. In the silver category, the first name is Animal Care Systems. BIK Industries, because GV Research Platform. GV Research Platform. Christian Biosystem. Christian Biosystem. Nomura Siam International Co. Private Limited. So that's all with the offline sponsorship. I'll just read out the online sponsors list. In the platinum category, we have Heterodrugs Private Limited, Virtuo Biotech Private Limited, Biologically Limited, Bharat Biotech, RCC Laboratories India Private Limited, iClass. In the diamond category, we have Natco Pharma Limited. In the gold, online, we have Vince Biotech, Sri Ananda Padmanabha Private Limited. And in the silver category, we have United Healthcare and Life Sciences Private Limited, Nectar Therapeutics, Krish Biotech, Labanims, Advitech Technologies Private Limited, and Elegard. Thank you.
Thank you, Dr. Thomas. Now this is the time to felicitation of the chief guest of this evening. Now I request Dr. Ramachandra sir to honor our chief guest with the shawl, and Dr. Arvind Inglesa with the memento, and all the Dr. Mahesh and Dr. Jayant with the flower bouquet.
Janaganamana.
I request Dr. Jaral Mahesh, Organizing Secretary, LASA 2022, to present vote of thanks.
Good evening to the Honorable Chief Guest, distinguished guests, speakers, delegates, and sponsors. On behalf of LASA, NIAB, NARBR, it is my honor and privilege to propose vote of thanks on this occasion of concluding the LASA conference on 2022. At the outset, I would like to thank Chairman CPCACA and Member Secretary CPCACA and expert consultant who have come forward to lead the conference. My heartfelt thanks to Dr. Krishna Ellagaru, CABD, Parathabaitak International Limited, and icon of our LASA conference 2022 for gracing this occasion despite his busy schedule. My sincere thanks to Director and Joint Director, NAM, for extending support of all fronts, providing this auditorium, conference hall, accommodation, dining space, exhibition space, logistic, and all other administrative support. The entire technical team for NAM, electrical, IT, horticulture, and housekeeping have worked day and night over the last 3 days to make this event successful. My sincere thanks to Dr. Taru Sharma, Director NIAB, and Dr. Ramachandra, Director NARBR, for the wonderful mentoring and for the extending support with finance, logistic, and manpower. They were readily available whenever we approached for their guidance. My special thanks to Dr. Harishankar, Dr. Subhi, and Nagarajan for his wonderful guidances. My gratitude to all international and Indian speakers, chairperson and co-chairperson of all scientific session, participant in oral and poster session. Their contribution efforts are what made this vibrant scientific event. My sincere thanks to all delegates, participants of both offline and online events from various organizations, without whose enthusiastic participation, it would not have been this successful. Their participation holds great significance considering the big challenges of conducting a large event post-pandemic. We thank profusely the generous contribution and participation of all the sponsors for the events. My sincere appreciation to all the organizing committee members from various committees of this LASA conference. Everybody know that so many committees whose support we could not make such a massive event so smoothly and productively. With the permission of the chair, I announce the completion of the LASA conference 2022. Thank you.
Now I request all of you to stand for national anthem.
जनगण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे
Hello, all are requested to come on now for group photograph. Okay. Group photographs. Naam.
Thank you for coming to Hyderabad and supporting this conference. Thanks all.
Everyone, just one quick round of applause to Jayant and Mahesh. I think the organizers forgot to mention their names, and we really appreciate their untiring work over the last few months. Wherever you are, Jayant, Mahesh.
ये